merciful, Master of the day of requital. You do we serve, and you do we beseech for help. Guide us on the right path, the path of those upon whom you have bestowed favors, not those upon whom wrath is brought down, not those who go astray. Amen.
Thank you, Ms. Afrin. Now we will start the second technical session with a plenary lecture followed by paper presentation. I request Dr. Fauzia Ahmed, Assistant Professor, Postgraduate and Research Department of Zoology, to introduce the resource person and take up the session.
Assalamu alaikum and a very good morning to one and all present here. On behalf of the management, principal, staff and students of PG and Research Department of Zoology, I deem it a proud privilege to introduce the resource person of the day, Dr. Rajesh Lakshmanan, Biocompatibility Specialist, Cook Ireland Limited, National Technology Park, Limerick, Ireland. Dr. Lakshmanan is a doctorate in medical nanotechnology from Sastra University, Tanjavur. He has a rich research experience of 6 years at various national and international levels. In his career so far, he has received many honors and recognitions like the Young Scientist Award, DST Inspire Faculty Award, Young Investigator Award from Colorado Convention Center. He has a teaching experience of 7 years for PG and UG students. He is a life member of many societies like Indian National Congress, Society of Biomaterial Science, and Society of Tissue Engineering. I wholeheartedly welcome you, sir, to deliver the plenary lecture on the topic Tissue Engineering Strategy for Cardiac and Bone Regeneration through the virtual mode. Once again, I welcome Dr. Lakshmanan to deliver the plenary lecture. Thank you.
Thank you, ma'am.
Thank you very much. Can someone listen?
Yes, sir.
Students who have gathered over here on this fantastic morning. So without delay, I'll jump into the slides that we are going to discuss today on tissue engineering strategies for cardiac and bone regeneration. So before delving in, as you were acknowledging the Almighty, I would like to acknowledge all my research supervisors who have been providing me this platform for who I am here today. So first one would be from the Sastra University, where I had a great pleasure to work under the guidance of Dr. Swaminathan and K. Uma Maheshwari, where my PhD days were spent, followed by Yukon Health Center, where I had my postdoctoral experience gathered. And finally, another set of postdoctoral experience gathered at Trinity College Dublin. So most of the research slides that I'll be showing up will be from these institutions. So I'll directly jump into the tissue engineering strategies the main core that we will about to be discussing today. So those of you who were aware of tissue engineering before, excuse me, this is going to be like a overall introduction to the tissue engineering. But those who are not aware of, this would be very useful. So tissue engineering comes in as a modern science advancement that combines the principles of engineering and the core biological sciences. So this picture or this image in what you see here in the slide. So this is the maybe called a mother slide for all the tissue engineering what we are here today. So this concept was proposed by Vacanti way back in 1992 by Robert Langer and Joseph Vacanti when they were in Massachusetts Institute of Technology. So they just gave a rough idea of what would be like tissue engineering, like the triad of tissue engineering they call. First one would be the scaffolds. The second one would be the signaling molecules, and third one would be the cells. So these are called the tissue engineering triad. You know, so later on, guys started to working upon, and after 2 to 3 decades of extensive research, so this is where we are in this. Each and every single the triad becomes its own parent that leads to a really selected advances in the field of tissue engineering. So with respect to the cells, we have an advancement in iPSCs and stem cells and genetically modified cells. And with respect to the engineering strategies, we have more fascinating technologies coming through. Example, the 3D printing, which is like a state of the art as of now. And other engineering principles would be like getting into the signaling molecules like the growth factors and all the other therapeutic agents that can be delivered through this tissue engineering approach, which we'll be seeing quite a bit. So this is an overview of what tissue engineering is and where we are right now, right from the beginning to the end. And from here, we will be dwelling directly into the core concept of today's discussion, that is on cardiac repair and regeneration. So I'll just give a quick overview of what is a need for a tissue engineering approach to be in the field of cardiac failure. So in this slide, it's quite obvious. Hope you guys are aware what's heart failure. That's a lethal condition where a patient's left ventricle doesn't get enough blood supply due to the blockage of artery. So uh, so this is a fatal condition where the left ventricle doesn't get enough of oxygen. So what is the output of this would be the left ventricular muscle will gradually thinner, and the scar formation will be happening, which will eventually lead to thinning of the left ventricular tissue, and thereby leading to aneurysm, and that finally we call it as heart failure. So this condition is something very, very serious where we don't have an actual therapeutic treatment at the end stage failure. So that's where the tissue engineering comes in. Like, how can we come up with a novel ideas to treat this condition? So one such approach would be if you look into the molecular aspect of the heart failure here in this slide, you could see the myocardial infarction or the infarct site. That's what we call it as hypoxia. That is the lack of oxygen due to the lack of blood supply. There is no supply of nutrients as well as oxygen. So the orientation of the matrix, extracellular matrix, the collagen matrix seems to be different with respect to the disease condition compared to the normal condition. And in the normal condition, you get to see a randomly oriented fibers, whereas in the diseased condition, you get to see a more aligned and things are similar to the scar tissue, what we call in any of the tissue. So this is the change or the alteration happened during the ischemic condition. And what is the tissue engineering has it? So through the technology, nanotechnology advancement, we have the technology to get these nanofibrils through our laboratory setup. That's called an electrospinning setup, which is like a customized during our during our race. So this is one of those scanning electron microscopic images what you guys are seeing there in the slides. So the right image, which is output of the scanning output of the electrospun nanofibers. So this would exactly or partially mimic the native extracellular matrix tissue of the cardiac normal cardiac tissue. Moving on to further advancement, like what kind of polymers that can be chosen for this extracellular matrix preparation. So in research, wide range of polymers have been developed upon, like where they have the flexible nature similar to the extracellular matrix or the flexibility of the heart. From there, we tend to get a polymer, which is like a combination of two different primary or the polymers, individual polymers. It's the combination of lactic acid and the caprolactone. We call that as PLCL at an equal ratio, 50-50, and also in different ratios in the slides I'll be discussing upon. So we get to play around, have a chance to play around with the tunability of their mechanical properties as well as their for all the other biocompatibility aspects of it. So which I'll be showing up in the next slides. And in the parallelly, the signaling molecules when we are talking about the triad of tissue engineering. So one would be the extracellular matrix. The second would be the signaling molecules. So related to the signaling molecules, we try to pick up what would be the best angiogenic molecule that will be very efficient in bringing back the vascularization into the scar tissue, where the actual cardiomyocytes are dying. So we get to see wide range of growth factors have been developed upon, and onto which we have choose specifically two different growth factors there. One is the vascular endothelial growth factor and the basic fibroblast growth factor. So now I'll jump into the actual research output that that was that was done during my PhD days. So in the first half of the slide, like where you can see this was an output of an electrospun nanofibers, an electrospinning technology where the fibers are prepared with a different combination of the copolymer ratio, where the first one or the A panel shows about just the polylactate caprolactone, and the second one was with another polymer. We call it as a polyethylene oxazolane. So that's another polymer which we try to bring in up in order to bring in some of the hydrophilic growth factors into the matrix. So that was mixed at two different ratios. One is 7:3, and another one would be like 5:5. So two different ratios. We check their feasibility for electrospinning and also to load the growth factor molecules. And this is some of the other characterization studies that to show two different polymeric systems exist at the scaffold what we have prepared. And some of the other characterization includes the mechanical studies. As I was telling you, PLCL is quite flexible, and by the combination of polyethylene oxazolane into it, the elasticity can be much more improvised, but not to certain extent. Like there's a threshold where you can stop and you have to switch back, because at 5:5 where the properties are getting deteriorated. So 7:3 seems to be having a better elastic property compared to the PLCL, similar to the other properties. And this is some of the images of the cells, like the H9C2 is one of the cardiac cell line that usually will be first line of biocompatibility assessment testing will be done with these cells because the actual cardiomyocytes are quite fragile to handle, and also their adherence onto the scaffold is very challenging. So people go with any established cell lines initially to check the feasibility of these materials onto the cardiac environment, and that's where we try to work on with H9C2 cells, where we could see the cells were adhering onto the scaffolds fine, and then they try to proliferate nice. So on the first image where you could see the PLCL, that's the polymer matrix where the cells are much more having the staining, and they tend to expand their morphology, whereas in the B73 they tend to form their actin networks, like that's a characteristic morphology of the cardiac cells, followed by the B55. So with this preliminary assessment, we tend to know that the combination of different polymers doesn't affect the biocompatibility of the cells, and it also helps in the promoting the cell viability and cell proliferation. Um, the next step would be loading some good angiogenic molecules. As I was telling, we picked up two molecules. One is the VEGF, another one would be the FGF. At a certain concentration, we try to deliver it along during the electrospinning condition, and we tend to see its release profile, how these growth factors come out of the scaffold during the course of time. And this shows to be sustained, which is what we need in terms of a tissue engineering approach. Once you place the scaffold into the heart tissue, the growth factors or the therapeutic molecules that you have loaded in should be released in a very sustained manner so that the cues given to the cells in the healthy area to migrate to the infarcted area. That's the concept here, and that was kind of showing a good positive in our experience there while we do the the loading and the release studies. Similar characterization was done on the ICP OES once again to confirm the presence of growth factors onto the scaffolds. The other set of confirmatory tests would be the bio part, like how far, excuse me, how far the cells are going to recognize these growth factors, because we have made these growth factors to get into the scaffolds in a very harsh environment during the electrospinning. So maybe functional, may not be functional. That would be a dilemma. So we need to check either their functionality, the growth factor, or the signaling molecules functionality is not compromised during the electrospinning conditions. And that's where we try to see the phylopodia of the HUVEX, this time not with H9C2, whereas the HUVEX, because HUVEX are the right one to check with the growth factors VEGF and FGF, and they tend to show a positive response towards those. So first one would be the scaffolds onto which we have seeded the HUVEX and get to see the phylopodia formation, and the cells are trying to get into the pores of the scaffold, which is like a positive response, and they try to go in depth into the core of the scaffold. Whereas in the B73, where we don't load any growth factors, we don't see the cell's movement anywhere. So it's just sticking onto that, which was shown in the yellow marks there in the panel at figure 3. And then the blue stain, where you could see the elongation of the cellular matrix, is also confirmed by the nuclear level. That's a nuclear staining. So this was being quantified at a various level. That was the figure number 3. Figure number 4, this is a migration chamber analysis, where you have the scaffolds and you have the cells on top. You have the filter, which is like a very stringent pore size, like 3 microns, or very stringent pore size, where the cells should really have a positive signal to migrate within the pores to the other side of the migration chamber. So the other side of the chamber, we have kept our scaffolds, which has been loaded with the growth factor molecules. And we try to give a normoxia condition and hypoxia condition, and we get to see the cells have received the signal from the scaffolds that is released out, and they try to move the other side of the chamber, which has been quantified as well. So that's the figure number 4. And getting into the molecular level, on figure 5, we tend to see what all the molecules that will be showing a positive chain signaling after the receival of the growth factor. So we get to see all the other the growth factor signaling mechanism kicks in. So it shows a positivity of our growth factors is very functional there. Followed by which we have done our animal assessment in animal models. So the scaffolds, the dual growth factor loaded scaffolds, have been tested in an acute myocardial infarction model in rabbits. So this study was performed in Frontier Lifeline hospitals, where in collaboration with them, where we get a chance to deal with all these complex surgeries, where you have to do an open heart surgery in rabbits, followed by the implantation of the scaffolds, later on suture it and observe it for a trial of 4 weeks. So ECG and the echocardiography will be performed later, and we get to see how the heart is real-time improvisation of the heart is happening or not. So those were all the observations that we have monitored down the line. And we could see in various groups, the sham is the healthy controls, and the acute myocardial infarction, we have another control. It's a negative control we have there. And PTFE is something which commercially people or the surgeons suggest for the acute myocardial infarction patients. So we have that as a positive therapeutic control. So PTFE acts as a positive control there as well. And B73 and B73 growth factors. So two groups, one having just the growth factor loaded in the scaffolds. Another one would be devoid of any growth factors. And the last option would be just injection of the growth factors. This is to ensure that prior, like the cells, not the cells, the growth factors and the scaffolds in combination show a synergistic response to improvise the diseased cardiac tissue. And also this has been parallelly confirmed through the echo analysis, where we get to see a functional improvement in the echocardiography in the dual growth factor loaded groups. And in this section, where you can see at the end of the study period, we have sacrificed the animals, and we get to see the tissue adherence or edema, or we call that as where you get all the other layer of the tissue sticking onto onto the infarcted area. So this is quite predominant in the AMI, acute myocardial infarction model, and in the PTFE, whereas in the B73 and B73 dual growth factor loaded, we tend to see less of edema formation. And also we did a cross section and took a section of it and do a TTC staining analysis to actually quantify how much of the viable tissue now present, and with compared to the dead tissue or the scar tissue, we tend to see a good amount of increase in the viable tissue in the growth factor loaded scaffolds. Histopathological staining through hematoxylin, mesenteric and elastin myosin have also confirmed a good amount of cells have infiltrated into the scaffold where the scaffold was placed into the heart. So that was a in vitro confirmation what we did through our migration chamber analysis, but this is also confirmed through the in vivo studies as well. And in the mesenteric, we could also see a collagen formation or collagen deposition happening in the scaffold area. So the scaffold is kind of degrading and can tend to promote the secretion of the collagen. And similarly goes with the elastic vancomycin for the presence of blood vessels. So the ultimate target would be the blood vessels, because we want the scar tissue now to be repopulated with blood vessels followed by a live vasculature coming back. So this was happening, and it was confirmed through the elastic vancomycin, and the blood vessels has been further confirmed through the immunostaining of smooth muscle actin. And we here in the B73 dual growth factor groups, that's a good amount of blood vessel is positive in the scaffold region, and this has been quantified. Overall, this study, we did a nanotechnology or tissue engineering approach to load the growth factor molecules in the nanofiber scaffolds to put them onto the acute myocardial infarction models and tested it's working for the improvisation or for the functional repair and regeneration of the cardiac tissue. So this was a part of today's cardiac section, followed by which I'll move into the second part of the cardiac section, where the similar approach but with a different therapeutic molecule. This is resveratrol. This is one of the very popular and a very promising therapeutic agent known to repair, and it has a well-known establishing signaling cascade where it can induce angiogenesis. So that's why we know this molecule. This is a golden molecule. Sometimes people consider this as, and this time we try to put it into the scaffolds, nanofiber scaffolds, not with two different systems, but just the PCL, which is like widely FDA approved and widely approved for polymer for in vivo purposes. So here you could see MI, myocardial infarction was created in mice, onto which we have placed the scaffolds, just the PCL, and in the second one we placed also the resveratrol loaded PCL. And this is the second panel of monitoring or the echocardiography analysis that shows the cardiac output as well as the left ventricular left ventricular diastole, and the stroke volume, all the parameters that is involved in the echocardiography analysis seems to be quite positive towards the resveratrol loaded PCL rather than the PCL alone. So the improvisation, what we try to see here is due to the addition of resveratrol, it should signal the angiogenesis. So in my previous study, I was showing growth factor mediated approach, whereas in this case we don't go with the growth factor approach, because sometimes it's delicate or dangerous, because too much of growth factor would eventually lead into excess blood circulation again happening, which will lead to the carcinogen or the teratoma formation. So we don't want that to happen. Rather, we go it in a different way through a therapeutic molecule, and this is where we are with the resveratrol. And this is another confirmation through histopathological staining, hematoxylin and eosin and Mason trichome, confirming the presence of cells and presence of collagen deposition happening as expected into the scar area where the scaffolds have been placed. Further confirmation down the line, as we were expecting, the resveratrol should be released and trigger all the signaling cascade, which is again confirmed through the cardiac troponin T staining connexin 43, whereas in the lower panel from M to P confirms the presence of both connexin 43, the establishment between two cardiomyocytes that's happening, that is just vital for a functional cardiac tissue, that is to contractile and relax. So this is happening in the group where we have loaded the resveratrol, which is positively stained for both the green and the red. Whereas in the PCL only group, which is from I to L, we don't see this happening. So which is again confirming the resveratrol is quite promising here, that has been released and which is active. And further line of confirmation. So this is again, we are confirming all the signaling molecules down the path, which is mediated through the resveratrol, which is thyroidoxin 1, and the VEGF also shows the positive signal only in the thyroidoxin loaded, only in the resveratrol loaded PCL scaffolds, but not in the plain or the non-loaded ones. So with this, I'll shift gear to the second approach, that is for the bone, because I have very limited time. I've been given a half hour to wind up whatever I have to say. So I'll just shift gears to bone, which I have gathered some experience. Although exact my experience cannot be, like, my research output cannot be delivered at this time because of the unpublished data category, whereas I can able to show some of my lab where I worked in Danny Kelly's lab in Trinity College Dublin, all those approach how we tackle the bone repair and regeneration can be shared with you guys. So I feel that would be a right to get the things going in the bone tissue part. So on the first panel of the slide, you can see the hierarchical organization of a bone tissue, where it starts from a nanoscale, where you have the nanohydroxyapatite followed by impregnation into the type 1 collagen, and slowly getting into the fibrils form and then to the cortical bone form. So this is a nano-mediated approach of the macro level tissue, which we call it as bone in this case. So what happens during critical size defects? Critical size defect is a complex fracture where you have a complete loss of a huge chunk of a bone. Imagine in this case, the lower panel where you can see it's a femur bone in a rat, where it's like a 5 to 6 mm of the bone will be taken off, which is equivalent to a complex fracture in a human scenario. So the approach how we try to go with here is not the electrospinning, rather we used a 3D printed mediated approach, where the 3D printing is very, very widely fascinating and widely recognized polymer scaffold fabrication technique, and which has a high potential to get into the industrial preparation of the scaffolds and also in the clinical sectors. So we have a chance to work upon one of those 3D printers to get a scaffold which has a good porosity, the cells can get into the scaffolds and kind of come up with their own repair and the regeneration. So uh, in the left panel, you can see actual 3D printed scaffolds that we have generated, and this study was recently published in 2022 from our lab. And then in the right side panel, you have the histopathology section where we have the no scaffold implanted groups and then the scaffold implanted groups. So you could see the scaffold implanted groups in the like a dotted version or the plain non-stained portion. So those were all the polymeric scaffolds, the PCL in this case. Wherever the PCL is there, you get to see the staining. The stain has not picked up in the PCL part during the sectioning. This happens, and the ideal part would be the next slide, which I'll be showing here. So which is like a goldness trichrome staining, which confirms the presence of a blood vessel into the scaffold. So what we were expecting is through this approach, the bridging of the bone should happen by itself and eventually lead to a complete bone formation. So we have the nanohydroxyapatite coated as well as loaded into the scaffolds and bridged into the critical size defects. And in the CT tomography on the left side, where you can see small spicules of the bone getting into the pores, which I was showing in the previous slide, um, into the pores of these scaffolds and try to bridge the other end. So this is at the end of 12 week where we start to see a positive signal towards the bridging. And in some of the other studies later on in the lab, we have confirmed we can also bridge a complete by addition of some other growth factors like bone morphogenetic protein BMP2. The complete bridging can be possible. So this is another area of tissue engineering where um, we have explored the possibility of repair and regenerating a very complex clinical scenario. Um, yeah, with that, I would wind up my slides and I would be available for you to answer any of your questions if there are any on the floor. Are there any questions on the floor? Yeah, now I can connect. Are there any questions in the audience?
Sir, good. Sir, good morning, sir. It was a nice presentation, sir. I have a doubt, sir. Brain is a complex organ. It can either be transplanted or regenerated. Will it be possible to use bioprinting like technology to build brain cells? And one more question is that, I have not read some article. Zebrafish is having some regenerative technology, regenerative mechanism. The zebrafish of the art can actually regenerate. Will it be possible to use zebrafish stem cells like cardiomyocytes in our human cells using tissue engineering?
Thanks for the question. I really appreciate that question actually. So it was a fantastic thought process actually. So those were the lines that where you have to think when you come up with your own ideas. So coming to the question there, so you're talking about the stem cells in the zebrafish where it can regenerate its heart, can that be used, and also related to the
Bioprinting
bioprinting
aspects of the
aspects
brain
of the brain.
tissue during the brain failure condition. So there are two questions. Is that right? Am I correct in understanding your question? So there were two questions posted. One is related to the brain tissue engineering. Another one was for the heart tissue stem cells taken from the zebrafish.
Yes, sir.
Perfect. Yeah, I'll address one by one. So I think I'm hearing a repetition of what I'm saying, but no problem. So first point related to the cardiac tissue, the stem cells from a zebrafish heart. So see, so that's a different species, and we are a different. The humans are different species. So we have to understand it'll be practically not feasible to get the cells from a different organism, use it into human, because they have a different genetic makeup and the humans have a different genetic makeup, and we cannot use those cells directly. There is no practicality of getting the cells from the other organism, but we can understand the cellular signaling or the cellular mechanism that involved for the regeneration of the cardiac tissue cells. It's not just the the zebrafish, but there were other stem cells or different mechanisms that can able to repair and regenerate the cardiomyocytes. So here, the complexity of the cardiac tissue is whatever the stem cells you throw in, they cannot survive because it's a very dynamic tissue where it's continuous contractile in nature, like the beating. So like you it's very stringent for the cells you throw in because the cells need to acclimatize to the new area and followed by its proliferation. So once they have to settle first and then the proliferation happens. Whereas with respect to the stem cells, so far as of my knowledge is concerned, whatever the stem cells they have thrown into the cardiac tissue, they tend to be coming out of the tissue where it was being, or it's mostly like they get into mortality where they cannot survive the tough ischemic tissue or the scar unhostile environment that is prevailing in the heart tissue. So that's where we bring in first the room for a good environment for the cells to nurture. So first would be setting up a space if there is going to be any cells that is coming in. So coming to the question that you have asked, can we use zebrafish stem cells to be implanted for a human cardiac tissue engineering? The answer would be no, but the signaling or mechanism, how it works, can be extrapolated to understand how this can be extra, or the factors that drives the repair and regeneration in a zebrafish can be used in a human model. That can be a viable approach that can be tested upon. So this was a part of answer that your question for your question, and the second part of the question would be bioprinting brain tissue. So brain is such a complex tissue. First of all, you should understand the complexity of the brain tissue, where sorry, you cannot imagine something like an open brain surgery and do a 3D printing now. So 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 3D printing is something which has now the potential to print both like a polymeric system and also cells. So we call that as a bioprinting. So bioprinting is printing 3D printing the cellular morphology or the cells directly into the tissue or tissue like constructs and implanting this tissue like construct into the defect area. That's another way of approaching or tissue engineering approach. That's called a bioprinting area where you print live cells, 3D print the live cells. Okay, this can be a stem cell or any cells that is required at the particular area. So coming to the complexity of the brain tissue. So brain tissue, it's again it's a very dynamic and it consumes a lot of energy. And so far there were approaches like I wouldn't say there is no such approach has so far, but there are approaches they have taken how to a small portion of a brain tissue that can be 3D print upon like a hydrogel kind of a mass that can be 3D printable in a dish. But no such approach have so far been taken to implant or this 3D printed lab 3D printed tissue into a human system or anything of that sort. So that will be another set of complexity where you have to remove or you have to create a disease model, make it make a disease model viable and implant your the viable construct and test its efficacy. That's another challenge down the line. But there were options. People now trying to print the entire organ itself, like that's called an organ printing. More advanced in the 3D printing would be the organ printing. This is happening widely in the space stations when we move outside the Earth, where people, the astronauts and kind of stuff, where they have the capability to 3D print a part of the tissue. The imagination would be printing a complete organ. So there were potentials to print a wide complexity of a tissue. So definitely in future there would be a room for 3D printing brain tissue. That's the answer for the second part of your question, if I answered your question correctly.
Yes, sir. Thank you so much, sir, for answering the question so patiently.
No problem. If you have anything of further down the line, you can get my contact from Dr. Sultan Mubin Sultana, and you can contact so that I can put some of my known colleagues here, or whoever is expert. I can just show you a guidance towards your research, focus. You can look after certain groups, research groups, where they do exactly the same stuff, and you can have your thought process on it. So keep up your curiosity. That's the motto there.
Sure, sir. Thank
Thank
you,
you.
sir.
Thank you.
College for women to occupy the seats on the dais. Thank you, sir. So, thank you, sir, for accepting our invitation and delivering that lucid plenary lecture on tissue engineering.
Thank you. Thank you very much. Thank you.
I deem it my pleasant duty to introduce the chairperson, co-chairperson, and rapporteur of today's technical session. The chairperson, Dr. Bhavani Govindarajulu, is an assistant professor at Queen Mary's College. She has a rich teaching experience of 21 years and did her PhD from Pachepas College. She has undertaken minor projects and served as external examiner and doctoral committee member. She has guided 4 PhD and 7 MPhil students. She has undertaken lot of biodiversity studies on butterflies and served as a resource person in many conferences and webinars. She has been a BOS member, academic audit member, and editorial board member of many colleges and universities. I am also happy to introduce the co-chair for this technical session, Dr. N. Tirunavakarasu, Assistant Professor, Department of Zoology, Dr. Ambedkar Government Arts College. He has completed his PhD from Annamalai University in marine biology. He has a rich scientific experience in various aquaculture techniques and practices and has received the Best Thesis Award. He has attended many national and international workshops and conferences. I am also pleased to introduce the rapporteur, Dr. Aruna, Assistant Professor, Department of Biochemistry, JBS College. She has received a PhD degree from Madras University and has won proficiency prizes in UG and PG classes and has been awarded the Best Paper Award. She has a rich teaching experience of 20 years and served as a judge and rapporteur at various conferences and participated and organized webinars and seminars. I welcome you all. Now I request Dr. Hajira Banu, Assistant Professor, Department of Zoology, to do the honors of giving away the fruit boxes to our 3 renowned guests. Chairperson, Dr. Bhavani ma'am. Co-chairperson, Dr. Tirunavakarasu sir. Rapporteur, Dr. Aruna ma'am. Now I request the chairperson, Dr. Bhavani ma'am, to take up the proceedings of the paper presentation. Thank you.
A very good morning to one all assembled over here. It's my privilege and I'm really being honored to be the chairperson for this international conference. At the outset, I would like to congratulate uh, all the faculties of Department of Zoology and Dr. Dr. Mubin for being the organizer of this conference. And I'm happy to be among you all. And I'm also happy to chair the session with Dr. Tirnao Karasu and Dr. Aruna. Okay, so now let's start the session. So few words I would like to mention. So scholars who are presenting the papers, please stick to the timing and then 5 minutes. 5 minutes for your presentation and 2 minutes for discussion. Okay? Okay, fine. Please stick to your timing and wishing you all good luck. Now I call the first. Yeah, I would like to read out the names of all the participants of this session. First one is Ishwaran from Guru Nanak College, followed by Mohana Sundari Madam from Ethraj College, Muhammad Anwar Nawaz from the Department of Biotechnology, St. Joseph's College of Engineering, and then Niveda S from Department of Zoology, MCC, Subhashini Ravik Murugan from Department of Zoology, Gindi Campus, S.A. Vidya, Assistant Professor, Stella Maris College, Silmia Parveen from JBS College, and Janaki from Department of Plant Biology and Biotechnology, Presidency College, right? Okay, so wishing you all good luck, participants. So now leave it to the first participant, Ishwaran. Ishwaran is here. Ishwaran and S. Jayaraj. I'll move to the next participant, L. Mohana Sundari. Mohana Sundari Madam from Ethraj College. Yeah, ma'am, please come. Her talk, she is going to present the paper, Effects of Elysium verum hook of extracts on drug-resistant bacteria and estimation of its antioxidant efficacy.
Good morning. Good morning, chairpersons and the students assembled here. And special thanks to the team who have organized this and given me an opportunity to present my paper. And I'm going to talk about, present my paper on antibacterial activity and antioxidant activity of Elysium verum. Elysium verum, it is commonly known as star anise. Generally, we know it as anasi mogu in Tamil. And regularly we use it nowadays in our pulao, biryani and other spicy non-veg items. And it has its origin from China, but there are a few contradictions also that it has got its origin from India also. But in Chinese pharmacopoeia, about 3,000 years ago, we have this recordings about Elysium verum and various other species, and its medicinal and traditional properties are recorded in it. And traditional properties like anti-aging, antioxidant properties, and against cold, flu, and generally when you are considering the bioactive components of it, it is very good for viral infections, common flu, and it is the precursor molecule shikimic acid, we have it in this, which later helped to find out oseltamivir, the flu medicine. And with that note, I continue my work on Elysium verum antibacterial activity and antioxidant activity. And these are the properties like all that it is given here, like antioxidant, antibacterial, antiviral. It is exactly, antiviral is the most prominent feature of this Elysium verum. Antifungal, antihelminthic, insecticidal. Am I clear? And there are extensive research in this. Essential oil of Elysium verum is explored a lot, and it has proven to be very effective, and the bioactive components of that also has to be very effective again as a kind of alternative therapy, where you can use it instead of antibiotics, where because we have the emerging problem of antibiotic resistant pathogens and new diseases coming up, and we are forced to use of like alternate medicines where you will non-hazardous and non-side effects. So in that way, Elysium verum comes in various hands like that. You can treat it for many diseases. These are the references, review of literature, where the previous work shows that various extracts of Elysium verum have proven to be good against infections, and it also have proven to be good for as an antioxidant. My objective is this. We have bought this Anasi Mogu Elysium verum from the local market. We dried it, shade, and we powdered it. And fine powder, we actually sieved it. That we had used it for the soxalate extraction. And we used 3 solvents here: methanol, diethyl ether, and hexane. This we have chosen only after thorough references, like various other solvents could also have been used, and in this we found these three are less studied, and we wanted to choose this. And the methods I have given here: extraction of the plant by soxalate apparatus, and we had screened by standard methods phytochemicals, the bioactive components like phenolic compounds, polyphenols, terpenoids, carotenoids, anthocyanins, then various other bioactive components. I have given the slide here. Next. This is the soxalate apparatus and the 3 solvent extracts. And this is the phytochemical tabulation. What are the phytocompounds that we have identified, and that plus all that indicates the richness of these phytocompounds in these extracts. Out of that, we have chosen methanol, which is found to be having rich phytocompounds. Next. And we have subjected that methanol extract alone for the GCMS, and we found these are the estragol and benzene 1-methoxy, endinol, bicyclobenzene, acetic acid, all that. And in general, we have a very common and very prominent compound called anethol, anisyl alcohol, anisyl acetone, anisyl aldehyde. These are the major ones which are playing a very important role for the medicinal property of the Elysium verum. Next. We wanted to study the antibacterial effect by the disc diffusion method. Disc diffusion method, we use the Mueller-Hinton broth, and we studied with the filter paper sterilized discs on that and impregnated with the concentrations. Next, 20, 40, and this is what is the slide, zone of inhibition that we have got it. We have chosen this E. coli, Klebsiella pneumoniae, and Mirabilis and aureus. This we have chosen, why? Because it is in the view of WHO references that we have made, because they have listed in 2019 and also in 17 that these are the pathogens which are becoming more antibiotic resistant and unable to control them. And with that view, we have chosen to find an alternate medicine for them, like alternate antibiotics for them. And E. coli, pneumonia, and mirabilis, they are all gram-negative bacteria, whereas Staphylococcus aureus, gram-positive bacteria. And there are molecular studies saying that gram-negative bacteria are more resistant to it, whereas the Staphylococcus, which is gram-positive, is very sensitive. It can be actually treated very well with the Elysium verum. And the zone of inhibition, it's actually shown with the concentration 20, 40, and 80. And with the standard, we have chosen ampicillin antibiotic. Next. And this is the graphic representation of the same. And antioxidant activity, one of the most important, another problem in food industry where preservation has become a challenge. And also it has become a very important challenge in our body where you will be having oxygen radical, and those are causing damage to the cell and their metabolism and other physiological activities. So we need to find a scavenging one, and plays a very important role in anti-aging ways. And this extract has proven to be very excellent antioxidant, which can also be used in for as antioxidant for human as well as in food industry in preservation, where it can prevent lipid peroxidation and fatty acid peroxidation and all that. And we have, this is the representation we have chosen here, the graphical representation of antioxidant activity. And coming to the conclusion that we would like to say the Elysium verum has rich bioactive components which can be effective against bacteria, various bacteria, apart from what we have chosen also, and it is also exhibiting antioxidant property. Thank you.
The topic is open for discussion. Any question from audience, please. Please hand over the mic to him.
Yes. Soxhlet apparatus. Sorry? Temperature, it was 140 degree. Yeah. Yeah, thank you for the suggestion, definitely. But the thing is, actually, the bioactive components which are present in the Elysium verum are highly hydrophobic. And we had to use the non-polar solvents for it to dissolve them and get them in the fractionated ones. I do take this suggestion, and in upcoming work, I think I can go for it, thinking of this. But here we have chosen this hot method, Montex heater. With that 140 degrees Celsius, we have taken it, and we have condensed the vapors and all that, and we have taken it as this one and desiccated and kept it in the for the future use.
Any other question? Is it cultivable worm in our country? Are we
It's actually started in the eastern side. It has its origin. All herbal medicine scientists and all, they talk about this. Recently, even Shivaraman sir has given a talk about Anasi Mogu, saying that it is originated in India. But people from China, when they have started moving in, and they took it and they understood the value of it, and they started using it in their traditional medicine long ago, whereas we came to know only through them, in fact. In their Chinese pharmacopoeia, about 300 years ago, they have all the recordings. And their science of medicine and all that has started with such ginseng and other various herbs. In that, this is playing a very important role. It is cultivatable, now it is a very common one. It can be done, sir. And it is available in the local market, and we have also understood the seriousness of it, and we, because of the special unique odor that we have it. Elysium meaning is the special odor that it is giving, because it has got elysinone and elysiverin special, and that we started liking it and we are using it nowadays.
It was a nice presentation, ma'am. And to add on to your information, right now the Spices Board of India is also working lots on this particular species, and in Kerala they are cultivating this particular plant. And one question that I have is, when you have oils for this already available, the oil form is also available, what made you chose the whole plant, I mean that seed, instead of using an oil? Did you do any comparative study of whatever is available?
Yes, ma'am. Oil actually, extraction of oil is slightly not economic, and it needs a lot of refinement. And sometimes we lose a lot of various bioactive components when we are extracting in the oil form, because it goes a lot of fractionation and then comes in the refined. Whereas when we are doing this kind of extraction, most of them are retained. Even in this comparison, diethyl ether and hexane are not able to yield you the rich bioactive, whereas the methanol could give. So based on that, we wanted to choose. And references we have made, essential oil work is already being done a lot in that. I thought of doing this one. At the same time, we have more of this kind, though it is crude. But we will have more bioactive components which can prove the antibacterial effects and the antioxidant effects better.
Thank you. And one question, last question.
Yes,
The
ma'am.
clinical isolates that you were talking about here, you have isolated it from the clay, or you've just got the sample?
We got it from King's Medicine, Gindi, ma'am.
Oh,
We
the
got it from
strains were
the strains we got,
obtained
we
and
got
then
it.
used for you.
Yes, ma'am.
Yeah, thank you.
Thank you, ma'am. Thank you.
One question, one general. So, in this recent pandemic for COVID, was there any therapy through this anasi motu?
Ma'am, yes, ma'am. The extract of this actually, because it has got rich shikimic acid.
Okay.
Shikimic acid is very important for treating flu, common flu. And from that only we have a drug, precursor molecule oseltamivir and Tamiflu. That medicine is actually under registered name, it is coming under called Tamiflu. We are making that drug only through this component essentially.
Okay. And one more thing. From your answer, you said like extracts a bit expensive, right? Yeah. And now my question is, how many grams of anasi motu you use to take the extract for a solvent? Like,
Ma'am, I actually took around 300 grams, which I could have 20 grams for my extraction.
so you were able to obtain.
I had to
20 gram
finally sieve it with the muslin.
After with this other solvents when you used in the socks.
Actually I had extra also, but 300 grams.
So 300 grams. What is the cost of the 300 grams in that case?
300 grams. It was around 125 rupees.
Okay. For so me. Comparatively, does this work?
I got it from the Paris market, and it had come out in the sun.
No, so you mean to say it's cheaper than the extract then in that case for 20 grams?
No, ma'am. This is directly when you are using it in the rice or whatever the food item, you are not actually able to get the whole thing, ma'am. So when you are extracting it with the alcohol, this non-polar solvents, we are able to get the bioactive components fractionated. And with that only, we want to directly check it on the isolates.
Okay. Thank you, ma'am.
Thank you, ma'am. Thank you.
Thank you. And I would like to call upon the next. Any more questions from the audience? Yeah, please.
Good morning. So yeah, that was a nice PPT presentation. So here my question is all about LCM verum. Specifically, she just gave some differentiation about gram-positive and gram-negative. So about E. coli and Staphylococcus and Streptococcus. So my question is, in Staphylococcus, how this LCM verum has been, it's been applicable? Is this been presented here? Like how this process works? I want to know about that. Can you please explain?
Generally, phytocompounds have very good action, like protein inhibit alterations and inhibition. Bioactive components, when they interact, see, Staphylococcus is gram positive.
Ma'am, gram positive you said. So how this is applicable, this method, LCM verum? You said it has mode of bioactive as well as antioxidant property. So yeah.
Gram negative, we have that lipopolysaccharide, which is the hydrophilic compound LPS in their cell membrane, the cell wall. Whereas in the case of the gram positive, that lipopolysaccharide layer is missing, and that allows this bioactive component to enter into the bacterial body easily, and that helps in acting against it.
Thank you, ma'am.
Thank you.
Thank you, ma'am. The next presentation is by Muhammad Anwar Nawaz.
respected.
Kindly stick on to your timings, okay? 5 minutes for your presentation and 2 minutes for discussion, okay?
Okay, sir. So, respected panel member, respected faculty and scholars. So, today's topic I'm going to talk about is interaction between calanoid copepods and calanoid and cyclopod interaction in marine ecosystem, specifically in the Chennai coast. So, next. So most of us know what is copepod, but if you don't know what is copepod means, it's just one of the member of zooplankton. So when we collect a zooplankton sample in any marine environment, we can see like 70 to 80% of the marine zooplankton samples are made up of copepods. So copepods having wide application, but one of the most used field of application is as a bioindicator. So calanoid is one of the order of copepods. They make up like 70% of the copepods of marine environment. But cyclopod is another order of copepod, which makes up, which are dominant in, especially in the brackish water and freshwater environment. So the present topic, what I'm going to present is like, I'm going to see the interaction between the copepods, because the interaction between copepods is mostly studied in laboratory condition, but here I'm going to see how the copepods are interacting in the marine environment, in the ecosystem. So the zooplankton samples were collected from Chennai coast. So from January 20. So we have any mic, ma'am? Hello? Is it audible? So zooplankton sample were collected from Chennai coast, Ennur, specifically from Ennur and Kovalam, because Ennur is the most polluted place in Chennai. And Kovalam is one of the best polluted compared to Ennur. So calanoid copepods are identified up to species level, and enumeration was done in standard method. So I'm not going to explain the methods and how it was done, because to maintain the time limit. So to study about copepod interaction, 5 possible predators. So the predatory species are only selected based on previous literatures. So similarly, 5 abundant small size copepod. So they range between like 500 microns and something. So on cyclopod copepods, I studied all the cyclopod copepods. Like it consists of 10 species, but I only choose like 5 main dominant species and physiochemical parameters like temperature, dissolved oxygen, ammonia. So I studied about more than 10 physiochemical parameters, but I'm only going to explain about 3 of them because to maintain a time limit. So statistical analysis, I use Pearson correlation and principal component analysis. So these are the copepods which I was studying about. So predatory species, centropages, Tinurinis, centropages furcatus. So I will show a picture. So this is the centropages furcatus, Tinurinis, and it is Lepidoptera, acuta, Isiva, and these are all grazing copepods. So Acrocalanus species and Paracalanus and Pseudodiaptomus. This is cyclopod copepod. So you can see the difference in shapes of these copepods. So result is the most dominant species is Acrocalanus gracilis. So it comprises of like 41% of the whole sample, and so in the chosen predatory copepod, Lepidoptera isiva was the most dominant one, and Diotana rigida, this is a dominant among cyclopod copepods. So highest temperature was observed between April to June. So an ammonia level was high in water, especially during September to November. So from both the stations. So similarly, high DO was observed in February month. So in February, the temperature is low. It is because of there is a negative correlation between temperature and dissolved oxygen in the marine environment. So calanoid copepods showed a negative correlation between ammonia. So calanoid copepods are very sensitive to a change in environment. So it showed a very negative correlation with ammonia. And positive correlation was between predatory copepod is observed. So only in a Kovalam station. So negative correlation between calanoid and cyclopod were observed in both the stations. So cyclopod copepod observed in greater density at Ennur compared to Kovalam. So this is a graph. So you can see that So this is a grazing copepods. So they were in peak during the February month, grazing calanoid copepods. So this is a predatory copepod. So they are rising when there is a peak in grazing copepods, and it gradually declines. So and there is a rise when there is a small sized cyclopod copepod attains its peak. So you can see the similar arrangement in both the stations. So this is a principal component analysis. So we can see that all the calanoid copepods are grouping together here, and the cyclopod copepods are grouping on the opposite side. So there is a negative relation between calanoid and cyclopod is being observed in the marine environment. In both the stations, Ennur and even in Kovalam, the same kind of arrangement is observed. So even though there is a little difference, so but the arrangement is almost similar. So discussion is there are various factors responsible for determine density of and diversity of copepods and aquatic environment. So in the present study, I'm only going to observe what is the relationship like. So we can see that calanoid copepod rises during February and March, but the density of calanoid is gradually decreasing during the months of September, October, and there is a rise in cyclopod during that time because. So it's because calanoid copepods are sensitive to water temperature, but cyclopod are they are adapted to wide range of physico-chemical parameters. So cyclopod are tolerant to wide range of environmental parameters. Could be the reason for high number in Ennur, and also it is a reason for their peak during September to November. So limitation is just a basic study. It only explains about a new standard copepod like the surface copepod. So we can't apply in a deep ocean. So in future there is more studies needed to study in deep about this.
Thank you, Anwar. I would like any questions from the audience?
Yeah. What basis you have chosen the site? I mean, stations.
Stations. I was studying in 3 stations actually in Ennur, Marina, and Kovalam. So Ennur is like a most polluted station in Chennai, and Kovalam is like away from all the thing. So Kovalam also having some pollutants like fishing activities going on. But compared to other 2 stations, Kovalam is a distance from the Ennur and Marina. So I choose just to compare these 2 stations.
Okay. How, based on the diversity aspects, how you can relate with the pollutant area? I mean, the area which is polluted and which is non-polluted.
So based on diversity means, if you see Ennur means, in total it was like only 20 to 25 species was observed in Ennur. But in Kovalam and Marina, the species is obviously higher number. Like in Marina observed like 40 species comparatively, and in Kovalam it was like 35 species in total.
What manual you have referred for the identification?
I know Kasturi Rangan, and also I used this Copypod website. So they keep on upload the new characteristics in this website. So.
In the PCA analysis, the one plot you have mentioned,
Yes,
so
sir.
there is, the scientific names were not in italics, so correct it.
In that statistical software I'm using, there is no option to utilize it. So
okay. Yeah. Okay, thank you for your nice presentation. Thank you.
Thank you, Anwar. Next student is Niveda from Madras Christian College. Niveda, are you there? So Niveda is not here. We'll go to the next person. Again, from University of Madras.
Good morning, everyone. I'm Subhashini, a postgraduate student from the Department of Zoology, University of Madras, and I'm here to present on my work, which is the use of biofloc technology on the growth and survival of Lithopanaeus vanami by electron microscopical and histological observations. So let us see what is aquaculture, biofloc, followed by biofloc technology. Aquaculture is the culture of aquatic organisms like plants and animals for commercial purposes in an impounded environment. And biofloc are heterogeneous macroaggregates of planktonic materials like microalgae, phytoplanktons, macroinvertebrates, protozoa, fecal matter, etc. And biofloc technology is a new technique which is promoted by the enhancement of water quality by adding external carbon sources in accordance with high level of aeration to produce high levels of microbial bacterial floc in the aquaculture systems. So there are many advantages of biofloc technology. The main advantage of biofloc technology is the reduction of demand for water, because in biofloc technology we do not exchange water in our biofloc tanks. Reduction of emission of effluents, minimizing the impact of the environment, increases the biosecurity, reducing the spread of diseases, providing the nutritional benefits of the natural productivity of the biofloc. So the commonly used organisms and carbon sources in biofloc technology are as follows. Fishes like tilapias, common carp, and catfishes are being used in biofloc technology, and shrimps like Letopenaeus vanami, Penaeus monodon, and Pfaffenthais species have been extensively used in biofloc technology. And the carbon sources are maida, wheat, rice bran, wheat bran, even coffee dust has been used as carbon source in biofloc technology extensively. And the model organisms we chose for our study is Letopenaeus vanami, and the carbon source we used for our biofloc formation is rice bran. The objectives of my study are to analyze the water parameters of both control and biofloc tanks, to determine the growth and survival of Letopenaeus vanami, to study the electron microscopic observations of shrimp's gut microbiota, and to investigate the histological variations of secretory B cells in the hepatopancreas of the Letopenaeus vanami. The methodology we used are some standardized protocols from the literature. The experimental feed that we used to feed the Letopenaeus vanami was bought from Ciba ICAR Santhum, Chennai, and the carbon source we used is rice bran, which was soaked overnight before the experiment. This is the experimental setup in which we acclimatized the Letopenaeus vanami juveniles, which was bought from a hatchery from Kanjipuram. And the right side image shows the triplicate tanks of control and the experiment, in which the experiment contained biofloc. So these are the results. The water parameters variation in control and biofloc tanks. As you can see, there is no much difference between the water temperature, salinity, and pH of the control and biofloc tanks. But as you can see, the nitrate and nitrate levels have been reduced in biofloc tanks, and the alkalinity was also increased in the biofloc tanks, which shows that the biofloc tank had low organic load, and also we maintained the biofloc tanks maintained a less acidic condition, which is favorable for the animal to survive and grow, and with increased immunity. So this is the growth and survival of the shrimps in control and biofloc tank. The initial weight was 0.7 grams, but the final body weight increased 2 grams in the biofloc tank, and the survival rate also increased 10% in the biofloc tank when compared to the control. So these are the results of electron microscope, scanning electron microscope, in which I've put the scanning electron microscopical image of water in the control tank and also in the biofloc tank, followed by the same image of shrimp in the control tank and in the biofloc tank. In this, we observed some the microbes present in the biofloc tank also was observed in the gut of the shrimp, which was in the biofloc tank. Thus, it proves that the shrimp has fed on the biofloc. These were the results obtained from the histology of the hepatopancreas of the shrimp. The hepatopancreas of the shrimp in the biofloc tank had distinct secretory B cells with the large vacuoles, which proves that it has gained some immunity. So the conclusion, in our results, we conclude that the feed which we used, the carbon source which we used, enriched the biofloc, and also it contributed to the organism's feed, and it was better than the commercial feed. And the biofloc tank also had some enriched nutrition, which eventually led to the good growth, survival of, and increased immunity in the shrimps of biofloc tank. And also this study is a win-win situation for both the animal and the farmer, in which the farmer gets benefited and also the animal gets benefited by increased growth and immunity. And also, biofloc technology is a great opportunity for women entrepreneurs and village people, in which they use cheap carbon sources like rice bran to promote the biofloc and to get a good harvest. Thank you.
Good, Subhashini. You're a PG student, right?
Yes,
Good
ma'am.
job.
Yes, ma'am.
Okay, I'll leave it to the audience. Any questions from the audience?
Rice bran, sir. Rice bran. Yes, sir. Only rice bran, sir.
How much CN?
Sir, the CN ratio was not calculated exactly, but we maintained the carbon nitrogen ratio as in such a way that the carbon was more and nitrogen was less in order to decrease the organic load for the animals, sir. But the CN ratio was not calculated exactly, sir. We have to study further on it, sir.
Plaque volume, have you measured that?
The biofloc tanks volume, sir?
No, plaque volume. Suppose 1 hour you go to settle, the biofloc funnel will be there, no?
Sir, no, sir. We didn't do that study, sir. We didn't measure that one, sir.
Next time you come and meet SIBA, I will
Yes, sir.
show the technology.
Yes, sir. Sure, sir.
Biofloc technology.
Sure, sir. Yes, sir.
Any other questions from audience?
Hello?
Hi.
Hear me? Is it audible? Yeah. So my question is about Litopenia spinae,
Yes.
benamai. So it's also known as white-leg shrimp, as
Yes.
you know.
Yes.
So you basically said about, is it fresh water or salt water?
It
So
is,
what's the lifespan of this Litopenius?
Letopenes vanamis lifespan is, I think, 2 years, and it is a saltwater. It is a marine organism. It is not a freshwater organism. It survives in saline water, which has a PPT of basically 30.
Okay, biofloc technology, is it applicable for this? Like you said about reduction in the demand of water.
Yes.
So like, what's the reason behind this? Like why it leads to this process? I want to know about this.
Why there is a reduction in demand of water is in biofloc tanks we do not exchange water. Okay, the usage of water is very much less when compared to normal cultures. In normal cultures, we are intended to change the water frequently, maybe once in a week. But in biofloc tank, we do not change water. This will improve the flocation, and also it will not lead to the spread of diseases while transferring water from different source. So this reduces the water usage. This technology.
Water usage and pathological conditions, maybe other
Excuse me?
disorders.
Come again?
No, pathological conditions you said, like,
Yes.
you know, it leads to this. Yeah,
Yes, yes.
thank you. That's it.
Yes, sir, but it survives better in saline water, sir, due to according to literature studies. It also, yes, sir, of course, it also grows in fresh water. In 15 ppt also it grows, but it survives better in saline water, sir.
Rather than seawater. Because usually in seawater, WSSB, EHP, aquatic, other virus will be, you know, that virus effect will be less in vanamic culture in freshwater condition.
Okay.
We go find out, we go given seed to farmer also.
Yes.
One thing we go to acclimatize to freshwater condition. When before
Yes.
giving seed to the farmer, you go to acclimatize the seed to freshwater condition.
But the juveniles we bought, we acclimatized in seawater, and we did the experiment in seawater, sir, which had 30 ppt. And also, this organism is proven to withstand WSSV, as you said. Yes, sir. It has survived WSSV for the past decade, and also it thrives in seawater, sir. As per our experiment, it has thrives in seawater, sir.
Thank you. Which country is pioneer in this biofloc?
Sir, China, Japan, yes, sir. But this organism belongs to Mexico. It is an exotic organism. But recently, in India, there is an increase in use of Letopenis vanami in shrimp culture, sir.
Okay. The pioneer of this particular technology is Israel.
They are doing very nicely.
Okay.
And for your kind information, this particular species, the brooder of, the mother prawn, which comes from Miami. So we procured the Miami, the farmers, they are procuring the shrimp from Miami, and it will be quarantined in our Rajiv Gandhi Center for Aquaculture situated at Neelangarai. So from where it should be certified as negative. So then only the lot of animals will be distributed throughout our country.
Yes, sir.
Okay, and another information, like Haryana, Punjab and Chandigarh, near UP also, people are now culturing these particular bioflak technology with the help of this Litapnics phenomenon. Yeah.
Yes, sir.
Thank you.
Thank you, sir. Thank you, everyone.
Thank you, Subhashini. Next presenter is Dr. S.A. Vidya from Stella Maris College.
Good morning. The title of my presentation is AV Faunal Diversity of Pallikarnai Marsh. A wetland is an area that is covered with water for at least some part of the year. Wetlands are ecotones. They are transitional areas where aquatic and terrestrial ecosystems converge. And because of this union, wetlands play host to a variety of aquatic and terrestrial organisms. Wetlands also offer us a variety of ecosystem services. Recognizing the importance that wetlands play in offering solutions to the problems of food and water scarcity and climate change, most of the United Nations Sustainable Development Goals revolve around the management and restoration of wetlands the world over. Although most of these SDGs are relevant to wetlands, some are of particular importance, especially 2, 6, 11, 13, 14, and 15. The study area, Pallikarnai Marsh, is one of the last remaining natural wetlands in the city of Chennai. 337 species of plants and animals have been recorded here. Among them are 115 species of birds. Every year, hundreds upon thousands of migratory birds visit the wetland. But sadly, over the years, due to rapid urbanization and other anthropogenic threats, the wetland has deteriorated in quality and declined in area. These are the locations at Pallikarnai from which I observed the birds. The main objective of my study was to assess the avian diversity at the marsh and to correlate any decline in species richness with habitat loss. This study began in 2018 and it's an ongoing study. Assessment was carried out using the point count method. Birds were recorded by direct observation and by calls. The taxonomic order, nomenclature, and identification was based on what is given in Birds of the Indian Subcontinent by Grimmett et al. These are the gear that I used. All the bird checklists and photographs are uploaded to a global database of birds called eBird. I also looked at checklists uploaded by other birders to have an idea of species richness and diversity over the last 3 decades. Satellite imagery, both current and historic, were examined, provided by Google Earth, ArcGIS, and Zoom Earth, to have an idea of how much of the area of the marsh has been depleted over the past 3 decades. I also did a preliminary wetland assessment based on the wetland assessment form available at the Asian Waterbird Census website. So far, 81 species of birds have been recorded belonging to 38 families and 12 orders. 4 of these bird species are near threatened. That is the spot-billed pelican, the painted stork, the oriental darter, and the black-headed ibis. Of these 81 species, 63 species are resident, while the remaining are migrant. The further categorization was done based on the association of these birds with their habitat as aquatic, wetland-dependent, and terrestrial. This shows us the distribution and diversity of birds based on family. In 1984, something startling emerged. 214 species have been recorded so far, according to eBird, but many of these species haven't been seen for the past so many years. For example, the bird in the picture is the Eurasian hoopoe. This was last seen in 1994. Another bird, the curlew sandpiper, which is a near-threatened winter migrant, was last seen in 2004. Of these 214 species, 8 have been designated by the IUCN as near-threatened, 1 is endangered, and 3 are vulnerable. When I examined the satellite imagery, again I was in for a shock because right in the middle of the marsh, right in the middle of the marsh, there is a sandy area. That sandy area actually depicts the dump yard, which is in the middle of the marsh. And this dump yard has grown in size over the past few decades, and now it has seriously depleted the area of the marsh itself. Other threats that I was able to observe during the wetland assessment were large buildings encroaching into the marsh, lots of plastic pollution, cattle grazing in the marsh and disturbing the birds, vegetation like water hyacinths taking over part of the marsh, and worst of all, a busy highway intersecting the marsh. Care Earth Trust is an NGO that has worked extensively with Pallikarnai marsh, and according to them, their estimates of the depletion of the marsh area, it was pretty shocking to note that this marsh was originally 50 square kilometers in area, and now it has reduced to a mere 3.17 kilometers. And this was also corroborated by the satellite imagery that I looked at. The threats that I observed during the marsh were reported by earlier authors, and so the decline in species richness and species diversity could be attributed to habitat loss. The most important question now is, what is it you and I can do about the bird life in Pallikarnai? Can we do something to conserve it? Yes, we can. Become a citizen scientist, visit the Pallikarnai marsh, upload eBird checklists on this global database called eBird, and this will help conservation biologists plan to conserve the birds in a better way. This has been done successfully in Chennai for raptor conservation. Thank you.
Thank you, Vidya. So I'll leave it to the audience. Questions? Any questions from the audience?
What is the sampling, period?
So right from 2018.
No, I'm asking either it's a monthly sampling or
usually I go during the migratory season, that is from December to February, at least 5 times, and then during the lean period another 10 times.
Okay, so it means monthly sampling, am I correct?
Monthly sampling, sometimes during the migratory season, monthly sampling is more, but during the lean season, it's scattered. It's not very intensive.
Because I had experience with the Tamil Nadu government higher education department sponsored program. I did the survey from 21 February to 22 February. All the weekly sampling I have done. So totally 66 species only recorded based on my on that particular 52 sampling based on the weekly sampling. But okay, yeah, thank you. Again, regarding to the ecological threats, the metro train construction is also on the way. So, how we can, how it the birds again under pressure.
So many of the birds that are resident, they've become acclimatized to all of this development, sir. Okay. It's the migrants that are most affected. Yes. So what I found is a lot of the migrants, if they don't come to Pallikarnai, some of them go to Sholingnallur, which is nearby, because there is less development, but I think that is going to change as well because of the metro.
Thank you.
Thank you, sir.
Next person is Silmia Parveen from JBS.
Good morning to one and all present here. I am here to present the study on effect of aqueous extract of garlic on lead-induced changes in the tissue of rat. The heightened concern of reduction of environmental pollution that has been occurring over the past 25 years has stimulated active continuing research and literature on the toxicology of heavy metals. Here the lead is a bluish grey metal and most abundant one found in the earth's crust, and lead is a highly toxic element and cumulative poison to human beings as well as to the aquatic organism and plants. Garlic is a species of bulbous flowering in the genus Allium. Garlic has been used as a general food and remedy in the Oriental for a long time. My aims and objectives are short time oral toxicity were conducted in male albino rats to study the toxicity of lead acetate and to analyze the effect of aqueous extract of garlic, to study the toxic effects of lead acetate on rats and to study the effect of aqueous extract of garlic, to study the effects of aqueous extract of garlic before and after administration of lead acetate in rats during short-term oral toxicity, and this study was taken for the period of 28 days. And the materials, animals, healthy male albino adult rats of Wistar strain weighing 120 to 150 grams and they are housed in a clean polypropylene cages. The animals were acclimatized to the laboratory conditions. They were maintained under the controlled condition of light and dark, and lead acetate, commercially available lead acetate that are procured from Precision Scientific Co. Chennai, India was used in the present study. The aqueous extract of garlic. The most recommended preparation of garlic is the aqueous extract of fresh garlic. The garlic cloves were peeled off, weighed, sliced and macerated with deionized distilled water. Methods. The healthy albino rats were randomly selected and segregated into 6 groups. And the lead was divided into 28 equal parts and the dosage level is determined. Group 1 animals received only feed and water, and the group 2 animals received only the lead acetate for all the 28 days, and group 3 animals received aqueous extract of garlic, while group 4 animals with both lead acetate and aqueous extract of garlic simultaneously for all the 28 days, and group 5 animals received lead acetate for the first 14 days and it is followed by aqueous extract of garlic for the next 14 days. At the group 6 animals administered aqueous extract of garlic for first 14 days and it was followed by the lead acetate for the next 14 days. At the end of the 28 days, the rats were sacrificed and the tissue of both liver, kidney and non-target organs, thymus, spleen and bone marrow were taken and they are fixed in 10% formalin and they are used for histopathological studies. The results that I have got is in the group 1 in which the rat received only feed and water shows normal architecture, whereas in group 2 they shows vacuolar degeneration, and group 3, 4 and 5 there was no abnormality detected, and in group 6 vacuolar degeneration was detected. This was found in the liver of rat. And in kidney, the group 1 as usual shows normal architecture, group 2 shows tubular necrosis, group 3, 4 and 5 shows no abnormality and group 6 shows tubular congestion. And this is the photomicrographs of thymus where the group 1 shows normal architecture, group 2 shows the depressed activity and group 3, group 4, group 5 and group 6 shows no abnormality. This is the photomicrographs of spleen where group 1 which received only feed and water shows normal architecture. Group 2 shows focal necrosis. Group 3, 4, 5 and 6 shows normal architecture. This is the photomicrographs of bone marrow of rat. Group 1 shows normal marrow elements. Group 2 received hypercellular marrow elements. Group 3, 4, 5 and 6 shows normal elements. Conclusion. In the present study, it is confirmed that when garlic is administered simultaneously or along with lead, or when garlic is administered after the lead exposure, the animals were able to counteract the toxic effects produced by the lead. However, when lead is administered to the previously garlic exposed animals, the recovery is partial. Hence, from the present research work, it can be concluded that garlic can be a curative regimen for lead toxicity. Thank you.
Thank you, Silmia Parvi. Questions from the audience? Anyone like to ask some questions? Okay, Parveen, let me start. You have used new rat.
Yes.
So did you get approval from the ethical
Yes, ma'am.
committee?
My guide got approval.
Okay. And you're saying, and I appreciate your work. Being a PG student, you have done your toxicity work. And this garlic extract, you just gave the raw extract or use
Yes,
any?
ma'am, raw extract.
Raw extracts. And then you're talking about histopathology.
Yes,
Normally for histopathology, we use fixatives to fix the tissues.
Yes, ma'am, we use formalin.
But 10% formalin, I don't
And
think the purpose of using a fixative is to retain the condition as such. Formalin, I don't think it will help.
We used hemotoxylin and eosin.
No, that is stain, ma. You're talking about stain. I'm asking about, you said you sacrifice the rats after the experimental period and you happen to fix it. You give the tissues for sectioning, for histopathology. So the normal procedure is, once you cut the tissues, you have to fix it. So normally we use carnized fixative. We don't use 10% formalin.
We used 10% formalin.
No, you will not. So the main purpose of doing histopathology, you want to see the effect of that lead acetate or whatever. So the normal procedure for histopathology is the first and foremost step. You have to fix it. Okay. So the commonly used fixative is carnized fixative. So in future, I appreciate your efforts in doing this work. Okay. So lead acetate. So what is any common impact? So what
Yes,
are the common sources of lead?
Ma'am, it is it can cause cancer to the
common sources of lead. In our day-to-day life, we come across lead.
Yes, ma'am.
So can you say any 2 examples where we use lead?
Oh,
so you write pencil, right?
Yes, ma'am.
What is the pencil?
Lead, ma'am.
Yeah. So it's highly toxic as well.
Yes,
okay, fine. Thank you. Thank you, Sylvia.
Thank you, ma'am.
Thank you. The next person is Janaki. Janaki is here? Oh, she's here. Janaki, just try to make it short and.
A very pleasant morning, one and all present here. I am going to share few findings of my study, that is toxicity, cytotoxicity and biological effects of Albizia, Lebec leaf extracts. According to WHO, the alarming statistics of breast cancer is every 4 minutes an Indian woman is diagnosed with the breast cancer, and every 13 minutes one woman dies in breast cancer in India. So causes and risk factors of breast cancer is family history, gene mutation, late childbearing and early menstruation, late menopause, obesity, increased breast density, and prolonged use of oral contraceptives, alcohol intake and tobacco, junk foods and packed foods. There are so many treatments available in India, that is surgery, chemotherapy, radiation, proton therapy, targeted therapy and immunotherapy. All these are quite expensive or either adverse side effects. So we move on to the herbal compounds. It is quite low cost effective and easily available in our India. India has rich flora of medicinal plants, renowned attention of scientists, and there are spurt of publications on the chemistry and pharmacology and clinical investigations. In recent times, medicinal plants occupy an important positions for being paramount sources of drug discovery, irrespective of its categorized groups, herb, shrub and trees. Many plant extracts and phytoconstituents has been tried for the cytotoxic activity and potential, and most of these plants tend to exert their anti-cancer properties through antioxidant mechanism. Plant-derived antioxidants have advantages as they are less toxic and more effective and economical. Antioxidants reduce oxidative stress in cells and therefore useful in the treatment of many human diseases including cancer, cardiovascular disease and inflammatory diseases. Proper understanding of the complex synergistic interactions of various constituents of anti-cancer herbs would help in formulating the design to attack cancerous cells without harming the normal cells of the body. So this is the plant I selected for my study in and around the Presidency College campus. Plants were authenticated by Professor Jayaraman, Plant Anatomy Research Center, Tambaram. The plants belongs to Mimosaceae family and the local name is Vagai. This is the overview of my study. This is the standard methods I followed for my studies. Coming to my results, phytochemical screening of Albizia labec. Among the 5 extracts tested, the methanol showed the strong presence of saponin, flavonoids, terpenoids, phenols and steroids. In the antioxidant activity, DPPS radical scavenging activity, 60% of the inhibition occurred at the 100 mg per ml concentration. In the case of superoxide radical scavenging activity, 80% of the inhibition occurred at 100 mg per ml concentration. So based on phytochemical screening and antioxidant activity, we selected the best extract methanol. So methanol extract subjected into the GCMS analysis. In that we found pregnone 3,2-dione retention time of 18.82 with the highest peak area 28.14%. So this is the mass spectra. Based on the methanol extract, we subjected to the cell viability assay, Vero cells. In normal cells up to 600 microgram per ml concentration, there is no toxicity was observed. After 743.21 mg per ml concentration only, the 50% of the cells were shows the toxicity. So we take below 600 mg per ml concentration for the cytotoxic cells, that is MCF7 cell lines. In that, IC50 was observed at 182.38 mg per ml at 24-hour exposure and 93.73 mg per ml on 48 hours of exposure. So based on this, we conclude the study demonstrates that Albizia labec could be a useful pharmaceutical material to treat breast cancer. The methanol extract showed potent anti-cancer activity with more than 50% inhibition. There is also hope that this plant would be a cytotoxic on breast cancer cell and also an other cancer cell. The current data of in vitro work appear useful for further research on in vivo studies and clinical trials in the near future. Thank you.
Thank you, Janaki. Any questions from the audience? Any questions? Yeah, please come forward.
Hello, ma'am. So my question is, you said about vitral of Albizia labec. So what's the range of it, ma'am? What's the ratio you said about vitral cytotoxicity?
I couldn't come again.
No, ma'am. Albizia labec, you said about the vitral cytotoxicity of the ranges. Like, what's the range of this?
Leaf extracts.
Yeah, leaf extract from methanol. So what's the range of it? Yeah.
I couldn't get no.
What is the ratio? Range,
ratio of extract, ma'am.
You mean to say ratio of extract?
Yeah, yes.
Extracts is done using by Soxhlet apparatus. For 70 gram of powder inserted into the Soxhlet apparatus, then we get 25 gram of extract, ma'am. In the 70 gram of powder, dried powder, we used 1000 ml of methanol and get 25 gram of final extract.
So some of the examples for plant-derived antioxidant, ma'am. Plant-derived antioxidant, which you have presented here. You said an example of Mimosaceae, a sensitive plant. Like, how did you do the study for this?
In my study also, pregnone 2-dione is also a sterile compound. It is also a best antioxidant also.
So that's antioxidant, which you use for the study. Okay.
Yes.
Thank you, ma'am.
Thank you.
Okay, thank you, Janaki. Thank you.
On behalf of the PG and Research Department of Zoology, I thank all the participants of paper presentation for an effective paper presentation. I wholeheartedly thank the chairperson, Dr. Bhavani ma'am, co-chairperson, Dr. Tirunavukar sir, and Dr. Aruna ma'am for the smooth conduct of the paper presentation. Now I request Mrs. Asifa Ahmed, Assistant Professor, Department of Zoology, to present the mementos to the chair, co-chair, and rapporteur as a token of appreciation. Thank you.
Yeah, so it was really a wonderful session. So each and every topic was unique, and I congratulate all the students and staff who had presented. Okay, we really enjoyed all the session, and it will be an inspiration and motivation for many young researchers. Thank you all.
Thank you once again, the chairperson, co-chairperson, and the rapporteur. Now I request Mrs. Asifa Ahmed to do away with the honors to the three. Now I request Dr. Bhavani ma'am to receive the memento from Mrs. Asifa Ahmed. Now I request Dr. Tirunavukar sir to receive the memento from Mrs. Asifa Ahmed. Now I request Dr. Aruna ma'am to receive the memento from Mrs. Asifa Ahmed. Thank you all.
The paper presentations have come to an end. Now I request all the participants to join us for tea. But we are already running short of time, so I want all of you to quickly go have your tea and get back into the auditorium by 11:15. The tea is arranged in the eastern quadrangle, so I request all the participants to please quickly go and have your tea and come back because we are ready with the next plenary lecture. It will be given by Professor Balasubramaniam Senthil Kumaran, Department of Animal Biology, School of Biosciences, Central University of Hyderabad. Thank you.
Good morning to one and all present here. On behalf of the management, principal, staff and students of JBAS College for Women, I welcome you all for the International Conference on Frontiers of Biosciences and Sustainable Development. Today, we have amidst us Professor B. Senthil Kumaran, Department of Animal Sciences, Central University, Hyderabad. He obtained his BSc and MSc from Jamal Muhammad College and PhD from Banaras Hindu University. He is the distinguished researcher and a recipient of many awards. To name a few, Young Scientist Award from European Society of Comparative Endocrinology and JSPS Fellowship from University of Miyazaki, Japan. He has edited many books. He is an editorial member for Proceedings of National Academy of Sciences, India. He is the associate editor for PLOS ONE USA, Gene USA, and Gene Report USA. An editor for books entitled Sexual Plasticity and Gametogenesis in Fish, Nova Science Publisher. His research area include fish molecular endocrinology and reproductive biology and endocrine disruption. He has published more than 150 papers in national international journals. I welcome you, sir, once again. I request Dr. Mubin Sultana to do the honors to our chief guest.
We would like to welcome our guest with a box of dry fruits. Thank you.
Thank you, Mubin. Thank you, sir.
This is very good morning to all of you. It's indeed a great pleasure to stand before you to give my lecture entitled Controlled Osmotic Pump Mediated Controlled Delivery of Human Gonadotrophin to Induce Off-Season Breeding in Catfish and Developing New Strategies to Unravel Regulatory Influence of Brain Pituitary Axis. At the outset, I would like to thank Dr. Mubin Sultana, organizing secretary of this conference, to think about me to bring you before to give my lecture. And I'm also grateful to Dr. Amtul Aziz, principal of this college, whom I had met just now. And she was very kind, and then she was having a lot of praise for the college and the students of this college. And really, I feel excited to give my lecture here. And I'm also very much impressed about the college I visited for the first time. And you have a very lovely auditorium, very lovely environment, and really very good one for studies. And I'm really very happy that you are in a good college, good environment. And I'm also met few people here, and I feel that you are also having excellent teaching, and then the ideas, whatever they have, to bring in an international conference to you is really wonderful, actually. So I'm really pleased to be here. And then in today's topic, if you feel that you have any doubt, feel free to interrupt me in between. I don't mind. And then you can ask me, I can clear your doubts, and I can go along. So before starting, the topic is, inducing off-season breeding. Once you say off-season breeding, means, naturally it is away from the breeding season, actually. So you are trying to do something which is not usual. So we thought on this aspect because many of the times the fish, either they can breed by themselves, natural environment. Some of the fish which breed in the natural environment does not breed in captive conditions. You may have to induce with a methodology, and then you can make them breed. In that also there is a difference between male and female. Female will have the easy way of getting induction, but male do not. So again, there are a lot of difficulties are there. But I have one motto in my life: where there is a will, there is a way. So I always believe in that, and see that what best you can do in a given problem. And then once you think of what best you can do, the problem will get solved by itself once you start putting work on that. So that's very, very important. So that's how we conceived this work. And as per the topic, today's conference theme, what we are is focusing on sustainable development goals. This is the UN mandate, and one of the mandate today we are talking is life underwater, actually. So it is a very precise topic for this meeting, where we are talking about sustainable development. And I felt very happy that I had a point to prove that my lecture will exactly fit in for this conference, where in which we are thinking of life below water. And the important idea for SDGs, that is Sustainable Development Goal, is to bring in welfare for human. Either bringing in animal welfare, human welfare, ultimately focusing on welfare. And this one will certainly fit into SDG 14, where we are talking about the animal breeding, actually. So usually, aquaculture breeding practices, if you see, lot of different practices are available, actually, because the fish can either breed by themselves, or you can create an environment, or you can induce them. So here you can see there are traditional methods, what we call is upper breeding, where in which you can put the male and female, and then they can breed automatically. So which is, most of the times you can see the foot fish like carp, or even for that matter tilapia. Like some of these fishes have the habit of breeding naturally. Even if you create conditions simulated, for example, like Japanese medaka or the zebrafish, you can have the breeding can happen once you create an environment. But there are certain fishes, like for example the siluriform fish, which is the catfish. which cannot breed even if you create an environment of simulation. So you need to induce them to breed. Okay, so that's one of the aspect which is to be seen. Next one is induce in the season which is not naturally happening. So the idea for induced breeding started with these two great scientists, one from Canada and one from China. And Professor Richard E. Peter, unfortunately, is no more. And another person is Professor Lin Un, actually. So both of them formulated the idea of induced breeding in fishes, and it was named as Limpe technique. Okay, so named after the Lin and the P, so Limpe method has been developed. And it's one of the best method in induced breeding in fishes across country or across species. People have used this method, and nowadays if you see a drug called Oprim, which is used in the market, which is a wonderful drug to induce breeding, wherein which people use a GnRH, gonadotropin releasing hormone analog, coupled with the dopamine receptor subtype 2 blocker. Okay, so dopamine is a natural inhibiting neuromodulator or neurohormone in fish, wherein which it regulates or inhibits GnRH and gonadotrophins. So if you block dopamine, combination with GnRH analog, you can induce the breeding. That was the essence of this finding, and they have done this work, and then it has been proved across species. It has been proved that it can be a good method. But this method works in the natural breeding season. People have never attempted to try in the unnatural breeding season. I will come for that. Why we need to try in unnatural breeding season? Okay, something is happening in the normal period, but you don't want that it happen in normal period, but in another period. Why you have to try that? First of all, that question should be asked. Okay, so then we can see how it can be formulated. So another method before Limpe was the traditional method of hypophysiation, wherein which pituitary extract. Hypophysis is a zoological name for common name pituitary. So you make pituitary extract and then inject into fish or use human chorionic gonadotropin. Now you will be wondering why I am talking about human chorionic gonadotropin when I am talking about fish. So human chorionic gonadotropin is a wonderful gonadotropin molecule which can induce breeding in any vertebrates. That's a wonderful hormone, and it does not have its own receptor. It uses luteinizing hormone receptor and then transactivate LH receptors better than LH, and then starts inducing in a better way than what LH can do. This is something remarkable. For example, I can give a very nice, attractive example. You know, cricket is invented by British people, but now we have more cups than British people in cricket because we have adopted, we have done that one very well. So exactly, if you compare SCG with that, SCG is a hormone produced during the pregnancy, early pregnancy, the first 3 months, and this hormone is produced by placenta, but this hormone works better than what is produced by pituitary. So that's because it uses it efficiently. Somebody, learn from you and do better than you. Exactly like that. So SCG is a very nice molecule which can induce the ovarian development or testicular development better than what LH can do or even FSH can do. So that is the evolution actually. And then also you can see, for example, PMSG, which is comparable to SCG, pregnant mare serum gonadotropin. This pregnant mare serum gonadotropin is produced during the horse or pregnancy, like human, but this is not as good as SCG again. So that means the evolution has chosen SCG as a supreme molecule to induce the gonad at any vertebrates. And even invertebrates like starfish also been used and tested with SCG, where it can induce the gonadal development in starfish also, which is something marvelous, so that the molecule is excellent one. So we thought, instead of going with GnRH analog or dopamine, we thought to go with SCG, which is easily available in the market. Also, we can adopt a simple technology with SCG. Before that, I was telling you that why we should do this actually. Why can't you try in the normal time? Why you want to try in the abnormal time? That's very, very important question. But if you see in the breeding pattern, most of the fishes have got a breeding pattern of seasonal cycle. So that means they will breed in a particular season. And interestingly, subtropical countries like India, rainfall is the stimulus. So that means rainfall, flood is the stimulus for breeding. And then photoperiod, temperature, everything comes in play. That means all these environmental factors should be conducive for breeding. But occasionally what happens, we had a very less rainfall or occasionally drought. So during this period, the fish, instead of breeding, it'll stop breeding, and then the breeding phase is missed, and then the breeding does not happen, or if it happens, it's less efficient. Which means that the farm conditions, if somebody is breeding the fish in the farm fish does not breed because the environment is not conducive, then they may lose the productivity. So if you can make the fish to produce in an off season where the rainfall or environmental factors are negligible or you are negated, and then you want to breed, then it will be wonderful actually. So that's how we have planned to do an off-season breeding. Usually the fish breeds, especially the model fish, catfish, what we are using, breeds in July, August. Instead of that, we tried to breed them in the May. So that was almost 2 months ahead of what is its natural breeding. So now it is a very tough task. It is not very easy. First of all, the fish does not breed in lab. That's one problem. Now we make the fish to breed, which has been successful when it is pursued at the time it can breed. Now you want to breed it in another season where in which it is not ready at all. Now you have to answer two questions here. One is make the fish to come for breeding. Second one, the gonad should be appropriate. It is not as you think. You have to not only induce the breeding, also bring the gonad to develop much earlier than what you expect. Now, anything if you want to develop in a hastened way, that means you are trying to prepone, maybe instead of 12 months, you are preponing into 10 months. So when you are doing that, the natural question will come, can I get the same type of development? Do you understand that point? For example, if something is ready in 12 months, you are reducing into 10 months. That means whether we are, avoiding the quality, okay, or you are compromising the quality on this because you are reducing to 12 months. Naturally, if a building has to be made in 12 months, you say the builder to make it in 10 months, then the builder will ask, can I compromise the quality? Then we will say, no, don't compromise the quality, but hasten up everything and make it in 10 months. Now, I understand from Dr. Mubin Sultana when I was sitting here, she told she want to have 2 days international conference meeting. So there are a lot of papers and presentations were there. She said, I'm having a parallel session. So you finish with the parallel session 2 days. But still, there is some compromise here because some of the audience are there, some of the audience are here, not full are sitting here. Luckily, there is no parallel session running, so all are sitting with me now. Okay? But in fish, when you want to do this breeding, certainly it should be on time. In the sense that you should make the fish to come to perfect structured development followed by breeding. So it is not as you think very easy. Okay? So that is one point. Second point is how you can induce the breeding which is not naturally occurring. So these two questions are really an uphill task, like a man holding a ball on his single finger, like Lord Krishna. Okay, he took the Govardhan hill, on single little finger, and then make the rains stop. Okay, so something like that. Now, sometimes we can also have spawning failure actually. So when I had this problem, especially when I used to work on catfish and we used to breed in our lab, we also face problem actually. Sometimes, the fish does not breed properly, have this problem. So when I went to do a postdoc or an invitation fellowship with the University of Miyazaki, where I met, my former mentor, Hirohiko Kagawa, with whom I have done a lot of research starting from my postdoctoral career. So he had developed a plan in a similar way to induce breeding in a honeycomb grouper. Honeycomb grouper is a marine fish, which also experience breeding issues. So he had used a methodology to induce through GNRH analog using a osmotic pump mediated delivery. Okay, so osmotic pump is a mini pump wherein which you can fill the substance and make the osmotic pump to release that substance into the peritoneal cavity of the animal. Okay, and then make it to act like a normal, like a hormone or a substance or a chemical you can give in that. Okay, so this was the plan he had developed for GNRH analog implantation. So once when he did this, what happened is induction of sexual maturation of honeycomb grouper with the GNRH analog environmental factors modulation. He had done it, and then these are some of the stages of the fish. You know, this is the stage in which you can see the ovulated follicle has come off. So this is the natural breeding season. But when we could induce this with the manipulation of GNRH analog with the environmental factors, he could make them to mature. But he did not go till breeding, of course. Okay, but he could see that the females are early maturing. Then he also made some experimental groups in combination with various environmental factors with combinations. And most of the times, you know, everything has been improved in a condition wherein which photoperiod, temperature, plus analog implantation has been done favorably. In other seasons, it does not happen. When you make favorable conditions of environmental temperature, photoperiod, plus analog implantation, it should be able to induce the gonadal index. That means gonadal structure development is induced. And then also he calculated with various parameters like modifying temperature, modifying photoperiod, modifying all in combinations. He could able to induce the female animal to come into improved gonadal structures. That's something the initial study which has been done by him, and he had proved that you can always bring the gonadal structures to normalcy when you can induce with the suitable combinations.
Okay, thank you.
And also he measured the GSI again in various groups with the different dosages. Again, it proved that it is worthwhile to do it. So all these experiments which has been done, one or other way proved that if we can create an artificial photothermal conditions with the combination of GNRH analogs, you could induce the breeding. Now, for me, the photoperiod and temperature in catfish is important, but the difference in between the photoperiod temperature May versus July. You know, these fishes are marine fishes, so it has got a wide variations of temperature, photoperiod. So there is a possibility of modulation there. But for me, the difference between the photoperiod temperature May versus July is not much. So what we thought is, why not we keep that as a constant? Make it very simple. And then only modulate the gonadotrophin releasing hormone analog. But again, I told you, I don't want to use gonadotrophin releasing hormone analog. I want to use SCG, which is more simplified and where it will act at the level of pituitary. Because when you want to use GNRH analog, it will act at the level of brain. So you need to do brain triggering, then brain should trigger pituitary, then pituitary should trigger gonads. But instead of that, I want to bypass the brain and just use pituitary, that's SCG acting as an LH to trigger. So naturally, I told you that SCG is a better hormone than LH, and it uses this exact pathway of LH receptor and transactivate. So SCG is naturally, it does not have any receptors. It does not have any receptors. It uses only LH receptor. Also, it uses FSH receptors. SCG receptors are not at all there. Then again, the question will come, why nature did not make an SCG receptor? Because it comes on a brief period. So the nature or the system does not want to create a receptor just for a brief period of pregnancy. So it comes, uses LH receptors efficiently and goes off. So the evolution also chose a pathway where in which not much expense is happening. If you make a receptor, if you make it to functional, it has to keep or sustain them, which is not necessary because it's going to act like LH in the placenta. So the local control is happening, not going to the brain. So which means that why it should have its own receptors? Let us use LH receptors. That's how evolution has chosen. So we have extrapolated that and found out that SCG can act like that. And also instantly in fish, Dr. Fisher and Bogard from Holland proved that the SCG works well in fish. I told you already, SCG works well not only in fish, any of the vertebrates, including invertebrate like starfish. Okay, but this was way back in 2003. Fisher and Bogart proved that SCG can be a good substitute for the fish LH. So this is a fantastic discovery, and it's helpful for us to use SCG instead of GnRH. So I will show a movie here. What is osmotic pump? Osmotic pump, already I told, it's a miniature pump which is active physiologically. You know, what osmosis naturally do? Osmosis is create a gradient, right? One is hypotonic and one is hypertonic. So once it is hypertonic situation has happened, which means that you have to have an homeostasis. That's what osmosis do naturally in the body. So what we do is we fill this osmotic pump with human chorionic gonadotrophin, so that the human chorionic gonadotrophin is filled with pump. And now SCG or the gonadotrophin is not there in the fish, it will start to release. And it will release based on the body temperature of the fish. So you need no need to any pump or motor or battery, nothing is needed. Physiology is sufficient. So you drop the osmotic pump filled in with pump into the fish by doing a minor incision and dissection, you can insert the pump. And then this pump will start releasing the SCG into the body peritoneal cavity. Okay. Again, you can ask me a question, sir, you are putting in peritoneal cavity, how it will affect the gonad? How, or in other words, how it will affect the gonad? Remember, fish is a very primitive organism. Anything used to peritoneal cavity, it will go everywhere. Remember, the LH receptor is enriched in gonad, not everywhere. So the SCG will act like LH and it will go and act only in gonad, though you are giving in the peritoneal cavity. So if you are so much interested, you can also pinpoint this osmotic pump to go to gonad. We didn't do that intentionally because it is another complication. So we just left it in peritoneal cavity and then it start to release. We know SCG will only find gonad. It won't find anywhere else. Even if it finds anywhere else, the action may not be as good as gonad. So we are very happy that it let it be in the peritoneal cavity. So let me show you how this pump works. Can you please click that video to show that? You have to click that video, then it will start to show. So there is a commentary with that actually. So it's a self-powered osmotic pump. Can you put a volume for that and put a mic on that? So actually it's a fluid filled pump. Okay. So what you do is you fill the substance whatever you want, like human gonadotrophin or any chemical, and it will start to release with the temperature through that hole into the body. It will release a specific amount per day. That means if you fill in 50 microliters, we could able to keep this for the period of almost 21 days. Okay. But if you use a rat, it is only 7 days because rat has an higher temperature in the body. So it will be only 7 days. But whereas in the fish, it is 21 days. That means 3 times because the fish body temperature will be somewhere around 18 or so. Okay. So we can hold this SCG into the osmotic pump, let it release slowly for the period of 21 days. Okay. So okay. I think the video is not coming. The audio is not coming, right? So this is a drug reservoir actually, and this is an osmotic layer. And then semi-permeable membrane will be there. And then you have to fill in with the syringe into the, now you can see the fill in. And then you put the cap, and then it will start to release. Now you have to implant this, okay? It's okay. So now we have to implant. Can you play this one by one? This is Professor Hiroki Igawa. So now you can see I take the fish out. Catfish can live out of water for a while, so no problem. So I make a small incision. The next one, you please click. Now we fill the pump. So I already filled the pump. Now I am inserting this pump into the catfish. You can see very small incision is made in the gonad region. Okay. So I put the pump inside. Now I make a suture. So now I make myself clear that the pump goes into the peritoneal cavity. This is a female fish I am showing as an example. We did the similar one to male also. And then we make a suture. So we use an eye needle, make a suture, and then we make suture, and we can drop the fish back. Okay. The fish can be anesthetized using phenoxymethanol or MS-222, putting few drops of MS-222 or phenoxyethanol diluted into the gills, few drops. Then the fish will get anesthetized, and it doesn't even know that you are doing an operation. Afterwards, it may have some pain, but it will get relieved. Can you go to the next? This is Professor Hiroki Kagawa, who had generously gifted this osmotic pump to me after his work. So this is 3 days after the procedure. You can see the small suture, and this goes off. And then we maintained, we have used 2 species here. One is Claris batrachus, which is an Indian catfish, and Claris garipinus, which is an exotic species actually. So, but the Claris garipinus is now not allowed for research by Department of Biotechnology because it's an exotic species. It always eat away the Indian species, so they don't give funding to Claris garipinus. But the Science Engineering Research Board of DST does not bother to work on Claris garipinus. They don't enforce, but they encourage you to work on the Indian species Claris batrachus. Now it is known as Claris magur. The name has been changed. And you can ask me, sir, why you worked on 2 species? The main reason for me to work on 2 species because I want to see the reproducibility. Secondly, garipinus has got a long reproductive phase. It can breed from August to November. But whereas the batrachus can breed only July, August, and that too sometimes it shuts off in July itself. So I want to use 2 species of similar genus and having different reproductive phase. That's the reason why I chose both the species. And then these are the photomicrographs of SCG induced. You can see the male will have a milt. In fish, semen is not used because the fish do not have seminal vesicles. So there is no seminal fluid contribution. So we call it as milt, which is equivalent to semen. And why we call it milt? Because it gives a milky white appearance when you make it, sperm. Okay, so that's why it's called milt. And you can see the whitish color from the pale gray color to show that it's enriched with the milt or semen. And you can see the follicles which can mature in the SCG induced tremendously actually, where it is very small before induction. And then we also measured the sperm motility. Motility also increased when you do this induction. And we also measured the GSI and also measured the stage of oocytes. All of them show that the gonad is developing more faster than what is expected naturally. And then we also measured certain genes which are important for gonadal development. All of them were showing a correlative higher expression, both steroidogenic enzyme genes and some of the factors. And similarly in the Claris garipinus, and also the in the female also we could able to see this gene expression profile is increased. And then we also measured the steroids, which are very important for breeding. All of them shoots up. And also we did the histology of the SCG to show that everything you can see, this is SCG induced male, you can see the sperm is fully enriched, whereas this is control. So you can see most of them are in spermatid stage, extraordinary development. Similarly, if you can see Claris garipinus also, you can see enriched with spermatid, but it is not in the control. Similarly, histological analysis of ovary, you can see a lot of post-vitellogenic full-grown follicles are seen, full-grown immature follicles are seen, and in spite of all these things, we could not get them breeding actually. So this is one of the drawbacks. So we did all, we could show that gonad develops, sperm motility increased, stages of oocyte increased, GSI increased, but the fish is not breeding. So we thought, this is a big failure. After all, doing all these efforts, borrow everything, and we felt like we are repeating the experiments what Professor Kagawa did to show that it is progressing. Only thing difference is he did with GNRH analog, I did with SCG. That's the only difference. But the structure developed, GSI developed, Like for not at all possible, very, very difficult. Preparatory phase, certainly yes, we can try. But only problem with preparatory phase is you cannot have 21 days. Maybe you should have little more time, then maybe we can succeed. But at least I'm happy here that at least I could prepone for 2 months. And 2 months preponing is a boon for aquaculture farmers.
Our moderator
It's not
will
simple
shortly
because
join
you
right
can have
now.
one set of fish
Yeah.
coming for breeding in May
It's
and
just
one set
waiting
of
for the
fish
moderator to
coming for
join.
July. That means he can have a 2 harvesting very easily. Okay, so that was our first aim. Maybe if I can succeed in February or March, then I will have 3 times breeding happening in this animal instead of 1 time in a year. Now, this was published in General Comparative Endocrinology, and my student Muruganand Kumar and Pratibha worked on female fish, Muruganand Kumar worked on male fish, and wrote this paper together in this journal. And
I'm audible
of
to
course,
you?
this is one of the papers.
Yeah,
and
yes,
they are
ma'am.
working as a postdoc
Thank
in
you.
National Institute of Health.
Our
She's working
moderator will
on
shortly
in
join.
Princeton College, and they are now working on human. You know, they worked on fish. Now they are working on human. That's the beauty of research. Okay, if you have an experience, whatever animal you are using for your PhD or your research does not matter. What you know is more matters actually. So that's another example I can give you where there is a will, there is a way. So you need not think that what is the topic you are working, how much knowledge you have gained. So that's more important than what is the topic. I can give an example like where you are studying is not important. How you are studying is very important. Okay. So the place does not matter. Okay. So then what we did is we used this strategy. Now we know that we can induce osmotic pump and delivery. Okay. So we thought why not we use this strategy to understand brain pituitary axis regulation. When we talk about brain pituitary axis regulation, now my question is there any brain sex difference between men and women? Many of this debate is happening, and I see in the audience very few men, many are women because naturally it's a women's college. Now the question always come, you know, especially during 10th standard or 11th standard, 10th and 12th standard, I cannot say 11th, comparison will be always there. How many girls poised, how many boys poised, who was the topper, whether girl is a topper or boy is topper. This conversation will always happen actually. And many of the times, I have only son, so he doesn't like girls getting marked. And if I come back and say that, you know, one of the girl in my department did well, and instantly in biotechnology, you see a lot of girls only coming. Boys very rarely join. You see a lot of girls. Even in my present department, there are only 6 boys, 18 girls. Okay, most of the boys goes for engineering, but now engineering also you can see girls, but naturally boys are more there. Okay, medicine or biology subjects, you can see girls are more. So when I compare, come back to home and say that, you know, this girl did very well, then my son will say that no, there is no good boy in your lab. Don't say girl did well actually. So always this comparison happens, but in nature there is difference, gender difference. And whether you believe or not, the girls have really multitasking. The girls certainly do, women certainly do, men cannot do. I can give one example. Okay. How many girls sitting here, you got a call from your father, for example, you are going for an exam or something like that, how you did? I don't think so. Only mother will ask. Whether the mother is working woman or home staying, your mother will ask more questions than your father. Okay? Your father may be more affectionate, and that will be magnified if a boy is there. Suppose imagine you have a sibling brother. The brother will be asked all sorts of questions by mother. Father doesn't even carry. Doesn't even bother whether you passed. Oh, you passed. Okay. And today also I made more phone calls to my colleagues or somebody rather than calling my son sitting there. I called him only once. My wife would have called at least 3 times now. Okay. So sometimes she used to mock me also. Yesterday there was an exam for our son. Do you know? I will say, yes, I know, certainly. Then she will say immediately, not the yesterday's exam, it's today's exam. Okay. So that way I am caught also because the men do not have understanding of, multitasking. Men has got a weakness. Why? Because the gray matter is very high for the men, whereas the white matter is more for women. That's why women can do multitasking. So there is a difference in evolution. Because of the gray matter, you give a task to a man, he will only do that task. Okay? He won't bother. He will just think about that one only. On the other hand, a female will think many things. Okay? Anything, female will think so much. Okay? Suppose you want to do this, female will think so much. Female will, what will happen, what is happen, what is happen. Even future also they will think. But the boy is not like that. Anything will be quick. Even proposing a girl, he will be very quick. But the girl to accept that proposal will take years. My wife also took 2 years to accept me. Okay? Naturally, because the brain is highly complicated, multitasking. They have to think a lot. So, that's because nature has done like that. So that's why they are thinking so much. And also if you see, men is more aggressive than women also. In terms of fighting. Men like fighting, women doesn't fight. Women are very clever. They have men you can see two types, flight or fight response. That means they will either fight or run away from that fight. No intermittent. But the females are not like that, intermittent. They will fight for water in one place, or they will fight very hard on one topic. Next day they will see, they will put shoulder hand, they will go together for a movie. They will change very quickly, but men won't change because they think on one aspect, okay? And that's because the amygdala is enriched with testosterone, so they can think so much. And because of this, the men can do many jobs. You can see the cartoon here. She'll be thinking about this, laptop, phone, everything she can do. On one hand, cooking, everything she can do. But the man gets puzzled what I should do. He will do one thing correctly. That's all. He will follow that, he will finish the job. So men is like an isolated task governing, whereas she is multitask. Nature is like that. Okay. So then we want to see what happened this in the fish. Okay. So one is fish can change sex. Naturally, they can change sex. Now you can ask me, sir, changing sex means like human? Human does not change sex, only appearance change. A boy will become girl or girl become boy, only appearance. That also, if you watch very carefully, you will find out they are neither boy nor girl. They are transgenders. Because we don't have the ability of changing sex, but fish have excellent changing sex. If a male became female, they will be fully functional like a female. They produce egg. Similarly, a female become male, they will produce eggs, sperm. Okay, so they have fantastic sex reversal mechanism. And there are some animals live as a male for 1 year and then female as a 2 year. Vice versa, female as 1 year, male as 2 years. You can ask me, sir, why it is happening? Because of in the first group, the male is bigger and it is caught, so you have to supplement that.
Good noon, good afternoon, all the participants. Shall we start right now?
It is
Please
harvested or caught.
unmute your videos.
To the school, it should increase. So that's why it happens. And some fishes also change by hormones. You can induce them by hormones. You can change sex. Some of them serial sex change.
I'm
audible.
audible.
Very puzzling fish.
Yeah, ma'am. You are audible.
You can see you might have seen a cartoon film called Finding Nemo, a clownfish.
I start with
do
the
is one
quotes
male,
of
three females will be
Dr. APJ
in a
Abdul
nest.
Kalam.
The
The learning
fish
gives
will
creativity,
guide the female
creativity
fish
leads
for the
to the
purpose of
thinking,
spawning,
thinking
and male
provides
will do the
knowledge,
nurturing.
knowledge
So
makes
once
you
you
great.
remove that male, the
So I'll
biggest
quote,
female will change
I
sex to
would
become
like
male.
to start with the
After that, if you
Islamic
reintroduce
greeting,
that male back,
Assalamu
the
alaikum wa
reversed female
rahmatullahi
will reverse
barakatuhu,
female to male
meaning
revert back.
may
This is called
the
serial sex
peace
changes. So you have
and
all
blessings
sorts of
of
sex
the
changing
Almighty
mechanism
be
in
showered
fish.
upon
But
all of us.
remember, all
A
fish
very
do
pleasant
not
good
do all the
afternoon to
mechanisms. There are some
everyone.
fish do this,
Now
some
I
fish do
call
this, some fish do this.
upon
This is a very
Miss
puzzling,
Sayeda
interesting topic going on in my
Arshin,
lab.
a research scholar,
Fish
to
which I was
recite a
talking about,
few
serial
verses
sex
from
changes.
the
One
Holy
is a
Quran
coral
and
reef
rendered its
Okinawan fish.
translation.
Another one is Hawaiian blue ray fish actually. So they're very big, very small also you can see. They do that serial sex changes. So now what we taught is using this osmotic pump strategy. What will happen when you mismatch the steroids? So this was my postdoc Mamta who did this work. She mismatched the steroids. That means instead of estradiol, let us have testosterone more in female, estradiol more in the male. So when we did that, we got several changes in the regulatory mechanism of serotonin and catecholamines. And earlier we proved that serotonin is dominant in males, catecholamines is dominant in females. But when we mismatch with this strategy of osmotic pump, we got reversed. Of course, we did not change sex because it's an adult one. But we did a number of experiments with this osmotic pump. And then do many of the studies pertaining to the gonadotropin and GnRH. And then using this strategy, we have understand several regulatory influence between brain, pituitary, and
Translation.
so we have done a lot of
In
work
the name of
on
Allah,
using
the
osmotic
most
pump
compassionate, the
so
most
that
merciful.
we can do.
All praise
One
is for
drawback with osmotic pump is you
Lord
can't
of all
silence the
worlds,
gene.
the most
The gene
compassionate,
silencing
the most
mechanism cannot be
merciful,
done with osmotic pump
Master
because
of the
it
day of
requires
judgment. You
specific
alone we
influence
worship
of
and you alone we
gene
ask
like a
for help.
silencing RNA to
Guide us
pass into
along
the
the
cell,
straight
which
path,
is not
the
possible with
path of
osmotic pump.
those you
So you
have
can
blessed,
use either
not
chemical or
those
a
you
hormone
are
so that you
displeased
can
with,
manipulate
or
and
those
then you can
who
study
are
the effects.
astray. Thank you.
So even if we can use the steroids, you can make them to change sex. And certain fish which can change sex in adult, you can use this strategy. But this fish does not change. But still we can simulate the changes. If not
Today
reversal, but you
we
can bring
have an
about changes to show the
eminent, thank you,
dominance of
Arshin.
these hormones.
Today we have an eminent person, Dr.
So some of these works we have
Padmavati
proved that what
Srirao,
is dominant
moderator of.
in males and
I
females
take up
using this
the
osmotic
privilege
strategy. So
to
we used
introduce our
osmotic pump
judge for
for
the
oxygen
session.
breeding, then used this
Dr.
strategy to
Padmavati
understand brain
is
pituitary axis
currently
regulation,
chief
and then understood many mechanisms pertaining to
lead
brain
and
pituitary axis. So
consultancy
why I'm
proprietor
saying
of
this is that you develop a
Biosciences
strategy
of
for one
proprietor
mechanism.
and
Certainly, if you think
consultancy,
hard, it
Nautilus
can be
Life
used in other
Sciences,
ways also.
registered
That's
under
research. Okay,
MSME.
so that's why
Ma'am
I'm posing
started
this
her
at the top.
career as an
And then
assistant
we
professor
also
in
recently
US
used this
and
osmotic pump
has
strategy
16 years of
and then
teaching
develop a
experiences in
neurodegeneration
India
model
and
in catfish.
abroad.
You see,
Ma'am
usually
has completed
neurodegeneration
her
models are done in zebrafish
undergraduation,
actually. So we have
postgraduation, and
done in
PhD in
catfish,
Usmania
but in this case we have
University,
not used osmotic
Hyderabad.
pump. We used
She
essentially
has
delivery of
completed
drug
her MS
into the
degree
skull vein.
of
Now we can
cellular
also
and
use
molecular biology
osmotic
in the
pump not in skull by giving in peritoneal
University of
cavity, and
New
we can
Haven,
start
CT,
producing
US. Ma'am
substances
has
which can create
patented her work in
osteoporosis. We are also doing that sort of work,
non-invasive
also modulating brain behavior.
invasive
Many of the things we
method
are
of
under
sex
plan with this strategy
determination
actually. And we
in
want
the
to
fish
prove that
animals.
osteoporosis model in fish
Recently, Dr.
with drugs
Padmavati has
in the osmotic
designed
pump.
and developed a quick
So this is my research team.
zoology
Presently, I have
reference app
a boy and
entitled as
4 students who are
Octopus
doing
Verb
currently PhD with me. And
using
I have a
Visual
postdoc.
Studio
And the work which
2010.
has
Ma'am has
spoken to
also
you
developed
on
a
the
novel
osmotic pump
desktop
related sex-steroid
clock
mismatch.
called
Dr. Mamta was an RA in my lab,
Anjani
and also my
Zoo
wife,
Clock
who is also
to
RA in my
display
lab,
time
Dr.
by
C.C.
animal
Sudakumari, worked on that.
representations. Ma'am has
Grateful to
submitted
all of you for listening to my lecture, and I'll be very
CY
happy
gene in
if you have any
NCBI
questions to ask me.
GenBank. Her area
Thank
of
you.
research are cellular and
So
molecular
wonderful
biology and
presentation.
invertebrates.
What is
She
the
is the
size of
member
minimum
in
maturity for the
Society
catfish
of
for
Biology
inducing
London,
agent?
UK,
Usually 1 year
International
they mature.
Society of Zoological
So 1 year
Science,
usually like
Beijing,
something like 100
Zoological
grams, 150
Association of
grams of
America,
Claris babur
ZAA,
or Claris batrachus.
International
But Calypinus
Zoological
grow better
Society,
actually. So you may
US,
achieve 300, 400 grams
Lives
after
members
1 year
in
growth. So
association
you
with
can
the
either try after 1
Indian
year for the
Association
second
of
year breeding,
Aquatic
or
Biology,
within 10
India,
months of the
and
first year
Indian
of the
Science
breeding,
Congress.
you can also induce. We
Ma'am
did
has
both actually.
published 11
So in
international
the month that
peer
comes for the first time,
reviewed
for
journals
example,
like
if it born
Scopus
in July,
journals
it will
and
breed
3
in June,
national
July again. That
journals
means almost 1 year.
and
So what we
presented
did is the
many
born in
papers in
July,
the
we could distribute them
international
seed
and
in June.
national
So
conferences.
the
Ma'am
the
has published
size
3
will be
books
approximately
in
the weight will
cell
be like 100,
biology
150 grams.
manuals, laboratory
Dosage,
manuals, and
we have
picture
given
at
5,000
large.
international units into the pump to release for 21 days. And then we gave one shot of 2,500
Ma'am has
international
signed
units to
memoirs
induce
with
one
our
shot to beat the
college,
LS surge.
JBS College for
And
Women
equal
in
amount we have given
internship
in the male
and
also. But remember,
on-the-job
male
training.
does not
Also be
sperminate.
guide
That's
and monitoring,
difficult. We are
mentoring
still
the
finding a
students.
way to
Ma'am
sperminate.
has
Even
received
in the
many
normal induction, which is
awards in
40 minutes,
his
we have to
teaching
sacrifice the males.
excellence
But still, we
and
are
also
finding a way.
developed
But
educational
certainly, the male will have a sperm.
video
Maybe
published
you
on
sacrifice
YouTube,
still you have a
and
sperm in May instead of
contributions
June.
of my blog's title has Gabby
Yes, you
Hippocampus.
may be using
Ma'am,
overprime.
I
Overprime
welcome
also we
you.
can use. That's what it's a GNRH analog combination with chemozyde, which is called overprime. That's
I
what
call
substitute
upon
of Limpe method. That also works well.
Dr.
But I want to more
Dilshad
simplified, I went for ICG.
Dilshad Begum Assistant Professor, Department
Before
of
injection, 1 month. So
Zoology,
slowly it will release, like asthmatic pump. So it is
Padmavati
giving good
ma'am with a
results.
dry
But
fruits
for us,
mask
with
for her
the
appreciation and
implant, it does
gratitude.
not breed actually. So we have to give one more shot, and then it start breeding.
Okay, thank you.
So I don't know. Maybe your fish is more responsive. What is the
yes,
model you used?
Is that?
What is the model?
Model I got to ask sir only.
So maybe carp or
Ma'am, we are very
sea
happy
bass.
and
No, sea
proud
bass
to
is
have
can
of you
sometimes naturally
today,
breed, so it
ma'am.
may be much
Once
easier.
again, a hearty
But in this
welcome to
case,
our
you need a second dose
esteemed
of
guest.
quick injection.
Let's
Actually.
start our
That
oral
means you have to fill in
presentation.
that LH.
Participants,
We are also giving a second
let
dose.
me
Okay.
remind you the rules of the
Pre-dose. So same thing
presentations.
we followed.
Turn on your cameras for the entire presentations, and you can
Okay,
share
sure.
your
That's very
screen
nice
and
treatment.
unmute when your turn comes. Okay, presentation should not be beyond 5 minutes and 2 minutes of discussions. Okay. Dr. Padmavati ma'am, shall we ask the participant to begin the presentations?
Yeah, thank you for the good intro about me. And yes, without wasting any time. Yeah, thank you for the
First
good
person.
intro about me. And
Sir,
yes,
the first
without
in the
wasting
May you said
any
that
time,
by
let us
inducing
get
the
started now.
hormone, your
Yeah,
weight gain, will there be any changes in the quality? Because after 2 months again it's gonna
So the
breed
first
naturally, right?
participants.
So will there be
No,
any
no,
changes?
you are right actually. What we did is we made the animal to breed in May itself. It does not
I'm
come into
audible.
breeding in July. That animal is only
It's
May.
clear. My voice
Then
is
you have
clear.
to wait for next 14 months. But whatever eggs produced, like you said, what will
The
happen to the
first
quality?
participant
We
is
will be able
Biblio.
to see them growing, not much difficulty.
Biblio Bindu sir,
So
are you
there is
ready
not
to
no
present?
problem of
Yes,
ma'am.
you share
I'm
your
ready.
screen and start with the
the
presentation,
fishes
sir.
do
Go
grow
with
very well. And one of
within
the,
you
if you
have a
want
lot
to, if you
short
want to ask
durations,
me
so start with the
a
objectives
question
and
stating that
aims.
what is the
Yeah.
breeding
Uh,
efficiency in
Share
July
your screen,
compared
sir.
to the induced one?
may I start?
Certainly, it is not
My
equal.
screen is
Some
visible,
people get
ma'am?
100% efficiency of
No, sir. No, sir.
breeding
Not yet
or
visible,
release of
no?
oocytes. Here
No,
it may
sir.
be 80%. But I am happy with the
Screen not
80%
visible.
because I am getting another
No,
benefit.
sir.
That 80%, the farmer can have one set of fishes to breed in May itself and keep another set of
Now
fishes again
is
in
it visible, ma'am?
July. Like
Now it is
a scientist from
Okay,
SIBA
okay.
told me that they are supplying sea bass all
My
over
topic is
India. So
application
they
of
will have a two times supply now.
umbilical cord
So
blood
that will definitely
transfusion is
advantageous
post
rather than
COVID-19
so
anemic
if
patient.
you just want
So
the same
this is a very
efficiency you cannot do
interesting
that.
the,
But still it is a good number
idea
when you compare 100.
because most of
It's not bad.
the time
So that
the
way
cord blood
I am
that is
increasing my production.
discarded in throughout
Though
the
the
world.
production is not
But
actually
in
matching
our
with July
country,
and May,
there is a
but I
nearly
have one more
1.5 billion of
production
children
outlet.
are born
If I can do again
regularly
in March, one more, then I have a full
yearly
production outlet.
in
So
India.
that will be
But out
more
of this,
supply.
we needed
But the supply may
blood
be
units.
less
That
in
is
March,
near about
little more in May, more
2
in July.
billion units is
You may
required
like
for
that rather than only good
transmission
supply.
for each and for every moment we required nearly the 4,000 units per day
Usually,
throughout the
little
India.
bit tolerable
So,
to
but
stress.
inspite that, that we
Human.
have some
So
scarcity of the blood.
the
So,
stress is a
if
little
we
bit
use the
minimized.
umbilical
And
cord
all
blood,
my
that is
experiments have
if
got a
it's rich
control.
in
For
some
example,
products
I put
that I
asthmatic pump, one with
that
saline,
is
one with SG.
the
So
umbilical
the
cord blood that is
saline induced
contain
animal only I
the
showed here
not
and
only
then
the
compared.
RBC, this
So the
contain the
stress
others
is
product,
modified
just like
there
the
in
stem cells
terms of
also is rich
effect.
in stem cells,
But
and
some
there is a
increments
various
of
type of
stress
stem
may be
cells is
there.
present within the
Usually
umbilical
fish
cord, just
are more
like
tolerable to
the
stress.
hematopoietic stem cells,
That's why
mesenchymal
they
stem
are
cells,
good both
embryonal
for
stem
fish
cells,
industry and the man,
embryonal-like stem cells.
mammalian.
There is a after it may be classified different type of cells, that is the totipotent cell, pluripotent cells, and different others, the multipotent and unipotent cells
Thank
also
you.
contain the umbilical cord blood,
Thank you,
apart
sir.
from the RBC, WBC, platelets. And the plasma is contain the various type of the immunomodulating agents. So
Sir,
this
we would wish
agents
to
we are in my study
honor you with
we
a
are
memento.
uses
So I
this
request Madam
to
Principal, Dr. Ankel
combat
Aziz,
the situation
to hand
during the
over a
COVID
memento
situation.
to to the plenary lecture.
But in this situation, those patients are the suffering from the anemia. Nearly the overall the 10% population those are affected in COVID might have the some features of the anemic features, but because the pathologically that may be the chronic anemia of previously existing disease or due to the anemia due to the COVID related disorders. The
Thank
pathophysiology
you, sir.
here
I now request Dr. Ramkis
scope
Zahra
is
to
very
propose the
less
vote of
for
thanks.
describe the pathophysiology of anemia during the COVID. But in my study, we have studied that the umbilical cord blood to see whether the umbilical cord blood
I'm
is
really
safe or
privileged to
not.
propose the
First
vote of
one,
thanks
and
for this
second one
session
to see the
4
how much
of
the
Day 2
hemoglobin is
of the
increment,
International
and third
Conference
one
on
is
Frontiers of
any
Bioscience,
blood transfusion
Sciences
related
on
adverse effects or
Sustainable
happened or
Development.
not, because
Sir,
I
your
have followed up
enlightening
all
and
the
highly
patients, because
informative
most of the
talk on
that during the
plasma
adult blood
electrode-mediated
transfusion,
controlled
there
delivery of
as a transfusion
HCG-induced
medicine specialist, I am a
off-season
transfusion
breeding
medicine
in
specialist. I am
catfish,
working in the
developing
medical
new
college hospital
strategies to uncover
in the
regulatory
department of
influence of brain
immunohematology. There we have seen
pituitary
there is a lot of
axis
patients
has
they are suffering
definitely
from
aroused a
there is
great
a allo
interest in the
antibody and
young
auto
audience
antibody that
to
is with the
apply this
transfusion
breeding
related.
technique for
So how
commercially
can we
important
combat
fishes.
the situation? The because we
Thank you,
have
sir.
we are all are known that is the hypogenicity of the umbilical cord blood, because there is more than the
I call upon
350
Dr.
antigens
Karina
apart from
ma'am,
antigens
Dr. Santosh
present
sir,
within the
and
RBC
Dr. Anju ma'am
RBC
to come
membrane,
up on the stage
outside the RBC membrane.
for the
That's
paper
very
presentation
difficult
session, ma'am.
to counteract, because that's create the challenges to develop the allo or allo antibodies, and after that the immunological tolerance that immunology is so kind of that can initiate also the
I
allogenic
request the
cells
paper presenters to get
to
ready and
autoantibody
please occupy the
to
seats in the
stimulate
first
the
row of the
production of the
auditorium.
autoantibody also. But due to the hypoantigenicity of the umbilical cord blood, there is a less chance of the formation of the allo and autoantibody after the transfusion of the cord blood. So in this
Today's
situation,
session, we have the
I have seen there
chairperson,
is the most
Dr.
of the
Karina
patients, those are the
ma'am,
symptomatic
and
during
for
the
the
follow-up.
co-chair, we have Dr.
That patients,
Santosh, and for the
the
board chair, we
COVID-19
have
patient,
Dr. Anju.
the other features
Dr.
like
P.K.
the
Karina
cough,
is an associate
the
professor,
smells, and the
Presidency
features of the
College,
headache and the
Chennai.
chronic pains
She has 27 years
also
of teaching experience.
so reduces in
She has
the
contributed
we
for the
have the
book
two arm.
Simple
Basically, we have
Task
the
and
three
Great
arm.
Concept,
The some patients have
making life science
given
learning
the
everyday
transfusion of the
catalyzed and
cord blood
supported by
transfusion in the standard
BSD
medicine, and
Britain.
some patients
She has
get
been appointed as
the
resource
medications
person for
the
National
counter
Orientation
the
Workshop for
anemia.
resource
The most of the
for
patients, those are
resource
the
teachers
symptomatically anemia.
from
I
all
have
the states
chosen
and
them
UTS
after
on
taking the
Simple
consent
Task Great
to give
Concepts,
them cord blood,
coordinated
and
by
those patients,
EcoScience Research Foundation,
the COVID-19
BSD,
patients
New
have
Delhi.
compensated the situation of
Field of
anemia, and
specialization include
those
vector
patients have the
control and
hemoglobin more
environmental
than 8
management,
gram per DL. I
phytochemistry,
have
and cancer
preferred this
biology. She has
the
10
medicine
students
that is
for
just like the
MPhil.
hematinics.
She
We are
has
using the
completed,
most common ferrous
she has
sulfate
13 students for
preparation,
PhD.
one step
Yep,
body
9 of
for
them have
regularly
completed the PhD.
for 1 month,
She has
and
earned
the
Lifetime
follow up
Achievement
those
Award for
all the patients are
Education in
followed up for the
STT
four
Awards
months for
2020
consecutive 4 months
and
to see
awarded by
whether
Karunagaram
there
Charitable Trust,
the
Trivandrum.
situation or the
Her
any
publications include
improvement or
two
any
books, that
adverse
is
events
Artificial
occurs or not.
Intelligence Game,
But
Gene Genetics,
some
Cancer
we are
Biology,
using these drugs,
Marvels of
basically the
Artificial and
moderate
Computational Intelligence
COVID
in
patients.
Life Sciences,
Those are the
another book
classified
on an overview of
as per
nanoparticles from marine
the some
environment,
COVID
modern
guidelines. That is
technology,
the
modern
patient is suffering from the
nanotechnology,
mild patient, moderate
sustainable
patient,
energy,
severe
and
patient.
environmental
According to the mild,
application.
moderate severity,
She
the
has
oxygen saturations
published more than
levels
50 papers in
also
national and
increases
international
rapidly.
journals.
Those are patients
She has
where
applied for a patent
transfuse of the
in
cord blood,
artificial intelligence
and
based prevention and
in my
prediction of
results.
early stage breast cancer
So this is
by
my the aims and
analysis of
objectives. The
mammogram
umbilical cord blood has a
images
unique
using machine
composition,
learning,
and umbilical
image
cord blood transfusion
segmentation,
to
and
post
deep learning
COVID-19 anemic patient to be the
algorithms
as a for
published
the
in
treatment of anemia and
2022.
safety and efficacy of the human umbilical
Sir,
cord
you are
blood.
exceeding your time, sir.
Our
You are
chairperson is
exceeding your
Dr.
time. Please
Vinkin
go to the
Santosh.
results,
Our
sir.
co-chairperson
Oh,
is
okay,
Dr.
okay, ma'am.
Vinkin Santosh,
So,
assistant professor,
the results,
PG and
there is a
Research Department
significant
of Advanced
the
Zoology,
median
Government
deviation
College for
between the
Men,
hemoglobin
London.
follow follow-up and the hemoglobin, the initial group, that is the more than the 0.05 level. So significance of the p-value is, in my study, the p-value of those patients who are getting the cord blood, the p-value is 0.046. So this is the less than the 0.05. So the p-value is significant, and that is the statistically significant. The median increases in the hemoglobin, that is the 8.89 gram per dl when the patient accept treatment, and compared to the initial stage of treatment, the initial stage of the hemoglobin level is 8.2 gram per dl, and there is a median increase of an amount of 0.29 gram of hemoglobin. Those patients transfuse the cord blood. So this is the result, and after the transfusion of the cord blood, and we are statistically analyzed by the IBM SPSS 20 software, and also using for the graph and data tab online software for calculation statistical analysis, ma'am. So this is overall, this is a graph, some graphs, graphical presentation for the increment of the hemoglobin after post-transfusion patient. So I conclude that, in my conclusion, that is a significant increases of the hemoglobin level of the those are patients are transfused, and the after those are follow up the patient of the 4 months, consecutive 4 months, there is no adverse related infections are visible or recorded, and there is a significant improvement of
I
the
request
other
the
symptoms
chairperson
like
to take over
the
the
chronic
session.
pain, psychological symptoms, the cough, and the fever also. Thank you, ma'am.
Thank you, sir. Questions?
That was nice presentation. Thank you so much for your presentation.
Ma'am, not audible properly, ma'am.
Can you hear me, sir?
Yeah, ma'am.
Thank you so much for the wonderful presentation, sir. If the participants have any questions regarding this paper, you can go ahead. I think nobody has any questions, I guess. Okay, so thank you so much. We can go for the second presentation by Akshara. Evaluation of ethanol properties of cardiovascular, healthcare, and andrographis, molecular data against the bacterial axis of Feliscatus.
Ma'am, can I able to lift because some patients are waiting for me.
Presenting the paper.
Yes, sir.
If
Thank
you,
you so much, sir. It was a wonderful.
Paper.
Yeah, you can
We have
lead the
around
meeting.
2 minutes of discussion.
okay, thank you,
sir.
thank
Let
you,
us
ma'am.
start
Thank
the
you.
session. I now request Jaitun
Is Akshara present?
to come over to the stage and start a
Sir,
presentation.
close your sharing, sir.
Okay, ma'am.
Hello, ma'am.
Yes. Yes, Ma, you will share your screen.
Am I audible?
Yes.
Very good.
Good afternoon to one and all
Ma'am,
present
is
here.
the
I am
screen is
Jaitun of
visible?
First MSc Zoology of JBS College
No,
for
ma'am.
Women, and I am here to present my research work on the topic Inventory of Nucleotides of Indian Snakes in the National Center for Biotechnology Information. So, introduction. In recent times, biologists have been showing keen interest on sequencing as many life forms
Hello,
as
ma'am.
possible and entering the genetic data
Yeah.
in the NCBI
Is the
database.
screen is
But as
visible?
far as my study is concerned, it is
Yes.
particularly
It's
focused on
visible, you
Indian snake
continue.
species, and snake genomics
Okay,
is
ma'am.
still at its infancy level. So, this study will
Good morning,
trigger
all of you.
herpetologists
My name is
to
Akshara. I am
conduct
currently
as
pursuing
many research as
my
possible
bachelor
like
in
this at a
zoology
wider
at TK
range.
Madhava Memorial College, Nangyarkulangara from Kerala. My presentation topic is about Evaluation of ethnoveterinary properties of Cardiospermum helicacabum and Andrographis paniculata against the bacterial abscess of Felis catus. First of all, I would like to give an introduction to my topic. A bacterial abscess in a cat is localized collection of bacterial infection. It is important to treat a bacterial abscess in a cat. Veterinary practices are often based
Objectives
on the
of this
generation
research
of
work is
experience
to
and
get
are
an
tailored to
organized
the
data
specific
of the
needs of
sequence
local
endings in the
animal
NCBI,
populations and
clearing the
ecosystem.
choice of
Some of the
genes when it
common
comes to snakes,
veterinary
and to get a
practices
clear
include the use of
understanding
medicinal
of the
plants to
proportion of the
treat
Indian
various
snake fauna
ailments,
represented in the
wounds,
NCBI,
infections,
to indicate
digestive
and
problems,
channelize where
as
the
well as
lacuna in the
acupuncture,
genetic
massages,
information
and
lies
other
for
traditional
Indian
treatments.
snakes, and to help choose
Despite
the correct
their
species
importance,
to bridge the gap.
veterinary practices are often
And the methodology of this
undervalued
research
and
work is a
overlooked.
checklist of all the
Veterinary
Indian
practices
snake
hold
species was
the
sourced from the
potential to
Zenazai
contribute to the
compilation, and it
development
was
of more sustainable and
updated by
holistic
checking the
forms
reptile
of animal
database.
care.
And the cutoff date was 30th April
I
2022.
would like to give
And
an
each of the
introduction to
scientific name of
Cardiospermum
337
helicacabum.
Indian snake
This is the
species
picture
was
of
typed in the search box of
Cardiospermum
GenBank database.
helicacabum.
And then
It's the common name
there
of
was
the
no
medicinal
attempt
plant
of me to
Urinya,
verify or go through
which is
its
seen
any
in the
faunan
countrysides
population of
of
Indian snakes,
Kerala
such as
also.
sequenced or
Belonging to the
entered,
family
or the source the
Sapindaceae,
individual
is a
originate from within the
herbaceous plant
political elements of
extensively
India.
dispersed in
In the end,
tropical
a
and
tabular
subtropical
sheet was
areas of
created,
world.
inventory of
Grows
all
in
the
plains
genes
of
sequenced
Africa,
for all the
America,
Indian species
Bangladesh,
of
India,
snakes.
Pakistan too.
So
Commonly
here is the
using for
results.
rheumatism,
All the
nervous disease,
337
piles,
species
etc.
of Indian
Also used
snakes
in
was
homeopathy.
monitored in the
Roots,
NCBI
leaves, seeds
database,
of this
and this
herb are
table shows
employed
the
as
summary
herbal
of Indian
medication.
snake species'
Plants
genetic
possess activities
sequences
like
entered in the
antimicrobial,
NCBI database.
antifungal,
Here we can
antiparasitic,
see the
antidiarrheal, etc.
Colubridae family having
This
the
is an
highest number
introduction
of
about
genetic
the
data entries in the NCBI, and the
Andrographis
Pythonidae
paniculata.
family
This
has
is
the
the picture
least
of
number of genetic
Andrographis
data entries
paniculata,
in the NCBI.
which is
So
commonly
Pythonidae
known as
family is the most
chirata or
threatened species, and it has only
kiriat, which is
nine
also a
genetic data
commonly
entries,
seen
which is
plant in
represented
the
in the
countrysides
NCBI.
of
This table
Kerala.
too shows the
That
summary
is
of Indian
traditionally
snakes
used to
showing
treat a
number of
variety of
sequence
conditions
entries
like
and towards the
malaria,
complete
diabetes,
genome.
and liver
Here the
disorder.
Elapidae family
Belonging to
has
the
the highest
family
number of genetic
Acanthaceae,
data
which
and
is native to
highest
India
number
and
of
Sri
Indian
Lanka
snake
too,
species with
widely
complete
cultivated in
genome.
southern
And
and
Acroporidae
Southeast
family has
Asia,
only
1
one
minute.
species with
helpful in
complete
various
genome.
problems related.
Table 3 shows the ecoregion wise
Can you hurry
breakup
up
of
with your
no data entries
conclusions?
of
Results
Indian
are
Okay,
snakes
ma'am.
that are
Okay,
currently
ma'am.
absent in the NCBI
This
GenBank
is the
database.
materials and
So from a
methods.
methodological
The
point of view
research
or the
conducted at NIMS Center
biogeographical
for
point of
Genomic,
view,
Tirundapuram, and the
the
abscess was
highest number
collected from
of no
Veterinary
data entry
Hospital,
species, which
Tirundapuram
is
district.
represented in the area of Western Ghats, and then followed by Northeast India, and then further followed by Andaman and Nicobar Islands.
This is the different
So
pictures
this
of plant
chart shows the
extraction.
species with
Figure
no
1 shows
data. Here the
extraction of
Colubridae
Cardiospermum
family has
helicacaba and Andrographis
highest number
paniculata.
of
Figure
snake
2,
species with no
dried
data.
extract of Cardiospermum. Figure 3,
This
dried
pie chart
extract
shows the
of
number of species with
Andrographis.
complete
And
genome.
figure
Here this
4
Elapidae
shows
family has
stock extracts
highest
of
number of Indian
Cardiospermum
snake
and
species with
Andrographis.
complete genome represented in the
This is
NCBI.
the ethnoveterinary
This
activity which
bar
shows
diagram shows
a
the number of genes versus
tabular
species,
column.
that is
This is
family.
the four
Here this
strains of
Colubridae
bacteria
family and
A1, A2,
Acroporidae
A3,
family
A4.
has most
Here
number of Indian
U1,
snake
U2 and
species, which
K1, K2
has
are
highest
the
number of
plants
genetic data entries in
we
the
selected.
NCBI.
U1 is the
So
methanolic
this
extract of
project has
Cardiospermum
been done with the
helicacaba
help
in
of
200
Chennai Snake
mg.
Trust,
U2
and
is
these are the
also that
photographs
in
which was
400
captured by
mg.
me inside the
K1 is
Chennai Snake
methanolic
Park. So
extract
this
of
is the olive
Andrographis in
keelback belonging to the
200
family
mg.
Colubridae.
K2 is also that in 400
Desert
mg.
viper from
From this
family
tabular
Viperidae.
column, we
Spectacled
find that
cobra
the
from
highest
family
activity was
Elapidae.
shown by
Striped
K2,
keelback from the family
and the
Colubridae.
bacterial strains
Indian
A1 and
python from
A4
the
high
family
resistance in both
Pythonidae.
plant
Again,
extract,
Indian python
and
from the family
A3 is
Pythonidae.
sensitive to both
Indian
plants.
rat snake from the family Colubridae. Common
Then the
sand
final
boa
part
from the
conclusion.
family Araicidae. So
Bacterial
coming to the discussion
strains
part,
A1
the
and
research
A4
which
show
was done
highest resistance to methanolic
previously
extract
focused
of
on genetics of
cardiospermum and
snake, toxins,
andriographis.
sea snakes,
Even
kiwis,
though
GA
bacterial
species of
strains
snakes in
isolated
Bangladesh,
from the swab
neurotoxins of
samples of
virus, but not that of the
abscess of
snakes in
Felis
general.
catus is
So this
sensitive
exercise
in the
research
increasing
work
concentration
pinpoints
of the
future
methanolic
scientists and
extract of
herpetologists
andriographis
working on Indian
paniculata.
snakes to sequence
Further, the
the
group is
genes of
working on
all the needy
identifying the
Indian snake species
pathogens,
as identified
isolating the
and listed
bioactive
herein,
compounds,
which
and
have been
determine the
neglected till
mode of
date.
action through wet
So this
lab
study
and
talks
in
about the
silico
Indian snake
studies.
species with complete
That's all.
genome,
Thank you.
species with no genetic data entry, taxa or family with highest number of genetic data entries, taxa or family with lowest number of genetic data entries, and this on the whole level gives a clear understanding and idea about the genetics of Indian snakes. So coming to the summary and conclusion part, a total of 2,078 entries of genetic data for 337 Indian snake species in the NCBI were identified and observed. The percentage of
Thank you,
genetic
Akshara.
data
That
and the
was your
absence of
presentation to
genetic
find
data
the
for all the
conclusion.
Indian snake species were also calculated and
You have
recorded.
completed
This research
your
found that
presentation.
among the family of Indian snake species, for Pythonidae, Acrocordidae, Xenopeltidae, and
Can you
Paradidae,
hear me?
the entire genetic data has been
Yes,
submitted in the
ma'am.
NCBI. The gene CDS, that is the coding sequence, has been observed
Any
in maximum
questions from the
number of Indian snake
participants?
species.
Thank you,
So to
ma'am.
conclude my presentation, I want to quote Baba Doyam. He said that
Okay, so thank you
in
so
the
much,
end, we will
Akshara.
conserve
I
only
guess
what
there
we
are
love.
no
We will love only what we
questions for
understand. And we will
Thanks
understand
for a
only
nice
what we are taught.
presentation. Thank you so much.
Thank you.
Thank you, ma'am.
You can, you can not if you want to. That's okay.
That was an
Okay,
excellent
anyways, thank you.
presentation.
Thank you, ma'am.
Thank you, ma'am. Thank you.
The presentation is open for discussion. Any questions?
Well,
Any
we continue with the next
clarifications?
presentation. Please, it's a request for all the participants to please maintain the time constraint. Please do not start right from introduction. I think you can start right away from material and method. Read the results and come to the conclusions. It's a request because not many participants that we have to accommodate for the session. Just got Shastra.
Which is the least family that was ported here?
Pythonidae family, ma'am. It is the most threatened species, and only 9 genetic data entry has been represented in the NCBI, and its population is diminishing over the years.
This is with respect to the Indian population, isn't
Yes,
it?
ma'am. Only Indian
By
snake
chance,
species.
did you
Can we
get
have a
some
presentation
data
by
related to
Sashra?
India?
No, ma'am. That was
Yes,
not carried
ma'am.
out in this project.
Okay. Yeah, you can start when your time starts now. Please keep it.
Okay, ma'am.
Thank you,
Okay.
ma'am.
Hello, Title.
Yes, yes.
So yeah, actually you mentioned about 337 species Indian snakes. Can you list out a few, like apart from whatever you mentioned for now? List
Is
out
my
a few
screen visible,
species
ma'am?
of this what you have mentioned so far, Indian snakes.
Yes, ma, yes.
So there is
Just continue.
Indian rat
Continue
snake,
with the
viper, Indian
objectives,
viper,
aims and
Naja
objectives.
cobra,
Start
and
from
then
here.
we have
Okay, ma'am.
common sand
Good
boa
afternoon,
like
everyone,
snake,
and welcome to
like
my
sand boa.
presentation. My topic
And
is
then
analysis of
Just
antibacterial
say that your topic
rat snake, and then
and
there
start
are even
from
sea snakes.
Okay,
aim and
ma'am.
That's it.
objectives.
Analysis
On
of
your
Actually, there are a lot
antibacterial
of other snakes, but now I have mentioned only a few.
analysis of antibacterial activity of bioactive compounds from shell extract of goose barnacles, Lepas and Cerifera.
Jaitan?
Goose barnacles. India has reported to be about 144 species of barnacles.
Next,
According
we
to
have with
data
us
collected,
here,
Tamil Nadu
Hiba,
had the most species with 77,
present a paper
followed by
on in
Andaman
silico
and Nicobar
analysis
Islands
of the
with 62
genomes of
species.
SARS
Goose barnacles are
COVID-2
filter feeding and
variant
are
of
affixed to
concern.
hard
This
surfaces
has
of
been
rocks in the
reported in
oceans.
India with
By
respect to the
ingesting
reference genome
planktons,
and
goose
nucleotide
barnacles
level.
can act as water cleaning function. Goose barnacles are rich in calcium and considered as food for animals and in certain places like Spain and Portugal.
College for Women, here to present on my study.
Materials
In silico
and methods.
analysis of the genomes of SARS-CoV-2
Sample was
variants of concern reported in
collected
India.
in Casimiro fish market. The shell was removed, dried and powdered. The sample were fixed in 70% ethanol for identification. Shell extraction of methanol and ethanol solvents. Coarse powder were extracted by coal mercerization method. In 10 grams of shell powder were mixed in 100 ml ethanol and 100 ml methanol and left undisturbed for 48 hours. The obtained products was concentrated using rotary evaporator. Then ethanol and methanol extract of Lipa shell was obtained. Thin layer chromatography. Thin layer chromatography was done to separate constituents of the extract. Chloroform, hexane and methanol were the solvents used and the retention factor was calculated. Antibacterial activity. The pure bacterial culture of Escherichia coli, Pseudomonas aeruginosa, Staphylococcus aureus and Streptococcus pneumonia were obtained from MTCC. Subcultures of each species were maintained in lab. The antibacterial activity was analyzed using agar diffusion assay. 6 mm sterile disc was placed on each plate. A volume of 100 ml methanol and ethanol extract at concentration 100 mg, 200 mg and 400 mg were dispensed into the disc. And antibiotics, levofloxacin was used because it is the most common antibacteria, and the sterile discs were incubated for 24 hours. Gas chromatography mass spectrometry was analyzed for methanol extract because it showed the highest yield extraction. The runtime in GCMS was totally 60 minutes.
Of the genomes
Results.
of SARS-CoV-2 variants
The sample was identified as Lipa's anserifera with the help of an expert extraction of active compounds. Methanol showed the highest yield extract with 15.6%, followed by ethanol with 12.2%. This is the methanol and ethanol extract. Thin layer chromatography. Methanol extract showed 3 retention factor, whereas ethanol showed 2 retention factor band. Antibacterial activity. In methanol, the maximum zone of inhibition was seen with Pseudomonas aeruginosa, followed by Escherichia coli, and lowest in Streptococcus pneumoniae. This is the zone of inhibition for Pseudomonas aeruginosa, in which 14 to 14.2 millimeter band zone of inhibition was seen, whereas in Streptococcus pneumonia, ranging from 7 to 7.2 zone of inhibition was seen. This is the graph comparing Escherichia coli, Pseudomonas aeruginosa, Streptococcus pneumoniae, and Staphylococcus aureus, in which P. aeruginosa showed the highest zone of inhibition and lowest was seen in Streptococcus pneumoniae. And for antibacterial activity for ethanol extract, the highest zone of inhibition was seen in P. aeruginosa, followed by Escherichia coli, whereas lowest was seen in Staphylococcus aureus. Overall, the highest was seen in Pseudomonas aeruginosa in methanol, and the lowest was seen in Staphylococcus aureus. This is the zone of inhibition in ethanol extract for P. aeruginosa and S. aureus. This is the graph comparing E. coli, Pseudomonas aeruginosa, Streptococcus pneumoniae, and Staphylococcus aureus. In gas chromatography and mass spectrometry, 6 compounds were derived: diethyl phthalate, diphenyl dimorpholine, desoxoacetoxy, cyclopropyl N-dimethyl, butoxycarbonyl dimethylcycline, and cyclopentacyclopropa. This is the graph from which the compounds were derived in GCMS. Conclusion.
These are some of the
It
images
has been recorded that the crustaceans were good source for search of new substances for drug development. So, commercial antibiotics are highly effective to kill the bacteria and fungal pathogens involved in common infection. Microbes tend to gain resistance after repeated usage. So, it is inevitable to find alternative antibacterial compounds. This study determines that the shell of Lipas has actively acted against common pathogens, and they can be effective.
31 nucleotides were aligned with the reference genome, and 29,785 nucleotides exactly matched with the reference genome. And there were 33 mismatches or single nucleotide polymorphism that was seen in the genome of nucleotide sequence of Delta variant with respect to the reference genome. And the Omicron subvariant BA.2, it had the highest person identity with the reference genome of 99.88%, and the query coverage of the Omicron variant was just 99%, and it had 24 mismatches or single nucleotide polymorphisms. I'll just skip to the tabular column, which shows a summary of all the results from the pairwise sequence alignment. Here we can see that BA.2 variant that had the highest person identity with the reference genome of 99.88% had 24 SNPs, and followed by Delta variant, which had 99.85% identity and had 33 mismatches, and BA.1 had 99.81% identity with the reference genome with 56 mismatches. And finally, the Omicron subvariant BA.1.1 had the least person identity of 99.80 with 57 mismatches. These are the graphs representing the result from the pairwise sequence alignment. By doing such genomic analysis on the variants of concern, we are able to identify the mutations in their genome sequences, which can give us a better understanding about their structural and functional properties. When further extending this research and done on when analyzing the amino acid sequences, we can predict what kind of changes it can bring towards the structural and functional properties of the variants, like the spike glycoprotein or the structure of the receptor binding domain, etc. And it can help us to devise better
precise and targeted therapeutic approaches and diagnostic tools towards such variants that are emerging. So the conclusion is that from interpreting the results of this study, I conclude that the Omicron subvariant BA.2 had the closest nucleotide homology with the reference genome, and the Omicron subvariant BA.1.1 had identity comparatively than the other variants that were taken in the study. So I conclude that BA.2 subvariant is the most closely related genome to the reference genome from among the variants that were taken in the study, and BA.1.1 was quite distantly related to the reference genome than compared to the other variants taken in this study. Thank you.
It was a very good presentation. So, when we go for vaccination, it is very specific to the first variant what we had it. Okay, when the Omicron came in, so is there any way the vaccination pattern was changed based on the variant? I know that it is not from your subject, but I want to know because in it was only 99 point, it is 0.13 is a variant, right?
Yes, sir. Such position in which they occur, the open reading frame in which they occur, and what kind of protein structure they may alter can lead to the variation of the virus, and that is what has given rise to different variants. And Omicron variant is considered as an Omicron variant of concern. So studies are still going on to devise more targeted therapeutic approaches for such variants of concern.
Thank you, thank you.
Thank you, Hiba. Any questions? Okay, thank you. Next, we have here Nikita Vasu. She will be presenting the paper on metagenomic analysis on the gut microbiome of a diphtheria blowfly maggot rare in solid fish waste.
Good afternoon, everyone. Myself, Nikita Vasu from Madras Christian College, and I'm here to present my topic, metagenomic study of gut microbiome of diphtheria blowfly maggot reared in solid fish waste. Blowflies are diphtherians that belong to the family Calliphoridae, and they are present widely in the environment. They are one of the first organisms to arrive on the carcasses as they feed upon the dead matter, and also they lay their eggs on the dead matter. The species of flies that breed in the carrion, such as blowflies, are said to be one of the greatest in waste management, and also they could be used as a valuable protein source. Since they feed upon these meat, they could acquire a sizable portion of their microbe from these carcasses, which they then go on to disseminate in the environment. Now, antibiotic resistance has been increasing quite lately. Perhaps it's because of the overuse of antibiotics, especially in India. Aquaculture units that use these antibiotics could be a reservoir for antibiotic resistant genes. Hence, these blowflies which feed upon these antibiotics could be a potential carrier for these antibiotic resistant genes. This is the generalized life cycle of the blowfly, which happens in about 2 to 4 weeks. And I came across quite a few studies on blowflies reared in chicken and swine waste, but there was very little data on blowflies reared in fish waste, how their gut microbiome is, how they help in managing fish waste, and also their role in dissemination of ARG. So the objective was to study the microbial diversity from the gut of the diphtheria blowfly maggot using Illumina MESEC 16S rRNA sequencing, and also to study the dissemination of antibiotic resistant gene from the aquatic medium to the terrestrial environment. Moving on to the methodology, the adult Califora flies were procured from fish market and they were reared in the animal lab of MCC with solid fish waste. And the DNA from the third instar maggots was extracted using DNase ProSoil ProKits from Quagan. One of the samples was given for V3, V4 amplicon sequencing using the MESEC platform, and the other was amplified for the MCR1 gene. MCR1 is nothing but mobile colistin resistant gene. This is the gene that confers resistance against the colistin antibiotic, which is the antibiotic that is used as a last line of defense against multi-drug resistant gram-negative bacteria. So specific MCR1 primers were put and the DNA was amplified. The 16S rRNA sequencing was outsourced, where the sequence was stitched together to 400 base pair length using Flash program, and the taxonomic classification and relative abundance were done using the QIIME software. And the other DNA was amplified for MCR1 gene, and this is the PCR program that we followed. And the PCR product was verified using gel electrophoresis to detect the presence of MCR1 gene. This is the result that we got from the 16S rRNA sequencing. Some of the phyla that we got were Proteobacteria, Firmicutes, and Chloroflexi. And we also got about 10 genus, and some of the notable ones were Vagococcus, Galileocola, and Enterobacteriaceae. And this is the alpha diversity indexes. And the MCR gene, which was amplified and the gel was run, the gel indicated the presence of MCR1 gene from the gut microbiome DNA. To conclude this study, the analysis of V3, V4 regions was done, and firmicutes were predominantly found with 51% diversity, and Chloroflexi had the minimum domination with just 0.01%. Enterococcaceae and Vagococcus were the predominant bacteria at the family and genus level respectively. These results could open up results on using these maggots for fish waste management, and also could be used to study whether this fish waste can be used as a superior diet for culturing these maggots. The MCR1 gene was confirmed by the band visualized under UV transilluminator. Since these maggots were fed upon the fishes, it is possible that they could have acquired this gene from the fish, and there is a possibility of it getting disseminated in the environment. However, this cannot be confirmed because there needs to be an extensive study on both the fish waste as well as the adult flies to confirm the dissemination pathway, and also to establish the clinical significance. The bacterial strain needs to be analyzed, which is what we are planning to do further. Thank you.
Thank you, Nikita. Nikita, your title says the maggots were reared in solid fish waste. When you were presenting here, you said you procured the fish.
Procured the flies.
The flies. Yes. What about the fish from the waste? How was it? What type of waste you collected?
Gut and scales, ma'am.
Of the fishes?
Yes, ma'am.
And MCR gene, the dissemination of the MCR gene. You pointed out that it could be from the fish waste. How can you corroborate with your results then?
Ma'am, so for that we have to analyze the fish waste as well, so which is what we'll be doing
next.
A
possibility,
yeah.
Isn't it?
No, that was just a suggestion, like it could be probable, that's what I meant.
Any questions from the audience? Good presentation. Is there any particular reason you chose blowflies?
Ma'am, when usually people are working on house flies, but when I went to the fish market, more than the house flies, there were a lot of blowflies. And I went and read there weren't much data on them, like in regarding to this, so that's why.
And it is a good source of protein, you said. So does it have any impact to use?
Yes, ma'am, they could be used as a poultry feed, which has been done. So yeah.
Okay, thank you.
Just one thing. The last slide when you showed from your maggot to the fish, is it a GFP you would like to say? Is it how it will be the grown one, this one, the last one, right? From maggot to fish. So when you use green fluorescent protein, it normally glows. That can be taken as a biomarker. So here, what exactly would like to see?
No, we are just. I was just trying to say that the gene must be present. So the green just signifies the gene. Like I was just trying to bring the correlation.
Not glow.
No, no, no.
Okay, thank you. But your gel picture also, I could not see the.
I'm not sure why it's not. I'll
Because
show
because
the.
of light. No, no, no. Thank you, thank you.
Thank you, Nikita. Next, we have Jayanti. Extraction, isolation, characterization of bioactive compounds with anti-MRSA activity.
Good afternoon to one and all present here. I am Jayanti from SRM Arts and Science College. In recent years, there have been increase in development of pathogenic microbes towards antibiotic resistance. Among them, MRSA is an important nosocomial pathogen. It confers resistance to almost all available antibiotics. Hence, it is urgent need to isolate novel compounds to combat this pathogen. So, my research work is based on isolation of compound towards this MRSA pathogen. As we all know that extremophiles, that is the organisms present in high extreme condition. Among these extremophiles, I have chosen halophiles. Halophiles, we know that the organisms present in high saline condition. I have chosen halophiles because like many novel metabolites can be produced through these halophiles, and especially in our Indian salt pan, few reports are available. So to isolate the bacteria from salt pan, I have used Zobel Marinagar medium, that is the modified medium. Here we have isolated 324 strains. Among all these 324 strains, there is an isolate designated as MHB1. It confers resistant to MRSA pathogen. It's like it shows the zone of inhibition 14 mm, and we tested the salt tolerant of that MHB isolate. It tolerates nearly 18%. So it shows like it is like moderate halophile. The organism is present in high saline condition. To identify the MHB isolate name, the organism has been given before performing sequencing, we have performed basic biochemical tests like gram staining, then Imbic test, casein hydrolysis, starch hydrolysis test, then oxidation fermentation, nitrate reduction test. So all these biochemical tests indicates the presence of few tests like catalase, oxidase, then casein hydrolysis, starch hydrolysis. These two tests indicates the presence of like amylase and protease enzyme. So it means that it indicates it has same character like Bacillus species. To identify that genus name, that is like the complete genus and species name, we have given for 60 rRNA analysis. So this MHB1 isolate shows 99% homology with Bacillus amyloliquefaciens, and the strain has been deposited. We got the accession number. The next step is to isolate the bioactive compound from these Bacillus amyloliquefaciens. The first step is optimization of culture condition of Bacillus amyloliquefaciens. For optimization, I have optimized with various carbon sources and nitrogen sources. After optimization, the mass cultivation performed for nearly 5 liters. Then it was extracted with various non-polar and polar solvents. The next step is purification. Here two-step purification I have performed. The first step purification is silica gel column chromatography. Here hexane and ethyl acetate were used as eluent solvents. In the first step I have collected 72 fractions. These fractions were pooled together based on the same RF value. After the pooled fractions were like test for antimicrobial activity, among them fraction D showed efficient activity. That fraction D has been given for like a second step purification. In the second step purification, 4 fractions were collected. Among these, FD2 fraction showed efficient activity against MRSA pathogen. The next one is characterization of the compound. So I started with TLC. TLC, here I showed the comparison between the pooled fraction and the purified fraction. In the purified fraction, that is FD2 fraction, it showed the presence of clear yellow spot after spraying with perchlorate reagent. This perchlorate reagent indicates the presence of like a phenolic compound. Next one is HPLC profile. HPLC, it showed the single clear peak with the retention time of 9.7 minutes at the absorbance of 340 nanometer with no error, like it showed the purified compound. Then LCMS analysis, it indicates the presence of molecular weight of compound like 507 Dalton. Due to the electron impact on that parent compound, a small additional peak comes due to fragmentation, that is fraction 413. FTR spectrum. In this FTR spectrum, the broad stretching peak, that is 3379, it indicates the presence of OH group, which means the phenolic group, the OH group, it indicates. Then proton NMR, that is H1 NMR. Through this proton NMR, we can find out the how many H group, that protons we can check. So 6 to 8 ppm indicates the presence of OH group. The next one is cell viability. We have tested this MRSA pathogen with that phenolic compound. So within 2 hours, it shows death of almost 50%. For this cell viability, I have used propydium iodide dye. That dye binds to the DNA of dead cells and can emit fluorescence. Then electron microscope analysis, SEM and TEM analysis. So in the normal SRS pathogen, that is without treatment, we can see the caucus shape with no damage and membrane showing like intact one. But after treatment, some blebs formation appear on the caucus shape bacteria and membranes were started to damage and contents were released out from the cell. So this is like part of my PhD work. And the main thing is like we have to like isolate new compound from our like Indian salt pan towards like emerging red distress pathogens. Thank you.
Thank you, Jayanti. That was a very systematic work which you've done, and it's quite a good presentation. Any questions from the audience? So you were telling about moderate halophyte.
Yes, ma'am.
Can you elaborate on that?
Actually, halophytes is like 3 types: slight, moderate, extreme.
With respect to your strain which you...
Yes, ma'am. 0.5, 5 to 15, 15 to 30. It's like above 13, we can call it as a extremophiles. Moderate is 5 to 50. For my strain, I have checked how much like it can tolerate the salt concentration.
What was the salt concentration in the salt pan?
Salt in the salt pan, that is in the field.
Yes.
Field in the salt pan, I didn't check that one, ma'am. I just used only the microorganisms from that salt pan soil sample. Yes, ma'am.
This collection of samples, which season did you do?
Season, it's like July, August, like that, ma'am. Before starting my project work.
And you've placed this under moderate halophyte.
Yes, ma'am. Moderate
halophyte. MHB1 strain, you placed it under.
Yes, ma'am. Even so, like other bacillus amyloidocoefficients in terrestrial sources also we can observe, but it tolerates the salt concentration more when compared to the terrestrial sources.
Thank you, Jayanti.
Thank you, ma'am.
Next we have Narayani from Women's Christian College, Chennai.
Good afternoon to one and all. First of all, I wish to thank the Almighty, the esteemed faculty, principal and management of this college and the distinguished judges who have come here for having given me an opportunity to present my paper in this lecture. Now, today's work, I just want to present my work. It is the estimation of total phenol, alkaloid, and flavonoid content in the whole plant extracts of Anisomylus indica and the screening of its anti-inflammatory activity in vitro. So, the objective of the study, as I said, is to quantify the alkaloid, flavonoid, and phenol contents, and also to identify the solvent system which is suitable for the maximum extraction of the phytoconstituents in the whole plant, to identify the extract with greater efficacy in preventing actually I wanted to find out the extract which is more suitable for preventing the protein denaturation by using this particular albumin denaturation assay method. So the plant I have taken is Anisomylus indica. Not much work has been done in this particular plant, so I have chosen this plant. It is also known as Indian catmint, and it is mostly present in our Tamil Nadu. And also it is found throughout the Southeast Asia. It is a camphor-scented perennial woody shrub. It is found along the borders of the low and medium altitude places. So this has been traditionally used for treating as an antioxidant, antimicrobial, anti-HIV and Heliobacter pylori and anti-cancer activity and other anti-inflammatory activity. Only but very minimal work has been done. So, and the aerial parts, this plant has been used traditionally as a stimulant, expectorant, diaphoretic, and insecticide. The leaves are also used for treating chronic rheumatism, psoriasis, and other skin diseases. It is also applied in local snake bites. Yes, this is a picture of the plant. You can see it commonly here and also in Madras. And it belongs to the domain is given the taxonomical classification over here. The domain is Eukaryotae and kingdom is Plantae, phylum is Tracheophyta, class is Magnoleopsida, subclass Lamiaceae, order Lamias, family Lamiaceae, genus Anisomylus, the specific epithet is indica, and the botanical name is Anisomylus indica. The other synonym for it is Anisomylus ovata. Common name is Indian catmint. There are other vernacular names. I've just given the Tamil name, which is Vata Sadachi, and it's an undershrub, and this is a picture. So the methodology, these are the first basic steps. First of all, I have collected the plant and prepared the extracts, and then I've gone in for the total alkaloid content, total flavonoid content, and the estimation of total phenol content in the extracts. So the fresh flat samples were collected from Dindigul district because I could get it more from there, and then they were identified and authenticated by the Dr. J. Raman from the Plant Anatomy Research Center in Chennai. I've taken the whole plant, I washed it, shade dried, and then ground it into a coarse powder. Then I've defatted it with N-hexane. Then from the defatted residue, I've split it into 3 fractions. I've done this by soaking 5 grams of the defatted residue in 3 different solvents. These 3 different, before that I had done a preliminary phytochemical test, which I've not mentioned over here. In that I had a positive reaction for alkaloids, flavonoids, and phenols. And I got a better response for the more polar solvents. So that is why I've taken different types of solvents with varying polarity, especially towards the polar nature. So I've taken ethyl acetate as one of the solvent, methanol extract, and an aqueous extract. I've soaked this 5 grams of the defatted residue. I've done defatting because the other pigments from the plant, which should not interfere with my studies. Then after I've dissolved, I've taken 5 grams and I've dissolved it in the different solvents, and then the mixture was stirred using a vortex mixer, and then I've centrifuged it. The supernatants from the different extracts were taken and I've concentrated them at 45 degrees using a rotary vacuum evaporator and freeze-dried it at 4 degrees centigrade for further use. And the different fractions accordingly, based on the solvents, I've named them as ethyl acetate, Anisomylus indica, methanol Anisomylus indica fraction, and aqueous Anisomylus indica fraction. These fractions were further used for the quantitative estimations. Yes. So this is the estimation of total alkaloid content. So I've used for the alkaloid, I've used standard atropine, and of concentrations 25 to 400. This is the star. This is my standards, and then I have taken the different fractions, and then. Yes. Yes, this is the my phytochemical test. So wherein I had the presence of alkaloids, in methanol extract showed me the most concentration of flavonoids, phenols and alkaloids compared to the ethyl acetate. Ethyl acetate very little was seen and aqueous extract was more. Then I'd gone into the total alkaloid content where methanol extract showed me the most. Especially in the methanol extract, the content of the phenols were maximum. Then, yes, ma'am. Yes, methanol was most in the, phenol was present most in the methanol extract. Again, phenol flavonoids were most. Ethyl acetate had least quantity of least quantity of all the constituents. Ethyl acetate had least constituents of all the alkaloids and phenols and flavonoids compared to the other extracts. So then I went on to the next step. After quantifying, I had come to, I could at least made a preliminary decision that methanol extract was more potent. Then actually I went into the in vitro anti-inflammatory. Next slide. Yes, these are the graphical things by how I calculated. I used a linear regression curve, and then this is a comparative column diagram of the extract showing the concentrations, relative contents in the different extracts. This is my R square value, which showed that my line was more of a, the curve was better, the linear curve was more suitable. Yes, this is the phenol content, total phenol content. Here, this is a calibration graph of phenol. As seen, the phenol content of the methanol extract had the maximum content of phenols compared to the ethyl acetate and aqueous extract. Yes, next. Next. Just go to the mechanism of the slides are missing. Inhibition of albumin denaturation. Methodology for that. Yes, I've also done the methodology. It's not seen in the slides. Just go way back. Huh? No, after that I've done my albumin denaturation assay, ma'am, in vitro. Slide is I've done the methodology. Okay, ma'am. These are the results because the sites are not shown up. I've done it by the in vitro albumin denaturation assay, wherein I've taken different concentrations of the standards and the test extracts, and I will check the efficacy using ibuprofen as the standard drug, which is generally a non-steroidal anti-inflammatory drug, which is usually prescribed by the physicians nowadays. So I've used this as a standard and compared my extract's performance, anti-inflammatory efficacy against this particular thing, and I found out that methanol extract at concentrations of 1000 microgram gave the maximum. It could prevent the inhibit the denaturation of proteins. Next was the aqueous extract. Ethanol extract had very little efficacy. This showed that methanol extract might be due to the presence of phenols and flavonoids, which might be contributory for the activity. Discussion. I got it. So now, since inflammation is it is a mechanism, a compensatory mechanism of the living tissues in response to external stimuli which could damage our tissues. So these stimuli which cause inflammation can also disintegrate or denature the protein structure, which is also one other reason for causing inflammation. Now, because of chronic inflammation, we have disorders like arthritis, cancer, and stroke, and many other disabilities are occurring. So, when protein denaturation is inhibited, inflammatory activity also can be reduced. So, now in this study, this ibuprofen, which is a non-steroidal anti-inflammatory drug, has been used mostly, and it works by inhibiting the cyclooxygenase pathway. But since we are in need of more new compounds, especially from plant, which can prevent the side effects. But this ibuprofen is also known to cause serious side effects like bleeding, rupture, ulcers with long-term use. So compared, so in search of a conventional drug, I've tried to check this anisomless indica for checking to take out, extract the compound so that it could be used for in treatment, it could be used as a natural product to treating inflammation. Now, this particular assay is only a basic test. The high protein protective capabilities and also the lower IC50 values was almost similar or closer to methanol extract. So, which I conclude that it might be due to the flavonoids and flavonoic compounds. So, more additional research is needed to develop the bioactive principle from this plant, and so that it be used in therapy. Thank you.
Sure. I have two things. Number one, can you go? You have reference slide with you?
Yes, sir.
Number two, okay, just to make something. Okay, that is true. You said you are doing something with in vitro studies, right? It is on which cell line?
No, no, I'm not done. I've done egg album denaturation assay, sir. The slide has been missed out. Actually, it is there. It has not come up in the PPT.
No, because why I was talking about the two reference, it said anti-HIV, anti-inflammatory, and it said
yes, sir. This is the test I've done.
anti-cancerous. No, I'm talking about the reference two. Okay, in the introduction, you said this particular plan shows all anti-inflammatory, anti-HIV.
The references have not been mentioned there in proper order. That's why, sir.
Okay, so in vitro study, you didn't do it with any cell line as such.
So I've done it in the. I've done this denaturation using the eggs albumin. This is the protocol wherein I've done the assay, anti-inflammation assay. So this is my.
Ibuprofen showing more effect than the plan.
Yes, it is more effective.
Questions? Audience? Thank you, Narayan.
Thank you.
Abhisha B. She's from the Department of Biotechnology, Nurul Islam College of Arts and Science, Tirunelveli.
Good afternoon, everyone. A warm welcome to all present over here. I'm Abhisha from the Department of Biotechnology, a final year PG student, here to present on the topic Determination of Antibiotic Resistance of Klebsiella pneumoniae strains in diabetic patients. So before going into the presentation, I just want to give an overview of the work I have done. So this test is basically about to test the effectiveness of the antibiotics which are commonly used to treat the bacterial infections. So in this test, we have taken 5 strains of Klebsiella pneumoniae and 5 different antibiotics have been used. The antibiotics are gentamicin, neomycin, kanamycin, trimethoprim and oxycillin. So these antibiotics resistance is the commonly emerging health problem nowadays. So some bacteria that are capable of causing serious infections are becoming resistant to these commonly antibiotics which are used and are available antibiotics. So let's move on to the introduction part. So Klebsiella pneumoniae is a gram-negative non-motile encapsulated for lactose fermenting bacteria which are commonly found in the gastrointestinal tract and hands of the hospital patients. So this is the most common uropathogenic gram-negative bacteria which can affect any part of the urinary system, including urethra, kidney, bladder, and ureters. So antibiotics are the common medicines which are used to treat the bacterial infections. That is, they can inhibit or kill the bacteria. So antibiotic resistance is a form of drug resistance by which the subpopulations of microorganisms are able to survive or, to one or more populations. So aim of the present study is to detect the antibiotic resistance capacity of the Klebsiella pneumoniae strains in the diabetic patients, and objective is to collect those strains of the pneumonia from the diabetic patients and do morphological and biochemical studies, and finally to assess the antibiotic resistance capacity of the Klebsiella strains by the Kirby-Weaver disk diffusion method. So the materials and methods. There are actually 4 methodologies. The strains are collected from the urine samples of the diabetic patients from the Vivek laboratory,
at Nagarjuil, which is followed by the morphological and biochemical characterization of the Klebsiella pneumoniae strains, which were done according to the protocol of Cappuccino et al. Then the following tests are performed. The tests are gram strainings motility test, enroll test, MRVP test, citrate production test, H2S production, carbohydrate fermentation, and catalase test. So these are the mechanisms and results for this all these tests. Third is the confirmation of the Klebsiella and strains, which is done with the McConkey agar. So after biochemical characterizations, the collector strains were subjected to confirmation by quadrant streaking of these strains in McConkey agar, which is followed by the determination of antibiotic resistance capacity of the Klebsiella strains, which is done according to the protocol of the Kilby-Bewer disk diffusion method. So these antibiotic capacities of the collected strains were assessed using the standard antibiotic discs, and the antibiotics used here are gentamicin, neomycin, kanamycin, trimethoprim, oxycillin by Kilby-Bewer disk diffusion method. So these are the results for the morphological and biochemical characterizations. Colony morphologies can be identified by the McConkey agar, which is appeared as mucoid in shape, and these results, negative results, are shown from gram staining motility test, coagulase test, MRVP test, and H2S production, and the positive results are shown for catalase test, citrate test, and indole test. So these are the photographs for the morphological characterization of the Klebsiella strains. These strains colonies appeared as pink, which shows that they are lactose fermenting bacteria, and from this we can conclude they are mucoid in shape. As I already said, there are 5 strains. We have named for our convenience as MA1, MA2, MA3, MA4, and MA5. These are the photographs for the biochemical characterization. Left one is the test tubes for the INVIC test, and the right one is for the carbohydrate fermentation test. The sugars taken are glucose, sucrose, lactose, arabinose, and dextrose, and it is samely done for MA1, MA2, and MA3, MA4, and MA5 strains. So these are the results for the carbohydrate fermentation test. None of them shows gas production. And positive results were seen for glucose, lactose, and MA2 2 strains and 3 strains have positive for arabinose, and for negative results, sucrose, dextrose shows all the strains have negative results. In MA1, 4, 5 shows arabinose has negative results. And these are the results for the antibiotic disc. The zone of inhibition is measured. So the antibiotics trimethoprim and oxycycline shows complete resistance to all of the strains we have taken, and kanamycin and neomycin are at the intermediate level, which is between 12 to 19 millimeter. These are the photographs for the antibiotic disc. So the antibiotics are placed at regular intervals. The gentamicin shows relatively a larger zone when compared to all other antibiotics used. So oxycillin and trimethoprim shows complete resistance by the Klebsiella pneumoniae strains, and these for MA1, MA2, and MA3 plates. And this is for MA4 and MA5. In MA5, the gentamicin shows also completely resisted by the Klebsiella pneumoniae strains, and little larger zone of inhibition than the gentamicin is shown in neomycin. So the summary and conclusion of the work is, so from this work the morphological and biochemical characterization has been done to confirm this as the Klebsiella pneumoniae strains. This clearly indicates that they are glucose fermenting bacteria which can affect the urinary tract infections of the diabetic patients. The 5 strains, MA1, 2, 3, and 4, 5 strains are different strains used, and the antibiotics used are gentamicin, neomycin, kanamycin, trimethoprim, and oxycycline. So from all this, all the strains shows complete resistance to trimethoprim and oxycycline, whereas the MA1, 4, and 5 resistance shows resistance to kanamycin. MA2 only shows resistance to neomycin, which are moderately sensitive to gentamicin and kanamycin. The bacterial strains MA3 shows sensitive to neomycin and kanamycin and were sensitive to gentamicin. Thank you.
Thank you, Abhisha.
Thank you, ma'am.
What made you select urine samples of only diabetic patients?
Diabetic actually is the most commonly disease we have found. So, urinary
UTI patients.
urinary
Taken directly urine samples of UTI patients, isn't it?
No, ma'am. We have just collected only the strains from the laboratory.
From the laboratory,
you collected the strains.
Okay. And the rest of the work you carried out in your own lab?
Sorry, ma'am.
Was it done in your lab?
Yes, ma'am.
With Klebsiella?
So you collected from Nagarkoil, right? Sample. But your college is in Tirunelveli.
No, sir. It's actually wrongly printed. It's in Kanyakumari.
Kanyakumari.
Kumarikoil.
You have biosafety 1, 2, 3. You are good because this is pathogenic bacterium, right? So do you have? I don't think you have an approval to work with the pathogenic in your laboratory, Klebsiella especially. Yeah. Please check. Okay.
Okay, sir.
Thank you.
Thank you.
Any questions from you?
Hello? Am I audible?
I'm
audible. See, you have procured the strain as such from the lab that you specified, right? Or was it the sample which you took it for processing and you have isolated?
We have just bought the strains.
So when you have already got a strain, then what is the necessity for you to go for characterization and again prove that it is Klebsiella? Because they have already identified and they have given you the strain, then what is the necessity to further work with it? Probably you could have directly done your antibiotic resistance. That is fine. But the characterization part and again doing all biochemical and morphological characterization, is it necessary, do you think?
No, ma'am. It's only for confirmation.
Already the lab would have confirmed it, then only they have given you the strain, right? Okay, so that was from my part. Otherwise, it's fine.
Thank you.
Thank you, Abhish.
The chairperson to consolidate and say a few words. I request Dr. Anuradha ma'am to come up on the stage, ma'am.
A very good afternoon to one and all here. I would like to thank the organizers of the conference for inviting us to chair this session. On behalf of Madam and Santosh, I'm here. I just want to consolidate what has happened now. We've had 6 papers being presented, and each, there was more paper, there was about 3 to 4 papers on related to bioinformatic works, and about 2 papers which were carried out in the wet lab studies were there. And to sum up the presentation, we had postgraduate students presenting the papers. It was excellent presentation by a few of the participants who had come here. And also, I was very impressed with a paper on MRSA, nasocamal pathogen. That was an excellent paper that was presented here. And overall, the session was would have been very informative for the postgraduate students and the other participants who've come to attend the conference. And I hope you had an insight of what the different fields of work is being right now carried out in different parts of the state. And this session should have been an eye-opener for you and also to give you insight into what sort of work as postgraduate students you can go forward with. Thank you once again. On behalf of our team, I thank the audience for your patient listening, because it's almost lunchtime now. And thank you once again.
I extend my sincere thanks to our respected chairperson, Dr. Kalina, co-chairperson, Dr. Santosh, and rapporteur, Dr. Anju, for accepting our invitation and for the smooth conduction of this session. I request Dr. Anuradha Ma'am, Head of the Department in charge, PG and Research Department of Zoology, to hand over the mementos to Chair, Co-Chair, and Rapporteur. Thank you, ma'am. Meanwhile, I thank the judge for paper presentation.
So thank you all once again. So we have come to the end of the paper presentation of the forenoon session. We are actually running short of time. We are going to begin the next plenary lecture by 2 o'clock. We have Dr. Anish Nair, the Principal Scientist and Head, Center for Genomic Medicine, NIMS Medicity Hospital, Trivandrum, who will be giving us a talk on genomics and healthcare, the way forward. So I request all the participants to please go have your lunch and get back to the auditorium by 2 o'clock. It's a kind request because if we are going to get delayed, all the other sessions will also get delayed, and we have our valedictory function at 4 o'clock in the auditorium. Research Department of Zoology to welcome the speaker.
Assalamu alaikum and very good afternoon to all. On behalf of the management, principal staff and students of Postgraduate and Research Department of Zoology, Justice Bashir Ahmed Said College for Women, it is my gratitude to welcome you all for the 5th plenary session of second day of International Conference on Frontiers of Biosciences in Sustainable Development. The resource person of the session is Dr. Anish Nair. Dr. Anish Nair is currently working as a principal scientist and head Center for Genomic Medicine, NIMS Medicity Hospital, Trivandrum. He works on the development and launch of new diagnostic and prognostic tests using latest technology in genomics. He is involved with funded research grants from national and international agencies, scientific documentation, in-house biomedical research, and knowledge management across various forums. His doctoral research was on genetic diversity of pteridophytes and its bioprospecting using alternate endomycophyte technology against colon adenocarcinoma. His post-graduation was in molecular biology from University of Madras. He also holds a postgraduate diploma in bioinformatics and master's in international business management. He is also an NAVL certified internal auditor and an authorized signatory under ISO for medical laboratories. He has also worked as a scientist and head at Biozone Institute of Life Sciences Research and Training Division of Biozone Research Technologies Private Limited. He also holds additional responsibilities such as adjunct faculty, adjunct assistant professor, adjunct associate professor in many colleges and universities in Chennai. He has been a consultant with various organizations in Chennai. He has also been an invited speaker and resource person for many conferences and workshops at various institutions. About his research experience, he has 24 research papers published in reputed peer-reviewed journals at national and international level, and 28 NCBI GenVac sequence submissions. Now I request Mrs. Asif Ahmed, Assistant Professor of Zoology, to welcome Dr. Anish Nair with a box of dry fruits. Thank you, ma'am. Thank you, sir. Now I invite Dr. Anish Nair to take over this session. Thank you all.
Good afternoon. Can I get a good afternoon back? It's not loud enough. Good afternoon. So, I assume you guys are not ready for sleeping. Okay, so in the next, my time has been really cut short, so I'll be rushing through the slides, but then I am trying to introduce the concept of genomics. Many of you must be aware of it, but many of you might have a lot of doubts. So a quick, I should say the fastest presentation possible. I have a lot of things to share with you. So we begin with understanding what is clinical genomics all about. So basically clinical genomics, if I need to use a definition, it is an emerging medical discipline that involves using genomic information about an individual as part of their clinical care. Now, this kind of, starts with, so when you talk about clinical genomics, you need to understand the basics of genomics, where we come back to the basic of life. What is it that I'm referring to? I'm referring to nothing but the DNA, something that all bioscience students are very familiar with, and I'm sure pretty of the others as well. So if we look at the structural organization of the individual organism, so a human has individual cells. Inside the cells, you have your chromosomes. These chromosomes are basically in the form of chromatin network. Only during your cell cycle, it condenses to give you the centromere, the telomere, and forms the chromosomes. Which are where the histone molecules are basically the molecules which compact and present the DNA. This chromatin network contains the DNA, and parts of the DNA code for a protein which we refer to as a gene. And these DNA is nothing but a polymer which is made up of your nucleotides, comprising of your sugar backbone and your nitrogenous bases and your phosphate molecules. Now, all of you are very familiar. I'm just running through the basics before I get into the applications of genomics. All of you are very familiar about central dogma of biology, the whole concept of DNA to RNA to protein, where we say DNA to DNA, that is DNA copying itself is called as replication. DNA to RNA is referred to as transcription. RNA to protein is referred to as translation. So that is what is happening here. So inside the cell, if you see, nucleus is the site for replication and transcription, while your cytoplasm, in the cytoplasm, the ribosome is the site for your translation. So that's how a normal cell works. Now, why I am telling you this, you will understand in the further slides, where we look at, because any change in this pattern at different levels will lead to diseases. Okay, and that is what genomics has a role to play in healthcare, by detecting any minute changes, any spelling errors that's happening at any of these junctures. So to understand the concept of DNA, gene, and chromosome, now, like if I ask you what is the DNA, you will say genetic material. That's what you've studied in your textbook. If I ask you a gene, you will again repeat that it's a genetic material. So does that mean that DNA and gene are the same? No, they are not. Okay. So DNA and gene are terms which are used interchangeably, but they are not the same. Okay. Now, for example, DNA are like letters. Can you tell me how many alphabets are there in English language? Louder. Anybody who has a different opinion? Well, I differ on that. English language has one alphabet comprising of 26 letters. Alphabet is a collective noun. So please brush up your basics. English language doesn't have 26 alphabets. It has one alphabet. Okay, so DNA are like letters. When a set of letters come together to give you a meaning, you call them as word. Similarly, DNA is nothing but a polymer, a set of nucleotides. When a particular fragment of that come together to give you a meaning, or codes for a protein rather, it's called as a gene. And chromosomes can be something compared to as a book. You find a lot of words and spaces, etc. in books. Now, if you were to look at it in a little bit of detail, Can you read this? Since I'm running short of time, I'm not going to take it too interactive. This doesn't make any sense to you, but all you recognize is that it's a set of letters, right? From those letters, if we take a set of fragments here, right? Can you see it's AN? That codes for a meaning, right? It gives you a meaning. It's a word. HOUR, R, gives you a meaning. LATE gives you a meaning. So these are words. So similarly, this is a DNA fragment, and parts of them, when they code for a protein, we refer to them as genes. As simple as that. So coming to the human genome, 3 billion base pairs arranged into 46 chromosomes, information carried in pieces of DNA called genes, and the most important part that I wanted to share about the human genome is only 1.5% of human genome is protein coding, which we refer to as exons. You know, gene has two parts, introns and exons. Introns are the coding part and exons are the introns are the non-coding part and exons are the coding part. Now, why is this important in healthcare? Because most of your disease-causing genes are present, or disease-causing mutations are present in your exonic region. That is why when you go for a molecular diagnostic test, when you go for a whole exome sequencing, or when you go to any clinician, they first prescribe, or they tend to look at your whole exome sequencing. Why whole exome sequencing rather than a whole genome sequencing? Because whole genome sequencing is of course more expensive. Secondly, about 99% of your mutations, disease-causing mutations, which in technical language we call it as pathogenic variants, are present in your exonic region. That is in your coding regions. So that is why we look at the whole exomes for any disease diagnosis. Now all humans, okay, I'll come to this. To understand the difference between, all of you must have heard about genetics, all of you must have heard about genomics. You think it's almost one and the same, but no, it's not. Okay, there's a lot of difference between genetics and genomics. Now, the study of an organism's complete set of information is called as genomics, whereas the study or function or composition of individual genes is called as genetics. Now, to make it a little more specific, let's look at this. Now, I've already explained what this is. Now, your genetics is basically the study of single genes, or the function of single genes. But when it comes to genome, you can compare the genome with that of an engine, of a car engine. Can you see here? A car engine comprises of a fan, an air filter, an exhaust, an engine block, all sorts of things, right? Now, this is not just a set of one thing. It's a combination of all put together, right? So that's how your car engine works. Now imagine any of your one part, for example, the air fan stops or the air filter stops in a car engine. Do you think the car engine will be able to work? No, right? Now that is what is genomics, okay? Where we detect what is not working, and that is what we call it as variant, okay? It can be an external addition, deletion, all of that, we'll come to see it. So that's the difference between genetics and genomics. Understand that genomics is a broader view, and that is why it has more implications than plain simple genetics. Of course, genetics is the basics, it is the predecessor, but an application of genetics when it comes to healthcare, you have to look at as a whole. So it's not only the single genes, but there are other factors involved as well when it comes to genomics in healthcare. Now, a quick look at the other set of basics. All of you know this is a chromosome which has a short arm and a long arm connected by a centromere. Your short arms are called as your P arms, and your long arms are called as your Q arms. P here, actually, it's easy to remember. If you remember, P equals to petite. Okay, petite means small. Right. So if you see here, now why is this important? Because when it comes to many of the oncogenomics, that is cancer genomics, reports that you may end up seeing, or if you get into genomics and diagnostics, you will have, or even for reproductive medicine for that matter, the main source of recurrent pregnancy loss is genetic, wherein you will have these kind of translocations in chromosome 16. Okay, so this is how a translocation looks like when you karyotype it, and that will be detected by 1p36, 1p36 and 13q14. Now this is why I'm showing you this is, that's how you have to interpret your translocation. That is, this fragment is coming from here, okay, and it is interchanged from the first chromosome and the 13th chromosomes are compared, right? So if you see, there's a translocation happening between the first and the 13th, okay? These are your usual, if you get into the field of genetic counseling or any of the allied health paths, and if you happen to come across, situations where you have to write your genetic history of your patient, then these are the symbols that you will usually be using. The square, the circle, and the diamond shapes. Now, all of this, I'm not going into detail, but then few things that you should know is the uncolored ones are the non-affected ones, the colored ones are the affected ones. And consanguinity that you can see a lot in, more in your South Indian population. So I currently work on the border of Tamil Nadu and Kerala. Technically it's Trivandrum, but then it's on the border of Tamil Nadu and Kerala. So there's a lot of inbreeding happening there. There's a lot of consanguinity there. So you get to see a lot of consanguineous families, couples there, okay? Now consanguinity is a major cause of genetic diseases, okay? Another area where you get to see a lot of consanguinity is Maldives. We get a lot of samples from Maldives as well. So if there's a stroke in between any of your, okay, by the way, I forgot to tell you, the square is for the male, the circle is for the female, and if the sex is unknown, it's a diamond. The strokes indicate that the patient is, the concerned person, individual is diseased. And, whoever is providing the information, we have an arrow mark towards the diagram. Then you have other symbols of, if there's a marriage or a partnership happening, then there's a link between the male and the female symbols, so on and so forth. If they are relatives, that is, if you have to depict consanguinity, then you show something which you will understand if I say as double bond, okay? 2 lines. Right. Before we proceed further, a quick look at some interesting facts about your DNA. If all DNA in your cells were laid out in a line, it would reach to the sun and back around 70 times. So if I were to stretch the DNA in your cells, you can imagine how tall you would be, right? Now, what is the reason that you are not that tall? Maximum of 6 feet or maybe 6.5 at the max. I don't see any 6.5. Now the reason is your histone molecules, the wonderful creation. Now how do you understand that? When you buy thread, okay, for stitching, when you buy thread, right? You know how long the thread is, right? But how is it put? There's a conical, there's a cylindrical board in between and it's swirled around it, right? So that compacts it. Similarly, your histone molecules, octamers of histone molecules are put together and on that the DNA is twined, okay? That is the reason such big DNA has been compacted into cells which are not even visible with the naked eye, okay? That's the eternal beauty of life. Now, another fact, your genome is only about 0.1% different from any other person's, okay? Now that means the person sitting next to you, even if they look very different, even if they are of a different gender, there's only 0.1% difference between you and that person. Your neighbor, whoever is sitting here, will have the very similar, 99.9% similarity between two of you, but that equates to 3 million differences in your DNA, okay? Similar yet different, something like India. Now, another fun fact, if you spoke 1 letter of DNA per second, 24 hours a day, it would take about 100 years to recite the entire human genome. That's how big we are, okay? 3 billion base pairs. How much of data are we generating? Sequencing one person's genome generates around 200 GB of data, the capacity of a typical computer. So you can imagine, now we are sequencing genomes on a daily basis for diagnostic purposes. So you can imagine the amount of data that we are generating on a day-to-day basis. We as in anybody who is into the genomics field in healthcare. Some serious genetic conditions such as sickle cell diseases and achondroplasia are caused by a change to a single letter out of DNA of 3 billion. Out of 3 billion DNA letters, a single change, a T to a A, can cause a very major disease, as major as sickle cell. Why sickle cell? Even cystic fibrosis for that matter. Your CFTR gene, if there's a single gene, a delta 405 change, that leads to cystic fibrosis, and all of you know the consequences of cystic fibrosis. So that's how amazing genetics and genomics is. a very interesting field, especially for bioscience candidates. So please make sure you get into the depth of it. So genomic diagnosis of human disorders is referred to as the detection of various pathogenic mutations in DNA and RNA samples in order to facilitate detection, diagnosis, subclassification, prognosis, and monitoring response to therapy. So when you talk about genomics in healthcare, it's a whole cycle. We are not just detecting the disease. All these are possible. Detecting the disease is one of the aspects, one of the applications possible through genomics, right? So that is what is the reason of the genomics industry booming up, especially post 2020. Also, expand scientific knowledge of the natural history of diseases, identify people who are at risk for acquiring specific diseases, that's prediction, and diagnose human diseases at the nucleic acid level. Now, when you talk about the genomics per se, the uses, where are the uses that in generally you see? You can do a better diagnosis, you can do a better prognosis, you can avoid adverse events, you can optimize the treatment selection, especially when we are talking about modern day oncological treatment. Modern day cancer treatment does not follow one drug fits all concept. We have moved to precision oncology, where you look at the genomic signatures of the tumors and give the drugs according to that. It is no more, okay, you are, okay, XYZ is a cancer patient, has breast cancer, BRCA positive, give the drug. No, doesn't work nowadays. Better disease monitoring and effective clinical trial participation. So how does it begin? It begins with the genomics in the clinics, and that is basically understanding the genome of the index patient, then going to the patient and the families, describing the phenotype. Very important is to clinically correlate your genomic data with your clinical manifestations, which we refer to as the phenotype. Then identification of the genes, which are I can't say genes, the errors in the genes which are responsible for the characterizing mechanism from the DNA to the phenotype. That is what is the entire genomic medicine cycle. Now, where are the applications in terms of diagnostics of genomics? You can find genomics having applications right from pre-conception stage. Even before a couple gets pregnant, they can use genomics from that stage itself. So pre-conception, post-conception, artificial reproductive technology, pre-implantation, prenatal, newborn screening, adulthood. The entire life cycle has applications by genomics, okay? So if there's a preconception couple coming to, in fact, nowadays, I remember when a couple of months back when the sequencing costs were reduced, I had actually put up on my status saying that why are we going to match horoscopes? We should be matching genoscopes now to avoid genetic disorders. So genomics basically has a huge impact. Now how? For example, if there's a couple who's planning for conception coming to the counselor or the clinician, what we do is we basically understand the genetic history of both the the male and the female counterparts, do a whole exome sequencing of both the future parents, followed by the karyotyping, couple karyotyping. Now, all this will tell us if there are any spelling errors in their DNA or the structure or function of chromosomes. Anywhere, any error. If it's the same error matching between both of them, then we tell them it's a high-risk pregnancy if in case they get pregnant, okay? So what's the solution in those cases? You move on to artificial reproductive technology, what you commonly call as IVF or very commonly or layman language, test tube babies, right? Now, even during that, even if you're going to opt for an ART technology, we suggest going for PGS, which is pre-implantation genetic screening. Suppose you have 10 embryos, okay, getting conceptualized in the lab, the zygote formation happening in the lab. If there are 10 embryos, they can undergo a sequencing. All of these embryos can undergo sequencing by a small fragment of the cells. Check if there are any spelling errors in them, and then we select which embryo can be transferred and which cannot be transferred. Okay, so that's pre-implantation genetic screening. Now, after that, once you, in case the couple is normal, then we ask them to go ahead with a normal pregnancy. Then next comes, once the lady is pregnant, within 9 to 20 weeks of gestational period. Okay, that is 9 to 20 weeks of your pregnancy. We ask them to go for NIPT, which is non-invasive prenatal testing, wherein just by drawing 10 ml of the mother's blood, you can understand if there are any spelling mistakes, any genetic diseases in the baby. That's called NIPT, non-invasive prenatal test. Now, how do we do that? There is fetal DNA circulating in the maternal blood. That is what we harvest and we analyze that. So that's NIPT. Now, why NIPT between 9 to 20 weeks? Because as per the government of India, if you have to go for an MTP, that is if you have to abort the fetus due to medical reasons, then you should do it before the 24th week. Okay, so that is why the NIPT is usually suggested between 9 to 20 weeks of pregnancy. Now, even after that, once you're continuing and then, postnatal, that is when the baby is born, you can do a newborn screening to again verify if there are any genetic errors in its entire sequence. Okay, that's called newborn screening. Newborn screening can be done with biochemical tests as well as genomics test. Now, this is still from the time the planning for conception till the birth. Even at any stage of adulthood or childhood, an individual can undergo a genomic test, and genomic tests are available across every discipline of modern medicine known to mankind. Cardiology, neurology, nephrology, anything, everything, we have answers in genomics. So preconception and prenatal testing, newborn screening, disease susceptibility. Newborn screening or even your later stage screening can also predict if there's going to be, if there's a possibility of the person having a genetic disease. Okay? Why is that important? Because you can delay the manifestations or you can manage the disease better. Screening and diagnosis, prognosis and therapeutic decisions, monitoring the disease burden and recurrence. All these has applications through genomics. Now, how do we look at genomics on a larger scale? The individual is in the center. You acquire the genomic data, you acquire the health data, you acquire the lifestyle data. Now, somebody had asked me yesterday during the panel discussion about the wearables, the health wearables, health monitoring wearables that you wear. Now, this is where that role comes to play. Everything in modern day is about data, right? So genomics alone cannot stand. Genomic data is not enough because we are, I'll show you that, we are as human beings, we interact with a lot of things. It's not only the genetic information passed on to us from our parents, which is going to manifest into disease or tell us if we are good at something or bad at something. The environment also matters, right? So that is why this health data or the lifestyle data plays a lot of role, which is being contributed by the wearables, mostly the wearables. Okay, how does that help? Decision making, research and education, health technology, evidence-based healthcare. These are the applications where we come to see. Yeah, so this is what I said. The human body acts as an integrated system. Why? Because not only is it getting the genetic inputs, it is also getting inputs from environment, nutrition, medications, interactions, so on and so forth. Okay? Even temperatures. Okay? Even temperature changes, climate changes, everything affects us in some or the other way. Okay? So that is why over the period of time, since our body has been integrating all these inputs, we get a lot of response in form of DNA changes, gene expression changes, and we also get to see changes in the effect of small molecules. Now, how can we use genomic information from an infective organism? We can look at the genomic information from an infective organism across the population to track epidemics. I think this I need not tell any one of you. All of you would have been reading in your daily newspapers how genomic surveillance was used to track the evolution of SARS-CoV-2, which you refer to commonly as the COVID-19 virus, right? So the SARS-CoV-2, the first variant, in fact, there was a paper early in the morning session when I walked in. There was a student who presented about the variants of SARS-CoV-2. So the first SARS-CoV-2 variant, which we refer to as the alpha variant, B1.1.7, from that to the Omicron lineages that we have seen now have been evolving, right? So all that could be tracked, all that could be used for the better applications for mankind only because of the genomic surveillance. Imagine if this was not available, if the genomic surveillance was not available, what would we have done? Every day we would be facing a new COVID strain, and we don't know that it's new, we don't know it's old, we don't know what it is, and we're just facing. It would have been like the earlier pandemics where there was no way to combat it. So hats off to people who have come out with this, and hats off to people who have applied this, and that is the reason we all were vaccinated and we were able to combat pandemics. And thanks to genomics, I should say, we are sitting here without masks again, right? Otherwise, imagine, first of all, this conference would not have been held. Even if it would have been held, Mubin ma'am would have saved a lot of money by making it online. And to generate faster and more accurate diagnosis, to choose the target treatments, that is from an individual. So at an individual level and at a population level, the applications of genomics clearly has its differentiation. Now, to personalize treatment and surveillance, we can use genomic information from an individual, from a person's tumor, or from an infective organism to develop new treatments based on the effect of genomic changes. Okay, a constant monitoring, a constant tracking of all these levels can easily give you a better solution and help you with therapeutic decisions. Now, how do we compare genomic information to identify genetic alterations? You have a cohort of patients without the condition, people with the condition, you compare their genetic informations and come out with the genetic alterations which is getting associated with the disease. Now, quick look at the history of molecular diagnostics. Started at 1865 with, of course, Father Gregor Jorgen Mendel giving us the laws of genetics. From that, we moved on to purification of DNA by measure in 1866. The sickle cell anemia mutation, of course, 1953, a milestone year when Watson and Crick gave us the DNA structure, double helix, rDNA technology, Sanger giving us the sequencing method in 1977, PCR given to us by Carey Mullers in 1985, and of course the human project, genome project starting in 2001, and from 2005 to 11, the sequencing technologies and genome sequencing, the advancements in that. Now, for your kind information at this juncture, I don't know how many of you know it. I would like you to put up your hands if you know who won the 2022 Nobel Prize in Physiology or Medicine. Can anybody give me the answer to this? Shortage of time, I'll tell you. It was Svante Pabo for his work on paleogenomics. Okay, please go back and understand what's happening in your field to be aware of it. A very common GK question. So genetic testings come in three categories. It can be predictive testing, where you can say if the next generation is going to inherit it or not, or even if the generation is there, then are they going to be put at higher risk for a disease later in their life? Newborn screening detects immediate diagnosis of any newborn errors. Carrier screening, as I said, the preconception screening. So even if the person as such per se has not manifested the disease, but could be a carrier of a pathogenic variant in its own genome, which can get passed on to the next progeny. Now, as I mentioned, going back to the central dogma, the levels of genetic testing, you can compare your DNA, you can compare your proteins, and you can compare your protein function. So when you're talking about comparison of DNA, the genetic material, we can do it two levels. One is the chromosomal level and one is at the sequence level. For the chromosomal level, you use the cytogenetic methods. For the sequence level, you use the molecular biology methods. For protein shapes and protein function, you have proteomic tests, especially MS-based tests. So what is a mutation? We have been hearing about mutations, SNP, CNVs, DELs, etc., etc. But what exactly is a mutation? A permanent alteration or an inheritable change in the nucleotide sequence of the genome is referred to as mutations. Two types. It can be germline or somatic, as I was mentioning yesterday. Germline is something that we inherit. Somatic is sporadic, which we gain after the embryo has been formed. Okay. Now, how can a person get somatic mutations? errors during replication, DNA repair, chemical mutation, physical mutation, exposure to radiation from UV rays, X-rays, and extreme heat. All these are ways in which you get your somatic variants. So if you see here, your germline variants are present right in your gametes, and it gets passed on, and the entire all the cells in the body basically will have the germline mutations. However, your somatic variation is absent in the gamete. It develops either during your embryo stage, your fetal stage, or during your childhood, or during your adulthood. And if you see, it's not, we call these as mosaic individuals. That is, they basically are not present. These mutations are not present in throughout the body. It is very localized. Now, if you look at the types of mutations, if you see here, the cat ate the rat. Now, this is a normal gene sequence. Okay? Suppose there's a substitution. The C has been changed to B. Now, that's called a single nucleotide substitution. So, the meaning changes, right? The cat is now the bat. Again, if it's an insertion, suppose I insert an S in between, the entire thing changes. The entire frames changes. Now, I can't even read this properly. It doesn't sound English to me. If there's a deletion, similarly, if this A is dropped, then this changes the, and then I can't read, right? So all these are types of mutation: substitution, insertion, and deletion. Now examples, so if you see the screen, you can see a normal looking DNA. If you see, there is a single base pair. I don't know if this is visible. There's an A which has turned red, which was G earlier. Okay, so this is called a single base pair mutation. A classical example of single base pair mutation is sickle cell anemia. This is a deletion, where you can see a deletion, as I mentioned. A classical example is cystic fibrosis. This is a major deletion, where you have your dystrophies, your DMDs, your BMDs, your Lager-Girdle syndromes. All of them are because of the larger deletions. Insertion, if you see, there's a set of A's which has been inserted here. And now that's called. Thank you. A has been inserted here. So this repeats are responsible for Huntington's disease. Multiple mutations at multiple sites lead to diseases like diabetes or breast cancers, etc. Now let's look at how do we look at the DNA sequence. I told you there are multiple ways of looking at DNA sequences. The first and foremost, something which we know very well, is your polymerase chain reaction. Then you have your RFLPs. Of course, polymerase chain reaction comes with all its variants. DNA sequencing, Sanger sequencing, microarray, next generation sequencing. These are different technologies that you use to see, that are used to look at the DNA, rather read your DNA. Now this is PCR, a polymerase chain reaction. As you can see, there's unwinding happening, binding of the nucleotides through the polymerase, and then extension. So this is a classic example where PCR, which is nothing but, as I mentioned yesterday, poly means many, mer means unit, ace means enzyme. So polymerase is basically an enzyme which makes many units. Chain reaction, a chemical reaction, which happens in a continuous manner. So that is what is polymerase chain reaction, makes multiple copies of your DNA. Classical example, melting happening, the double-stranded DNA getting separated, annealing, that is the binding of the primers at a set temperature, and then elongation or extension happening at 72 degrees. Again, this cycle continues, they break down again, they denature again, and then there's attachment again, then there's amplification again. So that's how you get multiple copies of DNA in a PCR. Now, how do we use PCR for deletion? How do we use an endpoint PCR? Now, endpoint PCR, or the basic version of the PCR, can be used to detect deletions. I told you there are multiple diseases which are caused by single deletion. So how do you do that? You design your primers accordingly. Okay, if this was the normal gene, and if this is the mutated gene, you can see there's a deletion. So you design primers according to that. You design your normal primers for your normal gene, and you design another set of primers for the mutated gene. So when you amplify and then when you electrophorese it, you will see there is a difference in the PCR products from your normal gene and from your mutated gene. So that is how it will look, right? A normal gene will be, will have more molecular weight than a mutated gene. So that's how you separate. Of course, you know about gel electrophoresis, and this is how your fragments can help us. So this is basically was to explain the concept of gel electrophoresis. Now, how does DNA sequencing helps us, or what is DNA sequencing? This is the control page, index reference data, okay? This is how your results of your sequencing comes. Now, what we do is, we, we take the, we need to understand, or we need to decode the patient DNA sequence to check if there's any mutation present in the, sequence of the patient. So how do we do that? Basically, denature the double-stranded DNA using heat, make multiple copies, attach primers, add to 4 polymerase solutions, grow complementary chains until termination die, denature the growth chains, and electrophorese the 4 solutions. That is, you basically have the unknown sequence, and the sequencing is done using multiple labeled termination added solutions. So what happens if you see here, the first one stops at C, the second one stops at T, the third one stops at G, the next C, A. Now this is what is getting detected, okay, because all of them will have different colors. Your C's have green, your T has purple, your G has yellow, and your A has orange, right? So similarly, this is how it's done. And then these products are electrophoresed, not using a regular agarose gel electrophoresis. We use a capillary electrophoresis, which that's why you see a sequencer machine, the most classical machine being the ABI 3500. And they separate because of the different sizes, okay? And that sizes basically is, the different fragments is detected by the fluorescence detector. If you see, the first one shows A, then C, then red is T, then A, C, so on and so forth. So that is how you sequence the DNA, or that is how you basically look at the sequence of the DNA. Then what do? You have already got the, the known gene sequence, and now you know the patient sequence, okay? Then you compare both and realize that yes, there is a change. The known one had G in it, while the patient has C in it, this is how you detect your mutations. Now probes, basically based on their complementarity, are used, I'm really rushing through it, are used in experiments called as microarray, where microarray basically is a set of probes arranged, spotted in a big chip. And then each of the wells have their own probes which detect different set of mutations. So why is this very important? Although this is expensive, but then this is very important in modern day diagnostics, because you can detect multiple variations at the same time. For example, here it's binding to a cystic fibrosis mutation. Here it is with the colon cancer. Here it is for a breast cancer. So in the same microarray chip, I can have parallelly analyzing the probable genes or the pathogenic mutations. Next generation sequencing, massively parallel sequencing technology that offers high throughput, scalability, and speed. And with last week, MGI coming out and declaring that the cost of a human genome is going to be lesser than $100, which is currently at approximately $200. I think that is the next thing that we are all looking forward to. 5 years before, or maybe 4 years before, we, for a whole exome, the charges used to be in lakhs. Now we are talking about a whole exome at 14,000, 15,000, and that is the reason that the whole exome, or the next generation sequencing, has invaded into the clinicians' prescriptions. So what happens? Why are we comparing? Why are we preferring NGS rather than Sanger? Because Sanger is one sample, one amplicon, and one sequence. NGS is multiple samples, hundreds and billions of sequences and bases. Okay. So at the same time, when you can get to see the whole picture, why only see the trailer? Right? That's the concept. Why we are preferring Sanger, NGS to Sanger. But of course, Sanger is much cheaper. NGS is expensive. But if you are lucky enough, if the clinician is lucky enough, if he's looking at a single mutation and the first Sanger gives you the result, fair enough. Otherwise, it will lead to a battery of visits to the clinician, a battery of visits to your lab, and I think that basically agonizes the patient. So depending on the patient's clinical manifestations, the clinician prefers to choose between Sanger or NGS, and of course looking at the patient's pocket. Analysis pipeline: extract the DNA, library preparation, sequencing it, quality check, alignment, coverage, variant detection, and copy number variant detections. So this is how your sequencing works. Okay, I'll quickly skip this. Yeah, so that was about the the regular diagnostic aspects of genomics. Towards the end of this, I'm just briefing out what are the other possible applications. So how can genomic information to help to develop new treatments? Okay, now this existing drug interacts effectively with the protein, right? If you see, this is the protein and this is the drug molecule, both can interact. Now when you have an altered gene and an altered protein, this altered protein is not able to interact with the same drug molecule. So what do? We get the sequence, from the sequence we predict the structure of the protein and then design the pharmacophore accordingly, and based on the pharmacophore, we either modify the same drug or we come out with new drug, or we screen for something we call as repurposing drugs, which fit into this interaction ability, and that's how you come out with your new drug. So this is another application of genomics in healthcare. Personalized medicine. I've got my warning chit already, so another 10 minutes, ma'am. Right, so personalized medicine is another application of genomics. The one drug fits all doesn't work anymore, so it's personalized medicine based on your genomic signatures of the individual. Now, for example, if you have a patient group and they are exposed to the drugs as required, they fall into 4 categories as for the analysis. The first one, where the drug is toxic but beneficial. The second one, drug is toxic but not beneficial. Third one, drug is not toxic and beneficial. Fourth one, drug not toxic and not beneficial. So depending on that, we can go ahead. Now, cancers, precision oncology is something which I wanted to talk for a longer time, but due to shortage of time. Different forms of cancers where targeted therapy is used. For example, if the patient, the tumor of the lung cancer patient has a tumor which is EGFR positive, you give an EGFR blocker for that. You do not need to look at other drugs. Increase the life expectancy of the patient. That is how you manage a cancer patient. So this is applicable to all of these cancers, not only the solid tumors, but your hemonc as well. I think towards the end, the other application is precision nutrition. Now, everyone responds to diet and nutrition differently based on factors including genetics, environment, exercise, microbiome, and health. So all these factors, okay, contribute towards forming a precise diet plan, precise lifestyle management for an individual. As I mentioned yesterday, precision nutrition is not only for weight loss, it can be used, which we call it as nutrigenomics, can be used for weight loss, to improve your skin, improve your immunity, predict your future possible genetic diseases, even predict cancer for that matter. All of that can happen through your genomics, and this is how it entirely functions. So it's basically genetics and epigenetics contributing towards a very personalized, custom-made, tailor-made diet plan. I sound like a marketing person. Okay, so good collection practices, all of genomics on one side, but make sure you do not make the mistake of collecting the wrong sample or collecting the samples in the wrong container or transporting it in a wrong temperature. All that matters. So depending on your samples, make sure if it's a blood, it's collected in the correct vacutainer. Your phlebos will be responsible for that, but then make sure that it's collected in the correct vacutainer, lavender, red, or green, depending on what test it has to go through. The container needs to be transported in the correct manner at the correct temperature. If it's a biopsy sample, then it's a tissue block that needs to be collected. Very, 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 very vital and important is your consent. Okay? Voluntary informed consent needs to be taken from the patient or the bystander or the guardian if the patient is a minor. Okay, without that, never ever accept samples for any kind of molecular diagnosis. You are dealing with very, very intricate, very, very sensitive data. Okay, now the next, a very controversial part of it, I am not getting into what it is, but then it is about data management. So the genetic data that we are getting from the individuals, where does this go, who manages it, what is the potential applications in the future, if somebody turns wicked, how can they use it? All that is very important, keep that in mind. That's a huge debate in itself. Now this is for somebody who had asked me yesterday the question on artificial pancreas when I mentioned it. So this is how it works. You basically have the sensor which keeps on monitoring your random blood sugars, which we call it as the continuous glucose monitoring, connected to a pouch of your insulin. So as this falls low, there's a beep happening on your smartphone, and this injection goes in. Okay, so this is how the artificial pancreas work. Last part, just before concluding, just to introduce where I come from. Right now, I'm at the NIMS Center for Genomic Medicine, Neyatingara, Trivandrum, Kerala. We are approved by Kerala government as the only Center for Excellence in Life Sciences, NABL, NABH, NACR, BHR, and ICMR. We have 4 verticals of consultation, diagnostics, training, and research. A lot of new age infrastructure, starting from automated nucleic acid extractors to next generation sequencers. Under diagnostics, we cater to infectious diseases, genetic disorders, reproductive medicine, oncology, transplant, immunology, autoimmune diseases, genomic wellness. Under our training division, we have options of hands-on training, academic dissertation projects, internships, workshops, value-added courses, research and analytical services, National Council for Vocational Education Training Certified Biotechnology Finishing School, and Faculty Development Program. A quick look at the facility. Thank you. I think I did a Shankar Mahadevan step today. My thanks is to the management, the Department of Zoology, Principal Ma'am, Amtul Ma'am, and a wonderful team at Zoology Department. I'm not new here, but then special thanks to Mobeen Ma'am for inviting me to this conference and giving me an opportunity to interact back again with these students. And special thanks to Dr. Lucky for having coordinated all of this. And on this note, thank you so much. I am open to questions, but I don't think Mobeen ma'am would like that. So it depends on her. Thank you. Is there any questions? I think by the time. Oh, yes. Go ahead, go ahead. With fingerprints? You're not audible. With fingerprints? So it's not with fingerprints, but with your cuticles, yes, that's possible. Just with the fingerprint, suppose you take the fingerprint onto, for example, a sellotape for that matter. In case that's done, only if your, the cuticles or the first epidermal layer, sticks, or some cells stick to that particular thing, from that you can, theoretically, nobody has done that, but theoretically that can be done. But just with the fingerprint, no, that's not possible. The reason is the cuticle will have some fragments of DNA in it. So that's why. All right, thank you.
Assalamu alaikum and very good evening to all once again. It's my pleasure to thank Dr. Anish Nair, the resource person of this session. Sir, your lecture on genomics in healthcare, the way forward, was very interesting and much informative. It enlightened us on basics of genomics and its various applications in healthcare, such as molecular diagnostics, which is a rapidly growing field, utilizes molecular techniques to analyze biomolecules and diagnose diseases, precision oncology, development of new cancer therapies, pharmacogenomics, diagnostics of infectious diseases, and finally, the benefits and challenges of genomics. Thank you once again, sir, for your impressive and lucid presentation and made the session alive. Now, I request Dr. Mubin Sultana, Assistant Professor of Zoology and Organizing Secretary of this conference, to hand over the memento to Dr. Anis Nair as a token of appreciation. Thank you, sir. Thank you.
Thank you, ma'am. Before we start the paper presentation, it is indeed a proud privilege to announce that the Postgraduate and Research Department of Zoology of Justice Bashir Ahmad Said College for Women will be signing an MOU with NIMS Center for Genomic Medicine, having its campus at NIMS MedCity, Thiruvananthapuram, right now on the dais. So I request Madam Principal, Dr. Amtul Aziz, Dr. Anuradha, HOD in charge, and Dr. Kanis Fatima, Assistant HOD, to come up on the dais. Now we shall exchange the MOU between the PG and Research Department of Zoology and NIMS Medicity Kerala. A proud moment for the department. Celebration continues. Thank you, ma'am. Thank you, sir. We request the chair, Azra sir, co-chair Dr. Jayanti ma'am, and the rapporteur to please come up on the dais. Dr. Sumita, yeah, please.
There is an announcement. I request the registered participants to kindly fill their feedback forms and hand it over to the volunteers. I repeat, I request the registered participants to fill their feedback forms, which is there in your folders, and please hand it over to the volunteers. Thank you.
Assalamu alaikum and very good evening to all. It's my immense pleasure to introduce the chairperson of the fourth technical session, Dr. Azra Sharif. Dr. Azra Sharif is an associate professor and head of Department of Zoology, the New College, Chennai. He completed his undergraduate degree in zoology in the year 1988, post-graduation in the year 1990, MPhil Zoology 1991, and doctoral degree in zoology in the year 2013 from the New College, University of Madras. He has 25 years of teaching experience and his specialization include fishery biology and aquaculture. He has participated in various training programs, conferences, seminars, and workshops at national and international level. He has published 35 research papers in reputed peer-reviewed journals. He authored 5 books and 6 chapters in books. He has been a resource person in many workshops, seminars, and conferences. He also received awards at national and international level. He was a reviewer of Asian Journal of Fisheries and Aquatic Research, editorial board member for International Journal of Scientific Review and Research in Engineering Technology, member for Board of Studies in Zoology in many colleges of Chennai, like WCC, Ethraj College, JBS College for Women. The co-chair for this session is Dr. J. Jayanti, Associate Professor, Department of Advanced Zoology and Biotechnology, and Dean of Research, Guru Nanak College, Chennai. She has 23 years of research experience and 22 years of teaching experience. She has published more than 105 research papers in reputed international and national journals and authored 4 books. About her achievements, she has received many awards. To mention a few, Young Scientist Award from Tamil Nadu State Council for Science and Technology, Fellow Award from Society for Applied Biotechnology, Srimati Amrita Devi Bishnoi Medal for Environmental Studies. She is an editorial board member for more than 4 international journals. She is also a member of Zoological Society of India, Society for Life Sciences, Indian Association for Science and Nature. Dr. Jayanti has also organized many seminars, conferences, and workshops. 20 PhD students and 13 MPhil students completed their degrees under her guidance, and currently 6 PhD scholars are pursuing their research under her guidance. The rapporteur of the session is Dr. J. Sumita. She is an associate professor of postgraduate and research department of applied microbiology, JBS College for Women. She is an associate editor for JBS College magazine from 2014 to till date. She has participated in many conferences, seminars, and workshops at national and international level. She also presented research papers in national and international conferences and seminars. Dr. Sumita has published 32 research papers in reputed and peer-reviewed journals at national and international level. She also received MRCC project grant under Capital Assistance Scheme for her project funded by MRCC of JBS College. Now I request Dr. Gausia Nisha, Assistant Professor of Zoology, to welcome the chairperson, Dr. Azra Sharif. Co-chair, Dr. Jayanti. And rapporteur, Dr. Sumita, with a box of dry fruits. Now I request the panel members to take over the session. Thank you.
This is for the benefit of the presenters. The presentation timing will be for 5 minutes and 2 minutes will be for the interaction session. Please maintain the timing. The first presenter is Brinda and Bhavani.
Good morning to everybody present here. Myself A. Brinda. I am from Queen Mary's College. I am going to present about the topic butterflies of Adaya Eco Park based on their abundancy and status survey. Butterflies are plant dependent and beautiful flying insects that are included in the second largest order Lepidoptera. Among the order Lepidoptera, about 1,50,000 species are recorded, and in butterflies 18,000 species are present, and the rest of them are moths. In India, about 1,504 species are present. In Tamil Nadu, about 325 species of butterflies are found. They are the prey for the certain migratory birds, reptiles and predatory insects. These are very sensitive creatures in which they react quickly to any kind of disturbances like changes in the habitat quality, climatic changes such as temperature, humidity, rainfall, availability of larval host plants. Present study work was carried out to understand the butterfly abundancy checklist, host and nectar plants, and status survey for about 72 species belonging to 5 different families such as Nymphalidae, Pyridae, Lichenidae, Papillonidae, and Hesperidae from January 2021 to December 2021 at Adaya Eco Park. Objective of my study, butterfly abundancy was I have done with the help of the line transect and direct visual counting method during the study period. Some of the host plants and nectar plants observed, identified, and photo documented. Study area, my study area is situated near the Dr. Ambedkar Law University. The total area is of about 358 acres, and only 58 acres are considered as eco-restriction. This is the map. Line transect count method was used to determine the butterfly abundancy. Survey was taken for 4 times per month, and these are the 3 transects: round transect, straight, and curved transects during the hours 10 to 11:30 and 3 to 4 p.m. Butterfly species were identified, and unidentified butterflies are identified with the help of the standard literatures and field guide book of Indian butterflies, Himkar, Evans, and Winter Blythe. Plant species are identified with the help of the PlantNet identifier. Butterflies observed are divided into 4 groups based on their abundancy and sightings: very common, occasional, and rare during the study period. These are the butterfly species. This is common, very common species, these 3. And this is striped tiger. This is chocolate pansy, small salmon Arab, common emigrant, cyc, common cerulean, lime blue, forget me not, gram blue. This is dark grass blue. This is this is gram blue, common mormon, common jay, blue mormon. This species is very rare, but in my study area I found this species very common. This is common lime, small branded shift, and grass demon. Actually, some of the rare species I have recorded in about 2 families: Nymphalidae, glassy tiger, blue pansy, and Lichenidae, but I can't take pics. This is family-wise distribution. This Nymphalidae accounts for about 30.55%, and followed by Pyridae 20. This is the family-wise distribution chart. This is current status. In that, very common species includes 30, and common 22, occasional 14, and rare 6. Conclusion from the study area, the present study includes 72 species of butterflies belonging to 5 families. Family Nymphalidae is more dominant with 22 species, followed by Pyridae with 20 species. Relative abundancy is more higher in Nymphalidae family with 30.55% compared with the other families. So from this, I conclude that ample of larval host and nectar plants are present in the large amount, so that large amount of butterflies, individuality of butterfly species gets attracted towards this region, and not alone ample of larval and host plants present, but also availability of water quality, sunshine, and calm atmosphere, and good climate also will attract the butterfly species butterfly species. So it is increased in that my study area. So the present information is useful for planning conservation measures in the Adaya ecopopulation. Thank you.
It's open for discussion. Any questions from the audience?
There's no question from the audience. I think we'll have to ask a few. Yes, sir. Yeah.
Yeah. Yeah, I can, but thank you, sir.
What sir says is try to correlate. You have studied abundance and all that. Try to correlate it with some of the important ecological indices that sir has mentioned. So were you able to correlate with any factor? You have mentioned that something is abundant, something is very less, and so on. So what do you feel is the reason for that?
Actually, butterfly individuals,
All are butterflies only. What is the reason for particular family being abundant and
can't understand, sir. Because for that particular species, larval and nectar plants are available in more number, in more amount. So it's get attracted. Actually, for overpollution, overpopulation, habitat fragmentation is the main cause for the butterfly uh, extinctions. So that for conserving that, I have done this method.
Adayar Eco Park? I don't think there is. It is a protected area. So where is the question of overpollution and all?
Maybe in future. Thank you.
Next presenter is Rakshanda Anju.
A very good evening to one and all present here. I am Rakshanda Anjum. I am here to present my paper entitled Comparative Study of the Aquatic Avian Diversity between Pulicat Lake and Adiyar Park. So we all know wetlands are doused with water. They are basically saturated with water. If not saturated, there is water in and around the wetland. That's why it is called a wetland. So wetlands are very important ecosystems in the environment. They play a vital role in controlling the bird diversity. Since wetlands do not have large predators, these birds they have a lot of space to stay in the wetland, and they breed, they reproduce there a lot, and they also have their nesting sites. So the aim of my project was I've taken two wetlands, one being Puliket, which is an unrestored one, which is not restored and it's accessible to everyone, and the other one being Adyar Eco Park. It is a restored wetland where the public is not allowed inside. It is a protected area. So I wanted to check the diversity between a restored and an unrestored wetland, so that what can be done further to control and to increase the diversity. So that was my aim. So I studied the diversity separately in Puliket and in Eco Park, and then I compared the diversity between both of them, and I also compared the checklist that I got with the previous work which has been done in both Puliket and in Adyar Eco Park. So materials and methods, I used point count method to do the survey, and the timings were early hours of every morning, that is 6 to 9. I spent 3 hours there in Puliket as well as Eco Park. It was not done simultaneously, but it was done one after the other. And in the evenings, around 4 to 6 p.m., the timings were like that. And this was the binocular used. And there were also another part of my project was to do the water quality analysis. I didn't take much complicated parameters. It was just pH, temperature, and salinity, just to check if this depends, if this is interrelated with the diversity that is present in both the wetlands. So this is Tolkapi Punga, the map, aerial map. So I took 4 sites in the park. Some of the areas are restricted. You're not allowed there. So actually, when I did this project, it was during the COVID. So to get the permission and to enter the park itself was such a big hassle. So I had to take 4 sites there. These were the first 2 sites. Actually, Adyar Eco Park has both freshwater and brackish water ponds. Like the ecosystem is like a Coromandel coast. They have mangroves, they have freshwater ponds, they also have brackish water where the seawater is connected to the lakes. So there's a passage like that. So this is brackish water. These 2 sites were freshwater sites. Coming to Puliket, 6 sites were taken since the area is vast, and mostly in Puliket people do the research in the sanctuary. So there is a lot of research done in the Puliket sanctuary, but the areas I chose, there has not been much work. The first 3 sites were in the MCC laboratory, which were mud flats, and the 4th, 5th, and 6th sites that was near the Puliket fish market. It was near the sea, so it was close to the sea. So these were the sites in the laboratory: Site 1, Site 2, Site 3. These were all mud flats. There was also less water during my survey, and the 4th site was near the Puliket fish market. 4th, 5th, and 6th. So results. Coming to the results, since Puliket is a vast area, and we found 22 bird species present in Puliket, out of which 5 of them were near threatened species. Spot-billed pelican, painted stork, Eurasian curlew, and curlew sandpiper. So 16 of them were least concern, and we found this great knot, which is claimed to be endangered by IUCN. It was in 2013 they claimed it to be endangered. So these are the 5 near-threatened species. So great knot. These actually in Puliket we all know it's more of this lab is mud flat, so we can see more of waders there. Instead of when the water is shallow, divers and swimmers are not much present there. There are only more of waders, so we could see all these birds there. And near the sea area we saw tern. And in the Adyar Eco Park, 20 species of birds were sighted, out of which again 5 near-threatened species were there: painted stork, stork-billed pelican, oriental darter, Indian spot-billed duck, and cinnamon bittern. So these 2, Indian spot-billed duck is very much present only in Indian subcontinent, and it's also seen in Pakistan as well. So this is a completely non-migratory bird, which was seen in the Adyar Eco Park. And Indian cormorant, this moves to the neighboring country, migrates, and it comes back. So these were all the birds: herons, These were seen on the mudflats. So comparing the results, duck species were high in the Adair Eco Park where there was fresh water, and in Pulicat, since it was brackish and the salinity was high, the count of the duck species were low. And next, waders were high in Pulicat. In Eco Park, it was less. And coming to herons, egrets, both had equal diversity, no much difference. And coming to darters, we could see darters, like a lot of darters in Adyar Eco Park than in the areas in Pulicat where I studied. So I did basic water quality analysis. So comparing the results with the previous papers, in Pulicat, the average temperature was high. And even in the Adyar Eco Park, comparing the previous studies, the same, the temperature was high and pH levels were high in Pulicat, but comparing the previous results, it was normal, it was average. Salinity was almost the same, no much difference in salinity. So discussion. Actually, in 1994, there's a paper by Krishnan. It was stated that there were 175 bird species in Adayar estuary. But now I studied only a part of the Adayar Creek, not the whole thing. But this survey was done for around 6-7 months. That's why I couldn't do the indices. So out of 175 species, we only found 20 species now, which means the diversity is decreasing. It is actually because of the drainage water inlet into the Adayar River. So whenever I went to the Eco Park, the river which was with the brackish water river, it was like too, it had too much plastic and it was not clear. The water was too in grey color. There was grey water invasion. There was also drainage water. Everything was mixed. There was also plastics. And during festivals, people dump all these things inside the water. So that was another reason. And in 2015, there was study comparing that with Pulicat. This was study in the Pulicat. Comparing to that, again we had less diversity. There was no much bird species present now currently, and I was surprised to see the birds were not scared of humans. If here in Eco Park, if we go near the bird used to fly. Like the bird, it was not used to human settlements. But in Pulicat, they're all near, they're all fishing. These birds have they've adapted to human settlement. So there's no change in the diversity because of human settlement. But in Adyar Eco Park, there are no humans. But since that, the birds are able to be there on their own. But still, there is less diversity. That is because the size of the area, the Pulicat is vast and Adyar Eco Park is a smaller area when compared with the size. So that's the reason why there is less diversity. That was my conclusion.
Questions from the audience?
Because actually, if you're not able to do the night, actually we have to observe at the night after in the later hours of the evening. Yeah, 6 o'clock was the average time. It started around 5 or 5:30, some like that. So, yeah. Sure, I'll follow that. Thank you. One part of my study was water quality analysis. The basic, actually, that was the diversity. I wanted to do the indices. Since the duration was short, I had to add up the parameters. That's the reason why I couldn't. But I am continuing the project, so definitely I will give you the result. Thank you, ma'am.
The next presenter is Ishwaran Rangasamy.
Good evening to all. Today I am trying to, my presentation topic is water contamination, tea garden area, a case study in selected tea plantation from southern Western Ghats, Tamil Nadu, India. My introduction, water is the most important constituent of all animals, plants and other organisms as well as crucial to the survival of mankind in the biosphere. Due to rapid industrialization, urbanization and modern agricultural activities, most of our natural water bodies are gradually degrading. In many fast-growing cities, heavy metal contamination in rivers is a major issue because of maintenance of water quality and sanitation infrastructure did not increase along with population and urbanization growth, especially for the developing countries. The report concludes that both the developing countries and developed countries are polluted physically, chemically, and biologically. Water quality is adversely affected by the intensification of agricultural activities with increased use of fertilizer and pesticide. According to our literature review, there has been no published data, physiochemical and heavy metal contaminants in selected water bodies of Nilgiris and Valpara division, Coimbatore district. That's why we should be concerned about studying the water quality of tea garden in this area. My objective is to find out the physiochemical parameter level analysis of tea plantation associated water bodies, to study the heavy metal level of adjacent water bodies, to find out the pollution level in selected tea plantation of Nilgiris and Valpara from southern Western Ghats, to find out the total pollution level in selected tea plantations in pre-monsoon season. My study area, Valpara, Kunnur, Kunda, Kodagiri, Udagai, Gurulur. Why I am selecting this study area? They are producing tea short period high yielding, and because they are using a high level of pesticide and fertilizer tea plantation area, that only I am selected this sampling site. So water samples were collected from selected tea plantation during pre-monsoon period by morning between 8 and 11 a.m. systematically following the appropriate methodology. Water samples are manually collected. The collected water sample directly transferred to the previously acid was amber polythene bottles. Oxygen will be fixed in the sampling site itself by introducing the 1 ml of MnSO4 and alkali oxide iodide solution, find out the DO level. This is the shape of my sampling method. Triangle shape used. Sampling location showing the longitude, latitude and elevation source of water. Materials and methods used. AFA, standard methods for use for the study, and water quality analysis laboratory methods near used. My statistical analysis used for IBM SPSS statistics. These are the analysis of parameter pH, alkalinity, COD, DO, hardness, heavy metal, fluoride, nitrate, phosphate, sulfate. So the 23 parameters are used for the study. My result and discussion, the average result of the physiochemical parameter of water sample under heavy metal sample average study. Some physiochemical concentrations nitrate is very high, especially COD and total hardness nitrate. COD in Kunda and Kunnur is high, and total hardness also Kunda, Kunnur and Kudlur is high. Nitrate only Kudlur is high. This is other parameter. The physiochemical parameter, that's showing the picture for some heavy metals are also high cadmium, iron and manganese. The cadmium showing all sides high, and iron also is category only showing the high, and manganese only showing the high in the Ooty. These are the heavy metal concentration levels sampling site. Conclusion: according to this study, heavy metal like cadmium, iron, and manganese were present high range of WHO limit in the selected stream water in this area. The pH value shows slightly alkaline to acidic nature, Kunnur, Kunda, Kotagiri compared to the other sampling site. But it is within this specified range. The total hardness very high except Kunnur, but calcium, magnesium are very harmful for drinking. Chloride and sulfate concentrations were found to be lower than the WHO tolerant limit for drinking water sources. There needs to be prominent attention given to the water sources iron level. In the stream water system, the good water quality help to the development primary and secondary producer and survival of plankton, flora, fauna, fishes, etc. There is an urgent need to conserve the solution of life to reduce the pressure on the stream. To ensure the safety of the streams, a strong legislation clause necessary with tight vigilance and people contributing to effort towards this study. So overall, the observe this study. Observe this study. Some physiochemical parameter and the chemical parameter are also increase the salinity of in water bodies. Yes, sir.
Any questions from the audience?
What you have checked all this in tea plantations, near tea plantations, I understand. Right? Tea plantations. Does the caffeine or whatever polyphenols in the tea has an effect on these heavy metals or whatever you checked?
Yes.
Does this have any correlation, or why did you choose tea plantation or stream near tea plantations?
Only there is high level of pesticide and fertilizer tea plantation. So.
I don't get you, sir.
Okay.
You mean to say that tea plantation has an effect on the heavy metal or the physico-chemical properties of the stream, or how is that? Yes?
Yes, ma'am.
But tea is not planted, they can be. Yes. What is the source of cadmium?
So fertilizer also highly used.
The question is different areas, but you have mentioned the word tea plantation, and your whole analysis is in the stream or physico-chemical and heavy metal population of the stream waters.
Yes.
I'm just asking, what is the correlation between tea plantation and your results or whatever you have done?
My tea plantation associated water bodies. So they're using chemicals.
Does it have an impact?
Yes, ma'am.
Yes.
The last presenter will be Gideon Moses.
Good evening to one and all. My research paper entitled as PCR-based detection of biocontrol genes in plant growth promoting bacteria. As we all know, our country is a land of agriculture. Every year we are producing, consuming, export several tons of agriculture products. But major constraints for the cultivation of crops are the diseases caused by various phytopathogens. Majority of our farmers are using chemical fungicides, but they are detrimental and have an adverse effect over crops, human, and also environment. Therefore, biological control of plant pathogen is the best alternative approach and an environmentally sound, viable, and sustainable crop protecting management. Among the various biocontrol agents, the species belonging to Bacillus pseudomonas have been exploited vastly, rapidly, and proved as successful biocontrol agents because they produce a variety of antibiotics such as bacillomycin, eturine, surfactant, mycosubtilin, fungicin, etc. with broad spectrum of antifungal activity and activate plant defense mechanism by triggering induced systemic resistance. Therefore, the detection of genes in plant growth promoting bacteria encode for the production of antibiotics enable us to comprehend the molecular mechanisms involved in the biocontrol of plant diseases. In the present study, we have isolated more than 100 bacterial strains from rhizosphere and philosphere bacteria and screened for their antagonistic activity against various phytopathogens. The bacteria which showed promising antagonistic activity and plant growth promotion were selected for further studies. The genomic DNA of selected plant growth promoting bacteria was isolated and amplified using bacterial universal primers. The bacterial sequences were deposited in NCB GenBank database and the accession numbers were obtained and presented in this table. The genomic DNA of selected plant growth promoting bacteria were used for the detection of biocontrol genes such as 2,4-diacetyl fluoroglucinol DAPG, pyrrolonitrin PRN, volatile compound HCN, bacillomycin, fengicin, and ytterine. The primers and PCR amplification conditions are given in this table. The gene-specific primers DAPG gene amplified the expected 745 base pair band in the DNA of Pseudomonas aeruginosa MML 2519 with the positive control Pseudomonas proteasin CHA0. The amplified PCR product showed 719 base pair band in the DNA of Pseudomonas aeruginosa MML 2515. The gene-specific primer HCN showed 570 base pair band in the DNA of Pseudomonas aeruginosa MML 2424. The amplified PCR product showed 875 base pair band corresponding to bacillomycin gene in the DNA of Bacillus thuringiensis MML 5816, Bacillus vallis mortis MML 5820, Bacillus velicensis MML 5745. The amplified PCR product showed 964 base pair band corresponding to fengicin gene in the DNA of Bacillus thuringiensis MML 5816, Bacillus siamensis MML 5729, and Bacillus ameloleukophaciens MML 5769. Similarly, the amplified PCR product showed 647 base pair band in the DNA of Bacillus subtilis MML 5809, Bacillus vallis mortis MML 5820, Bacillus velicensis MML 5745. Presently, we are sequencing the biosynthetic gene and the sequences are yet to be submitted in the NCBI GenBank database. Thank you.
Could you please connect me to this biocontrol genes? All you have talked about bacillomycin and could you please go back to that NCBA submission slide, please? All these are submitted, sir.
Yes.
You took which one as control for Pseudomonas?
Pseudomonas proteogens SAR0 as a positive control.
That served as the positive control.
Yes, ma'am.
With 1 kb ladder and DAPG primers, all these was in range.
Yes, ma'am.
Okay, sir. And what are you about to submit? Finally, for the last thing.
Bacillomycin fengicin. Currently, we are sequencing the biosynthetic genes.
Okay. How have you zeroed down on bacillomycin?
Pardon?
How have you zeroed down on bacillomycin alone? Fengicin. Bacillomycin you are finally working on, right?
Yeah.
How have you come to a conclusion that you're working on bacillomycin? Does
We have extracted
yes.
the antifungal compound bacillomycin. We have proved the
Antifungal, sir?
Yes, ma'am.
Antifungal, okay.
Antifungal. Because the antifungal metabolites produced by the bacillomycin ameloleukophaciens bacillus subtilis, we have proved their antifungal activity in well diffusion assay against various phytopathogens.
You worked on well diffusion assay for fungal plates, sir?
Yes, ma'am.
Bacillomycin. And so it showed higher activity,
Yes,
so you...
against various phytopathogens.
Okay, okay, sir.
Thank you.
I request the chair, Dr. Raza Sharif, to consolidate the session.
Assalamu alaikum and a very good evening.
I would first like to thank the organizers, and more than that, I'm very honored and happy to see two of my teachers as well as mentors, Dr. Sultan Ahmad Ismail, who doesn't require any introduction at all. Presently is the member State Planning Commission. Then Dr. Dawood Sharif, who was also my teacher as well as mentor. JBAS, always I appreciate as well as feel really happy in the sense that till some time back, they were the highest in the NAC grade. I think the number one college in the city with the NAC grade. Of course, now probably Guru Nanak has got slightly higher grades, okay, scores. Yeah, that's right. Okay, there's a correction by the organizing secretary that was for the third. In fact, new college is also, we are just awaiting the NAC fourth cycle, and we hope to get A++, inshallah. Okay. Coming to this paper presentation, already I think we are running short of time, and fortunately we had only 4 participants among the 6 listed, and my co-chair, madam, was also mentioning that there's a slight mismatch here. We had 2 participants who represent the PG course, and 2 who are research scholars. Okay. So that is evident in the work that was presented, because mainly it is because of the tenure. As a research scholar, you have more ample time to complete your work. Okay. And one thing I would, it's not a complaint, probably there is no binding theme for this particular session. Okay. Usually in most of the paper presentations, there is a specific theme, but here we had potpourri or miscellaneous. Probably it is because of the number of, people who wanted to participate. Okay. And I was also quite happy to listen to Dr. Anish. So we also had him as a resource person when he was representing Biozone some time back. Okay. Once again, I would like to thank everyone for giving us this opportunity on behalf of the chairperson as well as the rapporteur. And I wish the organizers as well as JBA's college all the best, and I'm sure they will continue to strive on the path of excellence. Already they have got the mark, or the, what do you call, the ranking. Plus they've also got that College of Excellence from the University of Madras, and so many other commendations from UGC, as well as I think even DBT, Star College, which is also a rare thing, because many are only in the first stage. Okay. In fact, there's no harm in mentioning that we also tried for that DBT Star College status, but somehow we were not successful initially. Okay. So once again, I would like to thank everyone for your patient listening and as well as for your presence. Thank you.
Assalamu alaikum and very good evening to all once again. I express my sincere thanks to the chairperson, Dr. Azhar Sharif, co-chair, Dr. Jayanti, and rapporteur, Dr. Sumita, for the smooth conduct of this session. Now I request Mrs. Parveen, Assistant Professor of Zoology, JBS College for Women, to hand over the memento as a token of appreciation to the panel members. Dr. Jayanti. And the rapporteur, Dr. Sumita. Thank you, ma'am. Thank you all once again.
Good evening. Very sorry to make you wait. Our principal is on the way. In just a couple of minutes, we'll start with the valedictory address. Thank you.
I request the dignitaries to come up on the dais.
Assalamu alaikum and a very good afternoon to all. We begin the valedictory function with a prayer. I now request Ms. Rifat Aisha of first year BSc Zoology to recite a few verses from the Holy Quran and render its translation.
Assalamu alaikum wa rahmatullahi wa barakatuhu. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم والله أنزل من السماء ماء فأحيا به الأرض بعد موتها إن في ذلك لآية لقوم يسمعون وإن لكم في الأنعام لعبرة نسقيكم مما في بطونه من بين فرث ودم لبنا خالصا سائغا للشاربين ومن ثمرة النخيل والأعناب تتخذون منه سكرا ورزقا حسنا إن في ذلك لآية لقوم يعقلون. Translation of Surah An-Nahl, which means the bee. Chapter number 16 of the Holy Quran, verse number 65 to 67. I seek refuge in Allah from Satan, the rejected. In the name of Allah, the most beneficent, the most merciful. And Allah has sent down rain from the sky and given life thereby to the earth after its lifelessness. Indeed, in that is a sign for a people who listen. And indeed, for you in grazing livestock is a lesson. We give you drink from what is in their bellies, between excretion and blood, pure milk palatable to drinkers. And from the fruits of the palm trees and grapevines, you take intoxicant and good provision. Indeed, in that is a sign for a people who reason. Jazakallahu khair.
Thank you, Rifat Aisha. I now invite Dr. Anuradha, Head of the Department in charge of PG and Research Department of Zoology, to deliver the welcome address.
A pleasant evening to one and all present here. On behalf of the management, staff and students of the Postgraduate and Research Department of Zoology, I deem it a privilege to render the welcome address to the valedictory program of the two-day International Conference on Frontiers of Biosciences in Sustainable Development. At the outset, I am immensely pleased to welcome our guest of honor for the valedictory program, Dr. B.C. Archana Kalyani, IFS, who is here to render the valedictory address. I also extend a very warm welcome to our principal, Dr. Amtul Aziz, our Controller of Examinations, Dr. Mercy Rajsekhar, our Financial Consultant, Mr. Selvam, our Vice Principal, Dr. Shaira Banu, our Accounts Officer, Mrs. Matina Fasi, and our Vice Principal for the Evening College, Dr. Firdaus Jahan, and the professors, scientists, and learned academicians, research scholars, and students of various colleges, and also to the student fraternity of our own college. A very special welcome to our mentor and guides, Dr. Sultan Ahmad Ismail sir, and also to Dr. Dawood Sharif for all their mentoring and the advice to revise and reform ourselves at all times. Now to introduce the guest speaker for valedictory function, Dr. B.C. Archana Kalyani. Dr. Archana Kalyani received her UG degree in botany from the Stella Maris College, PG degree from the Madras Christian College, and also a second PG degree in forestry from Dehradun Ministry of Environment and Forests, and the doctorate degree from CAS Botany, University of Madras. She also has got a diploma in computer applications from Biotech Institute, Chennai, and has completed a short-term course in GIS and remote sensing for natural resource management. She has also worked in various capacities as specialist, agroforestry in Tamil Nadu, watershed development agency, and also a deputy conservator of forests, deputy director in Aringarana Zoological Park, director tribal welfare, and district forest officer, deputy conservator of forests, and head of division land use in State Planning Commission, the conservator of forests, come additional project director of integrated coastal zone management program in the Department of Environment and Climate Change, and also a special secretary of the Department of Environment and Climate Change. She has also received various awards, to name a few: awards of books for academic proficiency throughout her UG program, and gold medal and silver medal at Stella Maris for her outstanding performance, and also KAK Ganeshan Award for proficiency in PG course at Madras Christian College, and gold and silver medal for being the first and for being the best all-rounder training officer, and also received prizes and certificates in memory of late S.P. Srinivas IFS and R.C. Kaushik Prize for proficiency in soil conservation and land management during her PG in forestry at Dehradun. She also has 3 book publications to her credit and 2 handbook publications. Her other interests include sports. She also has interest in instrumental music. She has also won prizes and shields for painting, singing, Villupattu, flower arrangements, soap carving, and won prizes in sports such as javelin, discus, shot put, carom, shuttle, table tennis, cycling, and running. I once again welcome Dr. Archana and all the other dignitaries for the valedictory function. Thank you.
Thank you, ma'am. I now request Dr. V. Anuradha, Head of the Department in charge, to welcome the guest of honor, Dr. Archana Kalyani, with a box of dry fruits. Thank you, ma'am. May I now invite the organizing secretary, Dr. D. Mubin Sultana, to present the report of the conference.
Assalamu alaikum and a very good evening to one and all. I'll keep the report as short as possible due to the paucity of time.
And it's a privilege to present the report of the 2 days international conference on frontiers of biosciences in sustainable development. The event started with the inaugural function, which was followed by 5 plenary lectures, one from Ireland and one from Singapore, which was on the virtual mode, and 3 from offline mode, for which one speaker was from Kerala, one speaker from Hyderabad, and one from Tanvas, Chennai. Followed by, we had 10 paper presentation sessions, out of which 7 were offline, 3 were online. Totally, there were 99 papers which were published in the abstracts, but we had presentations out of 81, 73 paper oral presentations, and out of 80, 2 were not presented, posters. 16 posters were presented. And out of the paper presentations, almost 18 city colleges had participated. And as far as other states are concerned, we had participation from Uttar Pradesh, West Bengal, Bihar, Pune, Hyderabad, Kerala, and Mumbai also. So the success of a conference is based on the participants, and I thank all the participants, both offline and online, to make this program a grand success. Thank you so much.
Thank you, Dr. Mubin Sultana. May I now take the pleasure of inviting Dr. Archana Kalyani, the guest of honor for today, IFS officer, Government of Tamil Nadu, to give the valedictory address.
Good evening, one and all. I'm sorry, I have a very bad throat. The topic of this seminar was sustainable development. All of you know what is sustainability. What is sustainability? Something which can be maintained over a period of time. So, what are we going to sustain in this world? What do you want to be sustained? And what is sustainable development? So, everything is anthropocentric. Only humans, like we want everything to be sustainable because we have done all damage. So we want something to be left for the future. So whoever has done the damage has to do the repair. So as human beings, we are responsible to actually patch up the damage which is already done. Now, when we were quite young, the air was quite pure, there was no pollution. The water was pure, no pollution. We used to directly take water from the well and drink. Now we have packaged drinking waters. There was a lot of immunity. Right now, I myself don't have immunity. That's why we are having cough, cold, fever. So, some 30 to 40 years before, the life was something very different. We were not behind things. We were not materialistic. We were nature friendly. Children never used to, don't take it otherwise, but I'm just telling. Children never used to keep on studying. Now it is always, study, study, study. It is a mad rat race. I still remember, when they were just reading my CV, people must have thought like I have got so many things. It's because, I used to go and sit and cry with my mother. I used to tell my mother, Mummy, why are you not asking me to study? Please tell me to study. Then my mother used to say, you will study, you go and play. I'll say, no, I want to study. You ask questions. But now it's the other way around. I keep asking my son, did you study? He'll say, I want to go and play. So it's all something different. We were so free, we used to play. We never had so many subjects. And mark was never a matter. My brother used to tell, even if you get 40, it's a pass. Even if you get 90, it's a pass. Pass is a pass. But now today, as a parent, what we are trying to do, we are asking the children, you should get first, you should get 90, you should become a doctor, you should become an engineer. So we are just thrusting. Nothing was thrusted on us when we were young. So at least the future generation should know we should not trust anything. We should live with the nature, enjoy the nature. Now coming again to sustainability, what do you want to sustain? You have to sustain the environment. What is environment made up of? It's made up of the trees, animals, flora, fauna, everything put together is only environment. We have already polluted water, We have polluted the air, we have polluted the soil, polluted land, everything. And now we are searching for a solution. How to reclaim water, land, air. The research is nothing but researching. Nature has solution for every problem. When you turn around, sit down and turn around, you will know what is the answer for that particular problem. The solution is already there. Now we are only researching for the solution. I'll just give you one very good example. Everybody will know. One is COVID. What did we do during the COVID? Everybody was back home. Nobody went outside. We were all confined. What happened? A lot of studies were there. What did they say? The ozone layer has patched up itself. Whatever damage we did, it got patched up. And there were a lot of pictures coming in TV. You saw like penguins were out, all birds were out, and then bear, lion, tiger, everything was on the street. So they were all very happy. None of the humans on the road, only animals on the road. They were too happy to roam around. We used to say that sparrows have gone, but during the COVID period, a lot of sparrows were there. I used to see sparrows, kingfishers near my house. So, left to nature, it can do everything it wants. It has got the totipotency to bring back the life. But beyond a particular thing, when we damage it, it cannot bring back life. So now we are at that stage. At least now we should realize that we should not cause more damage. There is actually a saying, nature has everything for your need, but not for your greed. Let's not be greedy. Whatever need is there, utilize it for that, and please leave the rest for the others. Please be more humane and considerate towards your fellow beings. There's nothing we are going to take. Our lifespan, it's just, what's the average Indian lifespan? 68, 70 maximum. But you just realize, we will realize one thing, like when something is with us, we will never realize its potential, or we'll never realize its, the greatness of it. But prior to COVID, if you see, in Delhi, there was a lot of pollution. And in fact, Supreme Court gave an order saying that like most of the schools should be shut down because the air is not congenial for the school children. So the schools were shut, and a lot of oxygen parlors were mushrooming there. And they had all types of oxygen: medicated oxygen, scented oxygen, normal oxygen. It is like when you go to a dosa corner, they used to have a variety of dosas. There was a variety of oxygen parlors. And you know what they charge for every 15 minutes? It was 299 rupees. Now imagine the lifespan of an average Indian is around 68 to 70 years. So if you're going to pay 300 rupees for 15 minutes, in your lifetime, you are inhaling oxygen from this atmosphere worth 73 crores. And that is not the only thing which the plant is giving you. It is cleaning up the air. Whatever carbon dioxide, now we are talking about carbon sequestration. All these carbon sequestration can be done only by the trees. And mind it, you can have oxygen producing factories, but the biggest oxygen producing factory is none other than the trees, which is free of cost. If you just leave a plot of area unattended, don't do anything, just close it, don't go there. After some time, you will see a lot of grasses coming up, then there will be shrubs, then there will be trees, then there will be birds, a lot of microorganisms. The whole area will be start bubbling with life. But once you start putting garbage into that particular area, you have doomed everything. Now you tell me, what are we supposed to do for this Mother Earth? Let's stop putting plastic. Let us be more prudent in using vehicles. I don't mean to say you should not use vehicles. Let's be more prudent. Wherever you can walk, please walk. Now everybody has been glued to cell phones, WhatsApps. We don't have time to talk even with our family members. Gone are the days when we used to sit together and have food. Dinner time is gone. There is no time schedule. You can take dinner at 11 o'clock. You can order for a pizza. You can go for Zomato, Swiggy. Of course, it gives livelihood opportunity for many. But please sit together. Have a small prayer at home. And try to talk with your parents. Talk with your siblings. So that's the time where, you get a lot of solutions also. So what we can do is, please reduce. This is also for sustainability of your life. If you want to increase your lifespan, please reduce on using your cell phones. Be more prudent in using water. Please don't waste food. There is somebody more needy who needs this food. Don't take anything beyond what you can. You cannot gulp things and then vomit. Nature can always repair itself. Leave the nature to itself. No dog or cat will go to a doctor for getting a fracture repaired or whatever it is. But for everything, we go to the doctor. Nature has everything. See, we used to have tulasi around us, omavalli. These were all for your throat infection. And tulasi is the one which produces oxygen throughout the day. And there used to be a people tree, arasamaram. Every panchayat used to have one people tree where people used to sit, congregate together. They will discuss all problems. And people tree again is the one which produces more oxygen. It can produce oxygen even during night time. Normally they say tree doesn't produce oxygen during. This produces oxygen, but not to a greater extent, but it produces oxygen. This is one which produces. Similarly, bamboos. So try to plant more trees around your house wherever possible. Everything has become a concrete jungle. Man doesn't, he's not satisfied with just one house. He wants to have many houses. There used to be a time when we used to cuddle together, all our, we used to put our leg on our parents and sleep. There never used to be a separate room for us. 5, 6 members used to sleep together in the same house. Now everybody wants a single room. 4-bedded flat, 5-bedded flat, and even for having dinner, somebody should make a WhatsApp call. I am in bedroom number 1. Let me come to the dining hall. Is this life? No. One house is enough, right? Let us not build houses for our kids. Let's build trees for our kids. Let them enjoy the nature. Concrete jungles are not the need of the hour. Nature is the need of the hour. Think of all those catastrophes we have come across. We talk about climate change. What is the reason for climate change? It is our own thing. It has caused this disaster. When you have something which is not suitable for your health, what happens? You start vomiting. Correct? Similarly, I feel tsunami, though you can say all theoretical things that tectonic plate movement and earthquakes, these are the results for tsunami. But what I think personally, whatever garbage you have thrown into the sea, it is getting vomited out on you as tsunami. And there used to be floods. What do you do normally when your house is not clean? You start mopping. You pour water and clean. Similarly, Mother Earth wants to clean this whole area. So that's why there is heavy rain. Now we keep telling there is rain which is unpredicted and there is heavy rain which is coming. There's nothing like that. If you look into the IMD details, because I just wanted to check and I was just checking. If you had heard the saying, like in Sangam literature, when somebody comes and visits the king, the king you'd ask, Madam Mummari Peidada. What is the meaning of Mari? Mari means rain. Madam is month. Mummari means 3 times. In a month, 3 times has it rained, which means per year there is 36 rainy days. Today we have 52 rainy days in a year, which is not something alarming. But the thing is, it used to rain on a consistent basis. Every month there used to be 3 rains. But now, on a single day, everything is poured. So what happens? You can't, Avayar used to tell, I don't know how many of you have heard of it. You can't have everything on the same day. You can't go hungry on a particular day. So how am I to stay with this body? ஒரு நாள் உணவை ஒழி என்றால் ஒழியாய், இருநாள் உணவை ஏழென்றாய் ஏழாய், இடும்பை கூறும் என் வயிறே உன்னோடு வாழ்வது அரிது அப்படின்னு சொல்லி. There is actually a saying. So similarly, if rain is going to be on a particular day and that is heavy and there is no capacity to hold it. Why there is no capacity to hold it? Because we have built buildings on all sustainable wetlands, on all water bodies. How many houses do we need? After all, like I used to keep saying, like as a human being, for us, after our death, even 6/2 is not there for us because they burn us and then they will put the ash into the river. So nothing is there. The kathoda kalandu pogudhu. We are just entering into the air. What do we take with us? So why should we have all this with us right now? So please leave something for your future generation. All you can do is, if you're quiet, without doing any harm, that itself will regenerate the nature. Please avoid using plastics. Please avoid throwing all your waste into the land. Be more prudent in using electricity, more prudent in taking food. These are the sustainable ways by which you can sustain yourself, sustain your future generation. Over and above, please don't forget God, because people keep saying whether God is there or not, but there is some supreme power beyond our control. We may think that we are the best. We are not the best. There is someone supreme. Maybe it is nature or it is God. It can be by any name. So try to have some prayer every day. Try to spend time with your family every day. That gives you a lot of encouragement. It gives you a lot of strength, and you will not break down at any point of time. See, if I've crossed so many hurdles in my life and I'm still quite brave in taking decisions, it's because of my mother. And incidentally, she's an old student of SIET. She was in the first batch of home science. And when I told her I'm going to her college, she was very happy and she remembered her principal, Rajam. Madam Rajam was her principal. Then she started talking about her Tamil teacher, Susila Madam. I was just taken aback. She said some 40 years before she studied here, she still remembers all her teachers' name. No, but how many of us are going to remember our teacher's name? We don't even remember, we don't even know who is there in our neighbor. We shut our door and there ends the matter. Try to mingle. Listen to the problem of others. So once again, though this is actually apart from what I'm talking about the last 2 days seminar. So I think this is the message we need to carry. Research is nothing but searching for the solutions which are already available in the nature. Sit for some time, meditate, you will get the answer. Please leave a better home for the future generation. Don't build houses, please plant trees. Leave trees for your future generation. And a well-grown tree will be able to take care, that is at least 15 to 20-year-old tree will be able to produce oxygen, which is keeping 4 members in a family to be alive. So that's one thing which we can do. So my humble request is, let's be more satisfied with what we are having. Sustainability should begin with us. Charity begin with us. Sustainability should begin with us. I take this opportunity to thank SIT College who has given me this opportunity to speak in front of you. My sincere thanks to Professor Ismail, who's always been my guiding star. I always envy sir how he is able to fit himself in every possible way. And he used to tell, he used to tell me how do you know whether a soil is fertile or not? How do you know whether a soil is fertile or not? He used to say, you just dig the soil and see. If there are any earthworms coming out of it, then that's the best soil. And you need not go and give the soil for testing. All you need to do is, you have to take a handful of soil, pour little water, and then try to make, whether it is, if it becomes like a mold or it is just dropping. So these things will make you understand whether the soil is healthy or not. So he is actually Manpudu Tata. I actually, I cannot talk much like him, but whatever I felt I should tell you, I thought I will let you know. My humble request again and again is, please leave this place as a better place for your future generation. Thank you.
Thank you so much, ma'am, for your wonderful talk. It was quite enlightening and inspiring to all of us. Before we move on with the prize distribution, which I guess every one of us are very eagerly awaiting for, We would quickly like to have a feedback from two participants from other states. If you're right there in the auditorium, can quickly come up and just share your views about this 2-day conference that you have attended. Just maybe a minute for each one of the participants. Please introduce yourself and then you can give us anybody who is interested and come up, especially from other states. If not, from anywhere in Tamil Nadu, you can come up on stage, introduce yourself and just give us a short feedback about your experience of having attended this 2-day international conference.
Good evening, everyone. It's been a great privilege and honor to be a part of this commendable 2 days international conference on frontiers of bioscience and sustainable development. Actually, this is a real good, real-time experience of attending this big conference with all the renowned speakers and scientists and the budding scientists who are sitting over there. Actually, the program was indeed very informative and well organized, I could say. The hospitality part, no words to explain. It was, I mean, I mean, those who have experience will explain it. Yes, yeah. I'm Kavita Devanayagam, PhD research scholar, Queen Mary's College, Chennai. When we just entered this premises, it was like, I don't know how to explain. And this is my first presentation. Maybe you all will feel that everybody was calling me as a ma'am. They were not able to distinguish whether I'm a ma'am or a student. In fact, after a 25 years of break, I mean academic this thing, I was in a position to take care of my children. As Sarah said, all our studies will ended up with our marriage and children. Yes, even I experienced the same. I did my MPhil in the year of 1998, long time back. After 25 years, when I put my son in the college, I just entered again into this academic field. I'm really, thank you so much. Thank you. Um, Today's younger generation, after all, definitely they are children to me. Please, there is no age limit for studies. Okay. Women can do anything at any point. So many were questioning me, ma'am, I can talk. So many, when I entered the research field, all the students, I'm not blaming them, but they were with a question in their eyes. Why are you here? What are you going to do? But I just asked them, what are you doing here? I'm doing the same. See, when a woman being educated with my MPhil education, I was in a teaching profession. Few years I was as a guest lecturer. I really enjoy teaching children. As Sir mentioned, teaching is the only profession where there is no jealous. When I really see my student as a doctor or something, I really just love them, rather seeing my son being in a great position. All the students are our children. So I want to educate much more. I entered in this field and I answered, let me be an inspiration, a small inspiration for many younger generation. We can do whatever at whatever age you can. Since you are in the younger age, try to achieve whatever possible. Thank you. Good luck for all the scientists and the prominent speakers over here. Thank you very much for this opportunity given to me. Thank you.
Thank you so much for that excellent feedback. Do we have one more person? If you can make it short and crisp because we are running short of time. I do understand all of you want to share your experiences, but I think that you should have written it down in your feedback forms. Thank you so much for that. Okay, if we have no more presenters, we'll just move on. Before we go on with the prize distribution, we would like to acknowledge the services of the judges of the paper presentation and the poster presentation. To honor them, we request our guest of honor, Dr. Archana Kalyani ma'am, IFS officer, to hand over a memento and a certificate of appreciation to Dr. Anandi Rachel Livingstone, Assistant Professor, Department of Zoology, Madras Christian College, who was one of the judges for the paper presentation. Okay, I guess she has left. We would also like to acknowledge the services rendered by Dr. Sumaira Rafiq as a judge for the paper presentation, Associate Professor, Department of Microbiology from Justice Bashir Ahmad Said College for Women. We would also like to appreciate the services rendered by Dr. Nalini, who is not present right now. She did the online poster presentation judgment. She is Assistant Professor, PG and Research Department of Zoology from Pumbuhar College. And the second judge for the poster presentation, which was online, was Lakshmi, Dr. Lakshmi, Professor and Head of the Department, Department of Pharmacology, Savita Dental College. Can we have a big round of applause for them? They have left for their work. Thank you so much. Now we come to the most exciting part of the conference, and that is the prize distribution. We have 3 different categories under which the prizes have been awarded for the best paper presentation, the PG and the UG category, the research scholars category, and the faculty members category. So let's begin with the students, because whatever we do is for the students, and we hope and pray that these students will come up to become great research scholars and scientists. So big round of applause to the entire participants. It is because of you we are able to do this, and we hope this outcome of this conference is going to make you do much more research in bigger areas to come. To start off with the PG and UG category, I'm very happy to say that the 3rd place goes to Atulya from Department of Zoology, TK Madhava Memorial College, Kerala. She also gets a cash award. I request the participant to please come up on stage. Okay, Dr. Anish is taking it on her behalf. The second prize goes to Sakshi Sandeep Raipalli, Department of Life Science, Aston from United Kingdom. It was online, so we would like to acknowledge his paper presentation, and I think the organizing secretary can take it on their behalf. And the first place under the UG and PG category is backed by Nafisa Syed Akhtar, School of Medical and Veterinary and Life Sciences, University of Glasgow, again from United Kingdom. So a big round of applause, because we had paper presentations both online and offline. Now moving on to the second category, which was of research scholars. Let's begin with the 3rd place. The 3rd place goes to G.I. Darul Rayyan, Department of Biotechnology, College of Science and Humanities from SRM University. The second prize is backed by Anupama Prakash, Department of Environmental Sciences, Bharatiyar University, Coimbatore. Come on, students, give them a big round of applause. The first place is backed by Mohammed Tamim, Department of Environmental Science. The first place is backed by Mohammed Tamim, Department of Environmental Science, Bharatiyar University, Coimbatore. Hearty congratulations to the winners. Now we move on to the last category, which is the category for the faculty members. The 3rd place goes to Sunila Kumari, Assistant Professor, Department of Zoology, University of Hyderabad. Dr. Padmavati is receiving it on behalf of Dr. Sunila Kumari. The second place goes to Dr. Sarasa R., Assistant Professor, Department of Zoology, Koidamillat College, Chennai. Okay, the first place goes to Seena KK, Assistant Professor, Department of Botany, Mercy College, Palakkad. Online. So anyway, hearty congratulations to all the prize winners and thank you so much, ma'am. Thank you all. So we now come to the end of this valedictory function. May I now request Dr. Hajira Banu, Assistant Professor, PG and Research Department of Zoology, to propose the vote of thanks. Before that, I would request Madam Principal, Dr. Amtul Aziz, and Head of the Department of PG and Research Department of Zoology, to give a memento to our guest of honor, Dr. Archana Kalyani, IFS, as a mark of our respect, love, and appreciation. Thank you, ma'am. Thank you all. I now invite Dr. Hajira for the vote of thanks.
Assalamu alaikum and a very good evening to one and all present here. On behalf of the management, principal, faculty and students of the PG and Research Department of Zoology, I am privileged and honored to get the opportunity to propose the vote of thanks on this special occasion of the two-day international conference on Frontiers of Biosciences in Sustainable Development. First and foremost, I thank the Almighty for the benevolent grace and mercy for the smooth conduct and completion of the 2 days international conference successfully. I would like to thank the guest of honor, Dr. B.C. Archana Kalyani, IFS, Chennai, Government of Tamil Nadu, for accepting our invitation in spite of her busy schedule and for giving such an enlightening lecture which has been engraved in all our minds. My heartfelt thanks to our principal and head of the PG and Research Department of Zoology, Dr. Abdul Aziz, for her continuous support and encouragement at various stages that radiated a source of energy to every one of us. My thanks are due to the Vice Principal of forenoon session, Dr. Shahira Banu, and afternoon session, Dr. Firdaus Jahan, for their gracious presence. I owe my thanks to Controller of Examination, Dr. Mercy Rajesh Shekhar, for her gracious presence. My sincere thanks to Dr. Anuradha, HOD in charge, for her support, guidance and cooperation rendered throughout the conference. I am very much thankful to Dr. Kanis Fatima, Assistant HOD, for her untiring help, support, motivation, which was a great source of inspiration. My wholehearted thanks and gratitude to the organizing secretary, Dr. Mubin Sultana, for organizing the international conference and bringing all eminent speakers from all over the world, both offline and online, from various fields under one roof to share their expertise and knowledge. I would like to thank all the honorable dignitaries for gracing the occasion with their presence. My sincere thanks to all the invited speakers of all the sessions for their acceptance to deliver enlightening lectures and chair, co-chair and rapporteur for the successful conduct of the sessions. I also thank all the judges of paper presentation and poster presentation, both offline and online. I thank all the paper presenters for their active and enthusiastic participation. An event of this dimension cannot happen overnight. The wheels started rolling months in advance. I am very much thankful all the faculty of PG and Research Department of Zoology for their untiring work rendered and to the motivated, dedicated research scholars, volunteers, students and non-teaching staff for their involvement and support. My sincere thanks to all the participants from JBS College and also from other colleges and organizations for their active participation and interaction. I would like to thank Mrs. Matina Fasi, Administrative Officer, for her support. I am thankful to the technical assistants, Mr. Sudalai and Mr. Sardar, for the technical support. Without them, this event was not possible. I thank each and everyone for making this 2 days international conference a great success. Thank you all once again.
At this juncture, my sincere apologies as I have forgotten to mention the prize winners of the poster competition. So I would request the poster prize winners to come up on stage. The third prize goes to J. Nushrat Fatima, research scholar from Department of Zoology, JBS College for Women. The second prize goes to Kritika N.S., undergraduate student from Department of Advanced Zoology and Biotechnology, Stella Maris College. And the first place is bagged by Shadan Haider, Department of Zoology, Patliputra University, Patna, Bihar. Hearty congratulations, and we are really sorry about this because I missed it. Thank you all. Hearty congratulations to all the prize winners. Now we conclude the session. We request all of you to please rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जाहे तव शुभ आशीष माहे गाहे तव जय गाता जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
I request all the participants to please have your tea before you leave.